nav GUI up to right, Tom? The little webpage one behind your NC repeater. You'll hit straps off on there. Yep. Cool.
Do you drop a target once D2's in the water?
Yeah, it should be, I think it's on the checklist, so it should be ROV 11 launch. Yeah, right there.
That was our launch.
I can hear you, Nav.
Sorry.
Yeah, I usually do it whenever we get in the water, but it's fine if you did that.
Yeah.
Yeah.
No, I haven't dropped it yet.
Okay. Yeah, you're good.
Pilot will wait for your confirmation of thrust before releasing the vehicle.
Roger that, dive suit. Pilot has control.
Pull the pin. The lift line is clear. Let's go ahead and lock in auto head inverse of ship and push center line.
Roger that.
That's a good heading there if you want to just push out.
Pushing out. Coming ahead full. Heading locked in, inverse of ship.
Copy that, pilot. Other than that, let's go off stick.
Off stick.
Looking good.
Auto heading in.
All right, deck. Let's go ahead and launch Sirius. Pilot, please provide some halfway as Sirius comes up and over the side.
Backing 30%. Backing 40.
Good copy, pilot. If you get up over 60%, pilot, let me know.
Roger that. Backing about 60 right now, dive suit.
Good copy. Pilot, we're easing forward way.
Copy.
Midwest.
Backing 50.
It's looking good, pilot. Let's maintain that.
Roger, maintaining. Easing.
Okay, pilot off stick.
Pilot off stick.
Winch, 0 reps.
Zeroed.
Pilot, 0 turns.
Turn zeroed.
Okay, pilot. Target lines are clear. Let's go ahead and descend down to 15 meters, 15 meters per minute. We'll call out every 10.
Roger that, dive suit. Descending. 50.
Descending.
Ready for monitors?
Ready for monitors.
Your auto heading's still engaged for reference.
Thank you.
Oh, you're on something.
10 meters, wire out.
D2, 17, slowing.
Just make out your oxide.
Two 0 meters, wire out.
D2, 23 meters depth.
Three 0 meters, wire out.
D2, 32 meters.
Four 0 meters, wire out.
D2, 41 meters.
Dive suit nav, good tracking.
Good copy, nav. 50 meters, wire out.
D2 holding, 5-2 meters depth.
Good copy. Winch, dive super.
Go ahead.
Can we get control pass to the control room at this time, please?
Control room has control.
Control room has good winch control.
Good copy. Pilot, dive super.
Go dive soup.
Control room's ready to continue with the dive. Let's go ahead and descend.
Roger that. Proceeding with dive.
Nav, dive super.
Dive super nav.
Would you suggest that we come into hold position back here on the AFCON?
I would come to an easy stop when able.
Good copy. Will do.
Did you copy, pilot?
I did. How are we looking right now? We
Yeah.
want to hold at 50 meters for a minute?
Nav, dive super.
We haven't gotten the all call to dive yet. Go ahead, dive soup.
We're in the process of coming to an easy stop at this time. Please watch streaming as we do so.
Good copy. Watching streaming. Yeah, you're probably going to want to push at some. We do have 6/10 of a knot stretching us out, though, and we're
Okay.
still in that surface current for sure, according to the ADCP.
Sounds good.
We have good tracking fix, folks.
Appreciate that. We'll hold here.
Did Dive Super not give permission to dive?
He did give permission to
Did he?
dive.
I thought he
I
did.
missed
Yeah.
that.
Thought he did.
All right, it's
but
on your call then,
I want
guys.
to make sure that, yeah.
Give me 2 seconds
Yeah.
of your time,
Yeah.
pilot. Verifying SCS outputs from HiPAC.
Mm-hmm.
Where do we do that?
SCS outputs from HiPAC should be these guys here, yeah.
So the fact that there's numbers
The
updating
fact
means
that
it's
they're
good?
updating, yep. Yep.
Okay, one other question is making ROV main vessel.
Vessels,
Vessels,
and then click on ROV and main vessel right there.
thank you.
Yep.
Okay, all
and
clear.
apply, yep. Good to go. And then you can change your layers back if you want. Now that we're 50 meters.
Nav, dive super.
Dive soup nav. Dive soup nav, go ahead.
Just so that you're aware, we are in full auto position. We are in the process of passing control to the bridge at this time. If you're ready, I'll pass comms off to you, and you can work with the bridge to make our way to the bottom.
That's a good copy. If you're ready, I will take comms.
Good copy, Nav. Navigator has comms.
Navigator has comms.
Pretty good launch for a new guy.
Not too shabby. Good job, dive soup. You guys ready?
I think so. How are you
Yeah.
looking in relation to the target?
We're right on it.
Right on it.
You were right
Perfect.
on it. We're
Awesome.
just going to go
I
straight
think we're
down.
ready.
Just keep it
Yeah.
stretched out, guys.
Copy that. I've got a. Let me dial in the Z-bias real quick here,
Yep. Let
co-pilot.
me know when you're on your way down. I'll
How much
follow.
have you guys been using to get down?
To get down?
70 or 80?
Saline water, I was at 63%, give or
63?
take 5%.
Okay, we can start
But
around
we're-
65 and adjust.
We're probably just a touch heavy today.
Nav, dive soup.
Dive soup nav.
Bridge has good control of the ship at this time.
Good copy. Bridge has good control. Thank you.
All right, co-pilot, you ready to come down?
Yeah, I'm ready.
Roger that. I'm pushing out just a little bit. I'm ready to engage Z-bias.
Yep, I will follow you down.
All right, coming down.
And paying out.
Descending.
Nope. You're all stopped.
Hold on one second. Auto depth is engaged. There we go. Shaking out full rest. There we go.
Copy. I'm going to start slow until you get a little bit of
Yeah, sounds good. You can tickle up.
52 looks good.
Increasing.
All right, and increasing.
Okay, you can see you're clocking around. There is a half a knot of current coming from the going to the west-southwest.
Copy that, Nav. Thanks.
When I look at the ADCP, it's definitely happening at depth. So that's probably why you're clocking around.
Copy that.
I'm at 25, almost to 30.
Roger that. Let me know when you have 30 dialed in and I'll fine-tune my Z-bias.
When do you usually tell the bridge to shift rotation? At the bottom or now to the A-frame?
I think we wait till closer to the bottom.
We're pretty locked in. We're not making any moves.
If we're not making any moves, you could probably go ahead and switch it.
Okay.
That's about 30 there, pilot. And
Roger that.
down.
Thanks.
Winch looks good. Coming out.
Thank you, Winch. Bridge, nav.
30 meters per
Fiber
minute.
values look good.
Go ahead, Nav.
I saw that.
See.
Yeah, bridge.
That's awesome.
Looks like we're holding position nicely. The ROVs are descending at this moment. We're just going to stay right where you are. This is perfect. Since we're not making any position moves, unless you need to change head, I'd suggest we go ahead and engage port prop and move rotation to A-frame.
All right, I have engaged port prop and port rudder, and I'll change the center of rotation to A-frame.
That's a good copy, Bridge. Thank you very much.
Thank you.
When did you notice those adjusted? Yeah. Gotcha.
As the slip ring's spinning, that's more the differential we've been seeing. Like a 2 dB differential. I'm gonna lower your aux a little bit there.
Copy that. Thanks for getting those lights on.
Of course.
And acoustic devices. Looks good.
Yeah, your blue view's pointed down. Let's see. You want me to range you out?
6 degrees, yeah, if you would, please. Let's pump that all the way up.
You want like 80 or all the way?
Yeah, whatever you prefer. I think like 90 is fine, yeah, where you have it there.
Great.
We're stretched out a little bit. Actually, clocked around to have me looking up slopes, so that's kind of nice.
Big clock, yeah.
Do we have any reading on the subsea currents, Nav?
I can investigate.
Probably doesn't go all the way down. We hasn't.
The 38 does. The 38 looks, well, maybe not quite. At the bottom, I'm looking at 0.15 to the south meters per second, so that's like 3. Oh, it just updated. Stand by.
Copy. Delta's good.
Yeah, 0.2.
Looks good.
So 0.4 of a knot to the south at the bottom.
Roger that.
So basically pushing you up the wall,
Okay.
if we can believe that. That's at a depth of like, well, that's at a depth of 250, so bottom's 485 here. Approximately, you are sitting over 472 meters of water where you presently are. Do you guys want me to back the ship up just a little to the northeast so that you land on the waypoint? It's a little flatter back here.
I wouldn't be opposed to that.
I would,
Yeah, we could back up a little
I
bit.
think so. I mean,
Yeah.
you could back
We
up
have
to
a-
Sirius, but let's creep in.
We have a long day today,
So
so
that's I'm
okay with
thinking.
that.
So you'll end up here once you give a little tether slack. So that's 40, 29, 30 meters. I'm going to make a 30-meter move at 035.
that sounds good.
Sounds real good.
Bridge nav.
Yes, go ahead.
We'd like to make one adjustment to the ship position before we land. Can you please make a position move 030 meters at 035 degrees, slow speed.
Okay, just to confirm, 30 meters with the bearing of 035 at maybe 0.2, 0.1 knots.
That is perfect. That's a good copy, Bridge.
And sorry, one more thing. We are center of rotation is currently A-frame. You're still good with that for a position move?
A-frame center of rotation is fine.
All right.
Thank you.
thanks.
Thank you.
Okay, pilots, move has been initiated.
Copy. Thanks, Sam.
Copy now.
We just
and I'll be listening. So if there's anything, don't hesitate to call. If you guys get to bottom and the current's ripping, we can readjust where we're at. We can always go to the flat surface, kind of like what we did yesterday. But great, thank you guys. Nice work.
Bridge Nav.
Yes, go ahead.
Yeah, I'd like to talk about our situation today and what the bailout location direction should be.
Yes, go ahead.
Okay, if you can look at high pack, you'll notice ahead of the ship to the southwest, we have a very steep wall-like feature. There will probably be segments of vertical face and other segments of softer slope, but generally very steep to the southwest. With that in mind, and given the wind and current, we're going to want the bailout basically dead astern. So let's say a direction of 035 astern.
035 for the bailout. Thank you.
Thank you, Bridge.
Let's see. And then D2, if it stays like that, as far as streaming goes, we could, if you give me slack in the tether, we could just spin Sirius.
Yeah, that sounds good.
Half turn. So then I want to spin, if D2 typically turns right, we would want to spin Sirius left. Is that correct?
Correct.
Okay. So half turn left.
And relative to you, yeah, I haven't turned. So yeah, we'll just let you come to port. I think that's a good plan.
Hey,
I mean,
pilot.
get eyes on the tether, make sure it looks good when we get down.
I don't.
Disabling sled course over ground.
That's always disabled. I think that's a relic.
Where would that even be?
Use cog for heading.
Okay.
Yeah,
Alright.
I've never seen that checked ever.
Okay, copy.
And then we did not have good visual yesterday in tether.
Yeah. Of anything,
so
huh?
Do as good as we can. I mean,
Okay.
we saw what was close to the vehicles, you know.
Okay. We'll do the best we can.
We didn't even see D2 pass yesterday when we set up.
So expected bottom nav is about 470 or something.
It should be
480.
485 at this waypoint 1.
Okay.
The ship is still moving northeast, so by the time they stop, you're going to be probably right around 480.
Okay.
And as you back up to get underneath, it'll be a little deeper. So,
Sounds good.
let's talk about what I noticed that happened yesterday and just general backing when you can't see what you're doing, right? And you start at a modest delta, and if you keep the modest delta and there's current underneath the water, what can happen?
Yeah, you can.
I don't know if
Oh,
you
you
caught
get-
what happened yesterday. The tether went underneath the light bar.
Oh.
Yeah.
Oh, I didn't see that was the
Yeah, I didn't
first.
see that either.
So to get that initial slack, you got to keep the delta modest, right? But then as you go, you do this, right?
Right.
And it's kind of up to both of you to play that
The
dance.
tether went. Is this the shift before us?
No, it's yesterday.
Oh, it's happening with us.
No, we were in the gym.
Oh, okay.
This was yesterday mid.
Not our
first one.
shift.
No, not our shift.
Understood.
There was some, anyway, it's that whole pendulum rather than a steady delta that keeps us safe. Because we can't see the tether. We can't see each other.
Hey guys, sorry, I want to
You're
interrupt.
drawing
We have
by
good altitude
instruments.
lock on Sirius.
I see that.
It
I-
looks fake.
Yeah, I think that's fake. I think D2's is believable though. We just got altitude lock with D2 as well. That would concur with bathymetry.
We're expecting slack tide in
I
a
don't
little
see
over
anything on
about
it.
an hour and a half, 10:15, nearest to tide gauge. See what that does.
So I'm gonna come down to probably at least 30 meters off the bottom. Would you be comfortable with me stopping at 30 and you could stop a little higher than
D2
that?
stop 30 altitude.
I can set up a little higher if you prefer.
No. I think with high currents, typically you want to be lower.
Okay.
Because I think. All right. Sirius lost bottom lock, which is good.
Yeah.
Yours DVL looks good right now.
I think it does. Yeah, it's looked really solid.
Yeah. So D2, I'll stop 30 and I'll stop above you.
Yeah, that sounds good.
Sounds
Just
good.
give me maybe 5-10 meters.
I did a bad thing.
Everything good, Ted?
Bumping
No.
your ox up.
Not good. Everything's good with you.
Oh, you just checking the CTDs? That's when you see that software up and running, that means that the auto CTD started successfully.
Oh, good.
Yep, you're in good shape.
I thought that was me forgetting to do it.
Nope, you're just
Okay.
supposed to verify.
All right. Thank goodness.
And you can check on both vehicles.
That was that script in the background, constantly
that's
waiting
right.
for the
For inputs,
vehicles on surface command or descent
Exactly.
command. All
Yep.
right, good. Jeez, I thought I screwed it up.
You're good.
I guess that makes sense because the SES has numbers, right?
Yep.
All right. It'll be on the stick. Whew.
D2.
D2 looks good.
Yep, looks like we're
5-0.
getting some readings, multi-beam.
That looks pretty accurate.
60 meters, yeah.
You're
Spot
going to be
on.
moving back some.
I'm really curious to see how the DVL behaves today, guys.
Looks good so far. They said it looked good too, acquiring bottom yesterday.
Oh.
At least the first bottom. Before we got on shift. Alright, passing 50 meters, D2. 20 more meters and I'll come to an easy stop.
When do you guys drop the bottom target? Once D2 is like within a meter or two bottom?
I wait till like 5 meters or less, yeah,
Yeah,
usually.
good viz of the bottom, like in
Exactly.
your face.
Exactly.
Okay.
D2 passing 40 meters. See you ready on the winch.
I'm ready. Delta 10.
Roger that.
Oh, big squid zeros.
Disengaging auto Z bias, locking in auto depth for a moment.
Slowing down.
We got bottom on blue view.
Yep, winch is all stopped. Alright, I'm going to crank your aux up a little bit so we
Roger
can see
that.
you.
Alright, we've got about 10 meter
So-
delta.
Yeah. Todd, say once again what happened yesterday while we're holding here.
I think as D2 backed up, they started at a delta 15, which was more than enough to start,
Yeah,
but then they didn't increase delta, and with the
Oh.
current like blowing the tether a certain way,
got
it
it.
ended up like, and there was a really quick heading change as opposed
Yeah.
to a nice steady heading
Got it.
change.
Okay.
Yeah,
Yeah,
and
yeah.
so there was a little bit of madness.
A little bit of
So
madness.
you know,
Okay.
when you can't see, you got to be safe,
Good, yeah.
and you got to go bigger delta when you're straight underneath.
Yeah.
Yep.
So what we're thinking today, tell me what you think. If D2's kind of has good visual of the slope ahead, I was thinking of if D2 just kind of backs up, but then Sirius does the spin.
Yeah, so you're spinning,
I would spin, Sirius would spin counterclockwise
Right.
left,
which is the same as D2 going right.
right? What do you think of that plan?
I think that sounds good. I'm gonna, whenever we start this, I'll start backing. Do you want to come up, or would you like me to push down a little bit?
So we have good lock on the DVL
Mhm.
in the blue view. If I'm just spinning, we could get. Would it be wild to acquire bottom like this? You can get visual bottom, and then
Since I'm already looking
Serious
at the
about
ROV
bottom,
nav bridge.
then you have an additional reference point.
Bridge
it'd
nav.
be unorthodox. But
Just wanted to
we
let
could
you know
do
that the move is complete.
Okay, good copy.
I feel
Move
comfortable
complete.
since we're-
Currently,
10
we are
meters
30
to
meters
visual.
off bottom, and the pilots are beginning to acquire bottom here momentarily. We're looking good. We're holding station.
Sounds good, thank you.
What do you guys think of that? Get a little lower till we get visual. You have an additional reference for D2, or we could do it at here and then just come down.
I think I'd feel better the second option. I like.
Okay. Understood. I
Yeah,
just
if-
wanted to put it out there.
Yeah, it's not a bad idea.
So let's see.
If you don't mind, I could back and you could just haul up on the winch as needed to manage the tether.
Like you go in auto depth. That's
Yeah, I'm in
fine.
auto depth, and I'll just back towards you,
Yeah.
and
So then,
and you'll rotate and haul up on the winch just a tad as I get closer to you.
yeah. Do you want to put auto heading in this time? Think your headings are
yes.
set?
You want me to lock an auto heading? We can both do auto heading. Yeah.
Yeah.
Sounds good. Why don't we do that? I'll slowly start backing. We got good visual on your tether cam of D2.
I do not
I see.
see you in the tighten it all.
Okay. I see you in your. I see me in your tether cam.
Let me put up the.
Yeah, it's really hard on in this turbidity to see with the Titan,
Let me see
but
if this picks up.
you will
anything.
see eventually. I think I see something, but that might just be particulate. Yeah, I think it's just particulate.
Copy that. So I think if we can get to like, if you can get to the bottom
Mm-hmm.
and we can use USB-L too.
Okay.
The bottom of that monitor.
Mm-hmm.
I don't think you're gonna see it, guys. I don't
think.
We lost Viz the other day, so I'm looking at. I think you should drive by a high pack mostly, but
I'll just start.
we have a relative distance of 48 meters, which I
48.
think is baloney.
Yeah, it's a
So
little
when
high.
D2 is somewhere like around where my cursor is,
Yeah.
I think you're going to be right underneath of Sirius.
Okay.
That's my.
So
Sounds
maybe
good.
halfway to where Todd's cursor is, you can start hauling up on the winch a little bit.
Okay, so I'm gonna preset my camera to about 4.5.
Okay.
So I'm at 5.0,
Yeah, that sounds good.
and I am ready.
You ready for me to back?
Yep.
All right.
As you back, I'll come up.
Coming out of joy lock and backing slightly. Keep an eye. I can see my aft light and your
Visual on
camera.
your aft, yeah.
Backing. Watching high pack. Should see that distance start to decrease. I see the tether slacking a little bit.
Yep, looks good
looking
there.
good.
I'm slowly
Yeah, you
coming
can
up.
probably start tickling the winch. Backing. Yeah, it's looking good.
Okay, you're closing range quickly.
Copy.
You're
You can haul
going
up a little bit, Sean.
Yep,
You're
Delta
moving with the
13.
current, so you're closing range quickly.
Copy. We got a 13 meter delta that's increasing. We're looking good. Yeah, it's looking real good. I can see most of the tether there.
Delta 15, you're at the bottom. What do you think about here?
Yeah.
Ready for turn?
I probably let me come back just a little bit more. You have your camera full back? Or no, you're looking
No,
at
it's at
45.
45.
Okay.
So when I spin,
Yeah.
you can come in.
I'd say
I'm
probably now you can probably start spinning. Yeah.
Delta 17,
That's
I'm
perfect.
spinning.
That's good delta. Yep. Maintaining.
I see you.
Yep, we see.
Down cam. Perfect.
It's looking good.
Like butter.
There you are. Nice.
I'm going to complete my spin. Point south. I'm
Could
south.
visit the tether there.
Delta 17. Rotating camera down a little bit. That
Do you want
looks
me to push
good.
out a little bit?
7.0 for reference.
7-0, copy.
Let's see.
You should be looking.
Video. Can you
With
bring
me.
out Sirius light on the Sirius D2 light on Sirius camera?
Pushing out a little bit.
Pied, do you care if I turn some Sirius lights
Yeah,
on?
just whatever lights you need to get good biz. I found, one light
I'd
bank.
recommend turning off the central bank and turning on the outer banks.
Copy. Central is off.
And then you can,
What's that? All right.
yeah, you can do just one bank if it helps too, but play
Yeah,
with it.
yeah. All right, so I'm going to rotate up to about 45 degrees.
'Kay.
And then I'm going to lock it in so you know for reference.
Roger that.
If you turn off all lights, you won't be able to see your umbilical, but I'll be able to gain up the
Copy
ROV.
that. I'm good with this setting. Are you good with this?
I like it.
I like it. All right, I'm ready to come down slow when you're ready.
Roger that. Do we want to, I mean, it looks like we're normal to the face here. I was going to say bathymetry otherwise, but let's go ahead and drive off of this.
Yeah.
Yeah, we can just ascend here if you
Yeah,
like
looks
it.
pretty good.
Ready to come down?
Yes, sir.
All right. Just gauging. Z bias in. Coming down.
I'm down. Delta 17. I'll let you know if I have to stop for tether maintenance.
Roger that.
And I'm going to keep my camera here, so you just try to stay center.
Copy that.
Looking good.
John, I'd probably recommend putting your HD between your toes. As
Copy that.
Choir bottom.
Yep, coming down.
John, I think you're getting
A little bit ahead.
yes, you're getting stretched out.
Slow down. Okay, we've got HD. We've got HD 1 and 2 looking
Here,
down.
I need to catch up. I'm not.
Yep,
our
sounds
delta's
good.
pretty
I'm
big.
slowing. I'll let you catch up.
Yeah, back up. I'm going to pause there.
That's starting to look better there.
Yeah, just back up for me. That looks better.
Got a little stretched out there.
All right, that's looking a lot better.
Okay.
That's a good thing. That's
I
what you're
think, when
supposed
you're ready,
to hear.
I'm
The
good to go down
pilot's in the
again.
right
Ready to
channel.
come down. Descending.
Shoot for
Video
about
survey
15.
one.
Go ahead, survey one.
I think we're ready for the pre-dive brief whenever the pilots are ready for the general announcement.
Okay, I'll check in. Pilots, it's good time for the general announcement.
Sounds good video.
I'm off wedge for just a little bit until you go down a little more.
This is Okeanos video engineer Roland Bryan making a general announcement to all those on the ship and the shore. This is a reminder that all audio associated with this dive will be recorded with our archive video, as well as being streamed ashore via the satellite and the internet. Keep that in mind as we proceed with the dive today, and enjoy the dive. Thank you.
All right, thank you, Roland.
Delta's getting a little high there.
So good morning, everybody, and happy Labor Day to those who are not laboring. We hope to bring you something fun to watch during your hopeful time off. So last night we transited away from our 10th dive site, which was in Beam Canal, and we're working our way back out to open ocean. Not quite yet.
You're getting a little stretched out again.
Copy.
Back up a little.
Zoom on over. So while we're waiting for the weather to dissipate a little bit more in the Gulf, we've pulled into Cordova Bay, which is a little bit more exposed, but still offering protection from the swells and the weather that we're seeing out, kind of more in the open ocean. And it should position us to get out tomorrow and begin diving in a little bit deeper and maybe a little bit less physically stunning places than these fjords that we've been spending the past couple days. We're really excited about today's dive here. It looks like it's going to be an interesting feature. Potentially, we should see a couple of different areas where different communities could be thriving, and hopefully some diverse geology as we land at about 450 meters or 480 meters, and then we're planning on ascending up to about 320 meters. And along the way, there'll be a couple of different steps within the bathymetry that we collected last night, which is really beautiful. We're looking at a 5-meter resolution with no vertical exaggeration. So this is what it actually looks like down there. So another very impressive feature that we'll be diving at. With that, I would like to pass it over to our science leads to introduce themselves. Jamie?
All right. Thanks, Sam. My name is Jamie Conrad. I'm a geologist with the U.S. Geological Survey. I work at the Pacific Coastal and Marine Science Center in Santa Cruz, California. And yeah, I'm looking forward to today's dive. We're diving just offshore of Long Island, which is one of the 1,100 or so islands that make up the Alexander Archipelago of Southeast Alaska. As Sam mentioned, we're still up on, geologically, the North American continent. We're about 40 miles or so, I believe, from the margin. And we're in, hope to see some rocks. I expect we will see some rocks. And these are a little bit better understood and more well known than the rocks we've dove on the last couple days. These are rocks known as part of the Alexander terrain, and these terrains, this is one of the many terrains that make up most of the west coast of North America and virtually all of Alaska. These are more or less, if not internally coherent, but internally related packages of rocks, bits of crust and perhaps some oceanic material that's been assembled or accreted up against North America by the northern movement of the Pacific Plate. In general, the process that happens is that the oceanic basement of basalt, which is created at the spreading ridges, does tend to subduct along these a convergent margin. But the things that accumulate on top of it, including the basin sediments, as well as anything else that's built on them, including volcanoes or bits of crust, tend to get scraped off and become part of the continent. And this is one of the primary ways in which new continental crust is made.
Bridge nav.
These rocks are fairly old,
Just making
that
you
we
aware
expect
the
to see
vehicles are
today.
now on bottom,
Ordovician and
right where we wanted to
Silurian
put
in
them.
age, which is
Bailout position
between
remains the same
500
at
and
0
400
degree,
million
5
years old or
degrees,
so.
All as well.
They've from the onshore mapping, I'm expecting that we will see some volcanic rocks, and hopefully not too metamorphosed, so we might be able to pick out some flow features and whatnot. This terrain has actually been described as an island arc or rocks related to, say, those that formed on this along the Sierra Nevada of California, but probably formed from somewhere much farther away. A modern analog for these rocks that we're going to see today might be the modern day Aleutian arc. So it'll be interesting to see what we see. But with that, I'll pass it on to our biology lead.
Hello, I'm Merlin Best. I am the biology lead on this expedition. I work for the for Fisheries and Oceans Canada out of the Institute of Ocean Sciences in Sydney, BC, on Vancouver Island. And yeah, this area that we've just landed in, I see, is the exact area that this coral species distribution model I've been talking about for the past few days has highlighted. So just to give you a bit of a background, it's kind of a series of calculations that took into account 20 years of bycatch records for coral and added in all these environmental factors like temperature, depth, and slope, and produced this map that gives us a gradient of probability where you could find corals in abundance, and it covers the whole Gulf of Alaska. The ones that I have access to have the highest numbers across the whole Gulf, pretty much right here in this area, Cordova Bay.
We can try to spread them out, maybe.
So since we're, kind of taking cover from that weather pattern that's moved through the Gulf, it's a really great opportunity to check this out. Whether or not we find a lot of corals, we're looking for those big primnoids, Primnoa pacifica specifically. But whether or not we find them, it could help us validate or ground truth that model, which is what we call it when we go to check out an area and see if it lines up with what the model predicted. So yeah, and I see right now we've landed on kind of the soft sediment base of the feature that we're going to be kind of climbing up in steps today. And yeah, we've seen a lot of this just... The water column here is full of marine snow, lots of turbidity and debris from biological activity, and a different kind of fish right now than what we've seen so far. I'm lucky to be joined by a team of shore-based scientists that I have access to via a chat room, so hopefully there's some fish biologists on board that can help us with identifications, especially when we see new things.
I could see him a little better if he backed up. But you're looking down 45 degrees still. Yeah.
So all of this beautiful marine snow that we're seeing is kind of the product of biological activity higher up the water column or further away that's been carried here by the current in this case. And it will all eventually settle. Some of it's recycled in the water column, but most of it will settle eventually on the sea floor and become habitat and become food for all of the different animals that live here. I think already I'm seeing some sea stars, maybe a little tiny bit of coral and some anemones, so it's already lots to look at.
Out for white bouncing?
There's a flatfish down there. We had a nice zoom in of this earlier, but you can tell by those little white dots around the edges of the fish. It's probably a Dover sole.
John?
John did it yesterday,
Oh.
2 days ago.
Yeah.
And nice to see that the current
On
is a
bottom
little
now. Murky like this.
bit down from
close
what we
to
saw
bottom.
yesterday.
How'd you have
Yeah, it
lights?
seems a little more calm. Things don't look like they're about to get blown off the side of a rock.
Yeah, a blizzard
Yeah.
of marine snow.
It reminded me of driving in a blizzard, exactly.
Once you get it out. I'll leave
Luckily, this D2's got four-wheel drive,
the droppers are out. Pointed out. Copy that.
four-thruster drive.
Hydraulics
Actually,
are
there's
up.
probably, what, like
Valve packs on.
8 on there?
So I'm gonna let D2 settle down here on the bottom just a little bit.
And then, yeah,
And then we'll be ready for a white bounce.
sounds good. DVL looked good acquiring bottom, right? I thought it looked pretty good.
Yeah, how's it been so
It's
far?
been really good so far, yeah. Locked on about 100 meters and kind of stayed that way the whole
You have the altimeter off, right?
That I don't.
No, I thought we
No.
were running
Yeah,
with it
kill-
on now.
kill- oh, we are?
I thought we just wanted to do it for a day.
No, I thought it was a definite interference. Is that not so?
Didn't seem to interfere today.
Okay.
I can have it off.
Yeah, we can
I
turn
thought
it
we were
off.
just
You
running
may
it.
want to run with your sensors page on, and you can
Yeah.
watch which one is
Yeah.
doing the right thing.
Well, it's off now, so.
Yeah. I mean, you don't need it on unless the DVL is being flaky.
Right.
So
I thought we wanted on acquired bottom because the DVL hasn't been trustworthy. So
Yeah.
we wanted
I have
the
a
altimeter
suggestion
We can
from
leave it
the...
off for now.
Oh, a
and
little
let's keep an
squid.
eye on the
Cute.
clock.
It's off for now.
Okay.
A suggestion from the science chat that those
feels like we got pretty
long,
stable
thin
here.
fish could possibly be herring. So if you see any more, once we get calibrated, we'll take a zoom and see if we can confirm.
Looking good, guys.
Roger that. I'm nice and settled here, just barely touching the bottom, bringing my rotator up. It was 3.5 degrees or so, right, Todd? For white balance?
The white balance is 3.5 up.
Yep.
Give or take a half degree.
Roger that.
Yeah.
How are you feeling, co-pilot?
I feel good.
Ready for a white balance?
Yeah, sure am.
All right. Could you spin up my hydraulics for me?
Hydraulics are up. You're ready to go.
Copy that. Thank you.
Is dissolved.
Oh, I can put your camera back down.
I'm done using that. Appreciate it.
Get it
Arm
to
coming
me.
live. 3, 2, 1. Arm is live.
Curious little herring, if that's what it is.
If you're watching at home, I believe on cam 3, if it's not there now, it may be there soon, is a 3D rendering of this face we're going to be climbing up. And right now, you can see
Our bailout today.
in real time a model of D2 down in the lower right corner, and you can see the up above us the orange-looking cliffier, possibly outcrops, but anyway, terrain we will be climbing today.
All right. Video, when you're ready, we can come in and fine-tune it.
There's some serious lighting necessary.
I've not done lighting yet.
If we zoom out a little bit, we can see this package is perhaps bounded by maybe a fault on either side that's juxtaposed this group of rocks in front of us from separate blocks to either side.
Looks a little slanted video.
In
Do you
those
want me to adjust that?
little canyons or valleys
Is that
that
good enough?
kind of are forming a little V shape now
Enough. We'll know when the lights
in the
are up.
view.
All right, I started. Adjusting light. Let me know what you need, video.
I need just a
Is that close
skosh
enough
more.
video? You need me to adjust
And
that?
that image on stream
looks
3
good.
that we're looking at
Pilot.
is
That looks good.
colorized by
Say a little
slope.
bit more light
So
on
that's
the
something
right
that we like
side.
to look at when we're
One
picking a
light
dive
on the
site
right,
for
stand
a number
by,
of reasons.
stand by.
One, in
Again.
some of these high slope
Turn down.
areas,
Can
you
you
have a
hear me
better opportunity to see exposed rock,
Okay, gotcha.
so
Yeah,
better
I've talked to you
attachment
for
points
long.
for a
Yeah,
lot
sorry
of the
about that.
organisms we generally look
I
for,
understand.
and exposed rock for our
yeah, good
geologists to look
positioning
for
of
as well.
Good.
As well as, perhaps more importantly,
Better or worse
is
video?
it gives us a little bit more information about the safety of the
High in the
area
middle and
as it
nothing on the
pertains to
outside.
piloting the ROV. So,
Currents come down just a little
for a
bit
number
off
of
the
reasons, we
surface.
like to have
Clocked around a little
areas
bit.
where we can
Hold
look
there.
Hold it.
kind of dead on and go upslope
with the contours, so we're not running into any surprises, and we know that we're always kind of looking up.
The camera.
And that's good for safety of the piloting of the vehicle, as well as for the quality of the imagery that we're collecting. Whenever we're looking down or going down a slope, you'll see a lot of blue water. You won't quite be able to see the bottom as well as when you're looking upslope. So we generally look at the slope maps to both tell us what directions are best for our view, as well as for the safety of the vehicle in general. And especially in these areas where we have high currents and potential for those currents to shift and be dynamic, we want to be able to make quick decisions if we need to bail out of an area for any reason, if we feel that it's become a little bit hazardous to the vehicle. And that way we can communicate quickly with those driving the ship to help with some of those moves and make sure that we are taking our time and being as safe as we possibly can.
And I'll point out now that what we're looking at here is from mapping that we completed just a few hours ago. As a geologist, much of what I refer to and much of what we know, almost everything about what we know about the geology of this area has come from exposures onshore. So what we're looking at here is essentially brand new information and essentially the baseline information we need to start mapping geology, and in that sense already we can see a little bit of new information. We're looking at a couple different packages of rocks and possibly faults that are brand new information for geologists, and ultimately will help complete those geologic maps out into watery areas where mostly we, in the old days, have tended to just color it blue and go, we don't really know what's lying underneath all this water. This is really neat to be out here.
Yeah, and then kind of similar to what Merlin was saying about ground truthing those models for coral distribution, the ability to go down and collect samples and take video of these types of features that we're seeing in the bathymetry allows us to ground truth the bathymetry. So then if we're seeing a similar type of thing in somewhere else, we have a better inference for what we might be looking at. So I think it's really important to help us better decipher. I've been looking at bathymetric models and maps for most of my career, so I feel like I know what I'm looking at, and it's good to get either confirmation or a recalibration of that as we go down and see what all those dots and pixels actually represent. Yeah, it's pretty cool to be able to do.
Do
things like turbidity, like all of this stuff in the water column, does that ever affect the scanning that you do?
It depends. And a lot of work with sonars, the answer will be it depends.
Yeah. Okay.
The frequency at which our sonars operate, so our multi-beam system, which generated this, is a 26 kilohertz system. And so with all sonars, you're basically balancing resolution versus depth. So you can get a higher resolution with a higher frequency system, but you're sacrificing the depth in which it'll go down, because the signal will attenuate through the water column. So with these systems, it can penetrate through this turbidity no problem. The bigger issue that we run into, especially in dynamic areas such as this, is salinity. Because basically what this data comes from, each of those points, is a measurement of time more than anything else. So the sonar sends down a signal, and then it basically just counts until that signal returns. It divides that in half, and then you get the depth. So I was always taught it in physics as the dirt equation. That's what stuck with me. So it's distance equals rate times time. And that's all that's happening here. You know the time because the sonar is measuring that, and the rate is determined by the speed of sound through water. And that's what can change. So depending on the density of the water, or basically how warm or cold it is, how much of it's stacked on top of each other, and what's in it, the salinity,
or the conductivity will determine how fast that sound can travel through the water column. So that's something that we measure quite closely, especially in these dynamic regions. And we can do it with what's called a CTD, which we actually have one on the ROV, and that's kind of ubiquitous in marine science research. You're always kind of trying to figure out those oceanographic principles. And CTD stands for conductivity, temperature, and depth. And through that, you can infer sound speed. But we also use, and I think you saw these yesterday, these XBTs, which are expendable bathythermographs. And that just measures temperature, and you can tease out some of those other parameters by looking at basically a database of other data that were collected by CTDs. And the nice thing about those is we don't have to stop the ship. We can do it while we're moving. So it's a really efficient way to collect that sound speed information.
Versus pausing for 6 hours or so to collect it with a CTD.
Wow. Yeah, and for the data that we're collecting at these depths, getting that CTD profile won't make the data that much better. Our uncertainties will still remain close to the same.
Oh, very cool.
But in areas like this, I spent most of my career before I came to NOAA Ocean Exploration on a different NOAA ship, the Fairweather, doing coastal ocean mapping in areas like this, including a little bit further up this strait, is where one of the first projects that I worked on was just, I think, 2 or 3 miles away from here. So it's kind of a coming home for me. But I do know probably better than most how dynamic the water can be in these areas, and especially when you're, I think, the more beautiful the place was, the more annoying the work was. So it kind of balanced that way. So when you're working by like
beautiful outside.
But working by waterfalls really makes that a lot more difficult. You get fresh water coming in, you get the tidal changes, you get all of that. So being able to model that well with the CTD profiles was pretty critical, and definitely took a lot of work.
Video's clear.
Copy.
Pilot.
Cakewalk. No problem. Well, watch this. Thank you.
Not as steep as that first.
Yeah, this visualization is new for me, being able to have a little mini D2 kind of climbing up these features. Really helps to just put it into context in a way that I've never seen before. You can look at a map and contour lines and sort of imagine, but this visualization is pretty incredible.
There's a ray.
I don't know how I'm going to go back after this. Oh, I think that's a skate.
See that?
Oh my.
Yeah, that's a skate. Just swam by.
Watch leads, nav.
Watch lead here.
Yeah, we are just getting settled on bottom. You just saw that we finished our white balance, and we're going to get moving here in just a minute. Is there an interest in a Niskin sample before we get going?
Ooh, yes. Yeah, I think so. Thank you.
Co-pilot will run us through that.
Copy that.
Perfect.
Hydraulics are still up, which is great. Reset. All right, Nav, are you ready for Niskin?
Nav is ready. Is survey 2 in?
2. All right. Video, you ready for Niskin? Caitlin gave me a thumbs up. All right, I think we're all ready for Niskin. I'll do it at 3-0 and fire. Good fire, Niskin 1.
So there was interest in looking at those fish that are swimming.
Roger.
So we're just, we paused for a moment to collect some water sample here for a study of its eDNA or environmental DNA. We'll take that water, and technicians up in the lab will filter it and keep that filter for genetic analysis. The idea being that anything that swam through or the current went past in the water column will leave a little bit of its DNA behind. And we have the technology now where we can sequence parts of all of the DNA, parse it out, and get a profile of the animals that are in this area.
Watch lead, nav.
Watch lead here.
Just want to confirm we have the sample ID number correct. I have it up on the HiPAC screen, top left corner. We got EX2306. D1101W. Is that correct?
Copy
That's correct.
Okay.
All right.
I'm just looking due south 180.
Yeah, roger that. I don't see.
You guys, let me know when you're happy to start bumping along, and we'll get us moving. But looks like you're.
I think we could start. I don't see anything.
Yeah.
Obstructions that
I just push straight ahead if you want.
Off screen, upper right.
Upper right.
Could we pan to the right if you're taking requests? What is that?
Probably because of low visibility.
Oh.
I think your best bet is
I think
try
it's
to,
just
like,
seaweed.
stay roughly.
A very
center of my
fish-shaped
screen.
seaweed.
Yeah,
After
so-
seeing that skate kind of cruise by.
Exactly.
Eyes
Now
are
I've got
looking
my
for
eyes
'em.
out. Yeah.
Yeah.
Your face
We
is
haven't had a
so-
really good
Yeah, is that
skate
working
sighting
out?
yet. Have
yeah,
found
the
a really nice skate egg attached to a
average
sponge
track's
a
gonna
few
be more
dives
like
ago.
210 to the next waypoint, but we could go local terrain up to the south, maybe 185.
Yep, that looks like
and
kelp
then
to
we're
me.
Start to arc around to the right a little more. 210.
That's great. Thanks. I don't think we need a close up.
As we
Pretty
Continue.
Thanks.
Yeah.
Sorry, I'm just volumes here.
Can we see what happens if we kill the main lights? Just
And we
have
got
the
a
outer
question in the
bars
chat
We can
if
give it
we
a go.
use ADCPs for current
Got it.
profiling.
Upper is so main's
And
off, yeah.
yes, so an ADCP
That's
is an
right there.
acoustic
Yeah, I guess
Doppler
that's
current
too much.
profiler.
Too much
So
probably.
basically, the same way that sound changes as a train
You
passes
can
by,
dim either
changes
of any of
pitch,
those if you want us to
the
try.
sound that we emit from the ADCPs will
Yeah.
change its pitch in general. And you can measure the speed of the currents through the
Alright,
water column
we
with that.
decided, was
We
that
generally
180 you
don't
guys
run
were
those
185,
all the time when we're
25,
mapping.
there.
One of
So
them
that's what it may-
interferes
call it in.
pretty greatly with our multi-beam because they're at similar frequencies.
Bridge nav.
But whenever the ROV's in the water and beforehand,
we were
when we're
ready for
doing
our
our
first move
tests
on
of the
bottom.
drift
If you're
and making
ready.
sure that we're seeing what we're seeing at the surface and what we're seeing down below
Okay,
either
let's
match
do
up or
20
something that
meters
we can account for with the vehicle
At
motions.
185
We'll have
degrees,
those running. And we
decimal
have
2
two
knots.
separate frequencies. We have a 38 and a 300 kilohertz. And
Good
the
copy.
reasons
Thank
that
you,
we run
Bridge.
both of them
Copilot,
are
if you
for the same
wanna
reason I
mess with
kind
that,
of started
I'll
the
scan it.
discussion
Feel
with
free to do so.
the range
The light.
resolution. So with
Dogs
the
cam, the camera, if you'd
300
like to deploy
kilohertz
that, I
system,
think
you're
we're
able to
in safe
see
condition.
a finer
sure can.
detail of the more
Yeah, understood.
uh,
I
the
put it out.
layers higher in the water column. And then with the 38 kilohertz, it penetrates a little bit lower,
the white
and you can see
dot is the
deeper.
bottom of the
And so
screen.
you kind of get a full look at what the currents are doing throughout. And we have that running through the entirety of the
That guy
dive,
right
so we
there
can
in the
see
middle.
how that's changing and then adjust the way we're piloting. And it looks like we got a little hermit crab making an appearance.
There he
A
is.
little hermit crab in some of those kind of pyramid-shaped shells, the Calliostoma probably. And I think hermit crab, I believe the family is
Sit down here.
Paguridae.
There we
And
are.
I'll probably leave it at that, but we're getting a nice close-up of this one right now. Maybe,
Maxim.
I think there are two separate shells that may be a little snail shell of some kind underneath.
Is he thinking about changing shells here?
I think there's two
Could be two crabs.
Or is it two
yeah.
crabs?
There we
Okay.
go. I was
Oh,
like,
yeah.
how many big claws
Oh
am
yeah,
I
there are all the
counting?
big claws.
Yeah.
Somebody shelping one another. Just holding on
Just
there.
visiting.
Clear.
Copy that.
Wow.
Can you come out whenever?
Never been able to get quite such a good close-up of the eyes of a hermit crab. These are obviously really small organisms. The zoom level that we
Sweep
can
the
achieve with
lasers
this camera
across.
is always constantly blowing my mind. So you can see those two laser dots, the red dots floating in the middle, are exactly 10 centimeters apart. So this is the tool that we use to kind of gain a little bit of perspective and get some detailed measurements of what we're seeing. Otherwise, it can be really easy to
Lifting off.
get a little bit lost.
Moves underway.
Yeah. Copy. I'm starting to catch up a little
Lots
bit.
of sea stars
Big
so
fish.
far. Oh,
Fish.
there's a
Come on.
big
Push
fish.
ahead.
Oh, that could be what we were actually, maybe what we were
it's a
seeing
sole.
before.
You can take
I think it could be
whenever you
a
want.
maybe a halibut. Oh, or a sole.
I
No.
think it's sole.
There we go.
Clear.
I'm seeing those little white markings around the side. I guess that could be what we saw
I
in
saw
the
that
distance earlier.
dorsal fin up and got excited.
Yeah. I
My
mean, the
camera's
soul is still
fine.
exciting.
It's
Look
very
at
exciting.
it. I
These are
love
nice.
how they're twisted around. So, yeah, the way that these flatfish have evolved with their two eyes on the top and then the rest of their body twisted around so that one side is flat against the ground.
That's another one.
And they don't start their lives that way. The eyes migrate over after their larval stages.
Oh, cool.
Even
I love that.
weirder than
Even weirder. Yeah, the mechanism that must get set off to make that torsion happen from between the larval stages, that is pretty cool. See, I think these are, again, the Pacific Dover sole.
If it's going to
Let's
sit
see.
there for us, there's also one directly behind that.
And I
Rain-
imagine these mouths to the sounds are probably a really great place for a flatfish to live. You get high currents, a lot of nutrients.
Yeah, this is a pretty high abundance compared to the, kind of scattered sightings we've seen so far.
No, maybe that's a different one. Maybe that's...
So I have a couple of facts. The Pacific Dover sole, Microstomus pacificus,
Hey, Roland,
ranges
we're about to
from
do a pilot
Baja,
change here, so
California
almost
to the
steady
Bering
up.
Sea.
Stand by.
Takes its name from a resemblance to the common sole of Europe, which is often called a Dover sole. So this is the Pacific Dover sole. Every now and then I have a few fish facts. We do see these from time to time on our seamount dives.
Wow, you might want to push out just a hair of everything over. Camera's come down to about 7.5.
Oh, there's a nice big jelly.
Maybe Solmissus.
Hey, watch these. We're going to do a front row change here real quick, and they'll be right back with you.
Sure, sounds good. Oh, there's another one of those white Henricias with the red spot in the middle.
Oh, yeah.
We saw, and maybe a Chiraster, the unidentified species that we were able to collect yesterday. Maybe. Hard to tell. It's a really big brittle star.
Still moving for another couple meters.
Apparently there are a couple of species of those Henricea sea stars that have the red dot right in the middle. It's very dramatic. I love it.
Nav, go. Understand. Move complete. Thank
So
you.
here we're kind of making our way up on the soft bottom
Pilot.
to the top of what we would refer to as a fan.
25.
That's probably getting a fair amount of sediment, seeing that we're kind of at the mouth of a valley, seafloor valley that's coming down from to south of us. So.
Ooh, I think this is one of the corallimorph anemones, just judging by those little balls at the end of the tentacles. A little bit different group from the anemones that we have been seeing. I think they're always really intricate and beautiful. You can see a couple of different size fractions of these tentacles. Really cool coloring.
Lasers.
And I referred to this as a soft bottom, but when we zoom in, you can see that it's actually a pretty rocky, pebbly bottom of loose pebbles and cobbles, which isn't terribly surprising, as we've seen the currents are pretty strong back in here, and likely any fine sediment is going to get swept away in these currents. So we're looking at just a little thin veneer here of mud or silt that's probably pretty ephemeral.
Pilot, can you hear me? I got you. All right.
Whoa.
Zoom if you want it.
Okay, this is so much weirder than what I thought it was. What are we looking at? I thought it was...
Get full screen.
Wow. Looks a little bit snake-like.
It looks very snaky.
Very snaky. Is it a snaky worm or a wormy snake? We have a ribbon worm, Nemertean. I've never seen them kind of look like they their sort of head is up and searching around. Very cool.
Fog lights
So,
up 30%.
Roger, for now I can get those.
We're going to get a little closer. Yeah, wormy snake was my gut feeling, but seeing it look around like that.
I know. Yeah,
about 30%?
a snaky worm, I guess.
40%.
Wow. But like even it looks like it has that little, almost like an eye spot. That's wild. So we have a suggestion from the science chat, a Nimertian ribbon worm. And it has some really distinctive patterning, that stripe along the end. Looks like it's kind of searching through the water column for, I would assume, bits of this detritus, but it doesn't have to look very far. It's pretty much everywhere.
Yeah, it looks like it can compress and extend its body to a pretty good extent.
Oh, wow. It does feel like watching a snake in the,
Which way should we
you know, in the grass. Very cool. I have never seen a worm that looks like this.
It's pretty large, too.
Yeah.
It was
Let's
greater
pan.
than the 10 centimeters.
Oh, was it? Oh, wow. It looks like it's injured back there. It's missing a bit of its, there's a break in the tail. Oh, no. Just buried. Just buried, I think. Wow.
Huh. I've never seen anything like this.
Yeah, me neither.
That is
amazing.
very cool.
Yeah, well, this close-up you can see, I mean, there aren't a lot of identifying features I wouldn't think that you can see from a video. I assume this has to be examined in the lab to take it down to a species, but that that white stripe that runs along the whole length of the worm seems pretty distinctive to me.
Well, actually, you didn't
Very
want to collect
cool.
this, did you?
No, it's not on my list.
Okay.
So I think we'll proceed.
Okay.
Thanks for that. Very cool.
I think it's about 10 long. Pretty small.
Clear.
I think there's another one there. All right. Which way should we be moving now?
Facing 180.
Yeah, the last move was 185, completed 5 minutes ago. Would you like another?
My rotator's at 46.
Oh, that's a shark, right? Right? What is that? You come in? Oh, it's the same guy we saw.
Another, a really big flatfish.
I am starting to
Yeah,
think
you
that
can
that
move.
skate we
what
thought we
direction.
saw before
It was
was one of those.
185,
Yeah,
20
me
meters.
too.
It almost tricked me again.
Todd was cheating you guys a little bit this direction so that you could square up by the time you come up to waypoint 2.
Great.
You okay with that, co-pilot?
Yeah.
All right, 20 meters, 185.
Copy, 20 meters, 185.
You'll out the water column.
Roger, eel pout.
Bridge, RB nav. Can I please get a ship move?
Yeah, I think we know
20
this one. The
meters,
eel
20
pout, they
meters,
make those
bearing
really
185
distinctive
at
little
decimal 2.
little circles
Takes it
in the water column.
That's a beautiful shot with all the
Wow.
marine snow.
Yeah, it just looks like
Yeah,
it's
that's right.
drifting
Yeah,
among
it's
stars
near
in outer
focus.
space. I love
Nice.
these.
You got fogs on around this stuff?
It's up in the
Yeah,
water.
I
Oh,
think
that's so
so.
cool.
Wow.
So I don't know. I've heard a couple of
Fogs
things
are
that could
off.
be kind of resting and slowing their descent,
In my direction.
or they could be a little bit stunned by the lights, and that's a
Diagonal
defensive
down
reaction.
left, pink, squishy.
Right here?
Yeah.
The eel pout version
big.
of the
It's
fetal
star,
position.
Squishy star?
Yep.
Really? Oh, good. Okay. Yes.
Closer to the
Okay, we've had a replay, and that was a skate.
Okay.
Great. This looks like a Hymenastra sea star, Hymenaster,
There's a
and a
fish that's
big
coming in.
scale
Lots of
worm
fish
towards the
going.
bottom.
Skate.
Skate.
Oh, do we have a skate? Yes.
That's a
Speak
skate.
of the
Oh,
devil.
beautiful. Wow, okay. Try to pull up my references. There are a few different skate species known from this area. Looks a little bit familiar.
Mitchell Hebner suggests a big skate is Raja binoculata.
Yeah, there's also the long nose skate, which I immediately thought of because of
The long
the
nose.
kind of longer nose. Raja Rhina. We can review the video and take a good close look. But I think I saw there's two darker dots on the wings.
Trying to get some light on it.
Look at those. What are the eyes on this one?
Down for landing.
Oh, I think they're so cool.
All right. Go for eyeballs.
So I think I'm seeing, I mean, the pattern on the scales is highly variable among these fish, but if I saw this in my annotations, well, no, I'm not sure. The other ones I'd probably lean towards deep sea sole, but it's giving me actually maybe more halibut. Let me see. This is what happens when you get an invertebrate biologist to check out vertebrates. I'm getting a handle on the skates, but we'll at least collect some really detailed video for others to examine later.
Pushing out behind.
Copy.
Video, he turned the lights off on stereo, so I don't know if you noticed.
Yeah, I gained it up 9 dB to give you a better view of this.
I'm kind of making my way uphill a little bit for Nando.
So
Copy.
C star upper left, maybe
Dale
E.
says 15 right now.
Okay. Going for the sea star.
Yeah, I'd love to zoom on this sea star. Thank you. Oh, I think another Henricia with that little red spot in the middle.
Are you still
Great, some fan worms, lots of encrusting sponges. I'm seeing brachiopods, I think. Oh no, maybe little scallops. A little gastropod shell to the right. Lots of empty tubes and full tubes that kind of fan shape coming out of that tube is a worm, probably a serpulid, I think.
The way the sea star is splayed out reminds me of those old crime scene chalk drawings.
Does look very dramatic.
Copy.
Think either a Hippasteria or a Ceramaster.
Go ahead, Bridge. Roger, thank you.
Oh, what's this?
Something coming by.
Oh, yeah.
Ooh, a really big rockfish. Wow.
Lasers are up. Try to splash them. There you go.
Okay, this could be a yellow eye.
I like the lasers now that we got the footage. I'm gonna try to get you closer, Ron.
Where do you want your sonars, or did you get it?
I got it. I think it's okay.
Guess I'll just have to keep all of my fish reference tabs open.
I'm gonna get close, Ron. You know if you want flags.
Yeah, this is definitely a larger individual. So some of these.
They're in the eyeballs.
Some of these rockfish have lifespans, close to humans, but also more. I think I read somewhere age estimates of closer to 200 years. But
he's got something on his face there.
Oh yeah, a little amphipod, I think.
I'll just try to hold
a little bit of damage too, maybe underneath. Okay. Looks like on this kind of cheek part under the eye. Hard to say. Ooh.
Staring contest.
Wow, look at the detail on that eye.
Yeah, just stunning.
Levi must be piloting. He's our eye guy.
Eyeballs.
You can see the spines on,
Roger.
kind of decorating all
left as you zoom out.
Different layers of this fish's head and the dorsal fins.
Keep rotating.
Understood.
And revealed.
Nice.
Lasers one more time.
Roger, lasers are up. Let me rotate down to splash them
squid.
on the subject there. They're on it.
So we'll definitely get some a fish biologist to look at these and confirm, but my tentative ID is a yellow eye rockfish, definitely in the genus Sebastes, but I may be a Sebastes ruberimus.
What's over here on the left? Tall.
Ooh.
Coral on a rock. That
It's
boulder in the
looking
lower
very
left.
coral-like to me.
Yeah. Look at my sound.
And the substrate is getting a little rockier all of a sudden.
I was going to say, it's a good sign when we see our first coral on our first hard substrate.
Yeah,
There
that's a great sign, and it's a pretty big one. This is the one that I've been keeping an eye out for in this area. This is a Primnoa pacifica, commonly known as the Pacific red tree coral, even though the ones that we've been seeing are a little bit paler, kind of a light orange or even a white color.
Okay.
All right, let's go for maybe the tip polyps.
Lights at,
Nope, they're still 30%. They
bring
want to
them up
turn them on.
another 20. There we go.
Beautiful close-up, so we can see the
150.
individual polyps, each of those little, little units sticking out off of the main stem of this coral are the polyps. Each of those kind of has the same basic structure as a individual anemone. So they've got the tentacles and then kind of a neck, but these are colonies, so they're all attached to the same skeleton, and you can see they're covered in these really hard, they're called sclerites, so like skeletal elements that are made out of calcium carbonate and add some structure and I would guess protection to the polyps.
Roger.
And you can see why we're interested in these corals as habitat forming structures. So they, provide a lot of height off of the seafloor as well as complexity. So you can imagine it'd be a great place for smaller fish or invertebrates to hide.
Okay. Full wide.
And lots of more of those Henricea, I think, sea stars around the bottom and encrusting sponges or tunicates. Very cool to see such a kind of a larger individual just right at the base in a pretty highly sedimented area. So these corals will need some area of clean rock in order to settle and start out their life. So it's very cool to see one right here.
Okay, my rotator is back.
More white objects ahead.
52.
Just going to turn starboard and look at some rocks here.
Squid in the center.
So this gentle rocky.
So you can see on high pack there is a nose or an outcropping to your
Ooh,
port.
cute little squid.
I think you're starting to see some of the
We've had a couple of these identified by experts afterwards, reviewing the video. Some of those are in the genus Gonatus.
I think our next move should be back facing uphill about 215.
Just covered a highly sedimented area, but at least clean enough to provide an attachment point for some of these little tube-shaped sponges. Probably in the genus Auletta. And this kind of branching sponge down there. Yeah, zoom would be great.
Lights at 30.
You can take it.
Ooh, very cool. I don't know this one. I think a Dima sponge of some kind. With lots of little brittle star legs wrapped around it, and kind of can barely see parts of their central discs. They're kind of entwined in this really thinly branching sponge.
Go full screen.
Lots of hydroids and I think some little branched bryozoans, but they're a few different sponges in this view.
215, somewhere around there.
Video's clear.
I'll lateral over to starboard with you.
All right,
Fernando.
I'll just follow.
Very cool.
You come over.
Thanks for that zoom.
Sit 215.
Yeah, 215.
Unless you guys prefer something else based on what you're seeing.
The pink brittle stars that we're seeing are all Ophiophilus in the genus Ophiophilus.
I'm going to move over this way.
Yeah, I'm rotating as well.
Yeah, watch it call out if you want to zoom on anything you see.
No, I think we'll just document some of these bigger ones. This is great,
Yep.
but I don't need a zoom.
Understood.
Lasers on.
Lasers are back out there.
Do you want me to turn off lights?
Yes, please.
That's exciting.
Okay,
It's always great
center
to see
on
some of those
215.
larger individuals.
Rotator at 52.
Big anemones.
On the dive
above
yesterday, we
lasers.
had quite a few sightings, but most of them I think were under 20 centimeters, so
Let me know when
maybe
you guys are ready for that move.
some
I
new
think we're
recruitment.
good.
20 meters, Fernando?
215.
I see 3 of these
Yeah, either way.
pinkish sea urchins. Actually, a quick snap zoom
Oh,
of those
yeah.
would be nice.
Let's do the urchin. Lower urchin.
50%
15
lights.
at 215.
Sounds good. Ready.
Bridge, RV nav. Can I please get a ship move? 15 meters, bearing
Beautiful.
215
I think I
degrees at
remember
decimal 2.
hearing that these were all likely Strongylocentris, but I'm hoping our echinoderm experts will
That's a good
pipe in
copy.
to confirm. There we go, Strongylocentris
Take more
fragilis.
zoom, Roland. Whatever.
Max zoom.
Yeah, I'm
Very
super
cool.
close
Look
to
at all of the different kind of red and
Alright.
white
I don't have
spines
visibility
on
of the
this sea
tether,
urchin.
so don't get too far out.
kind of slowly
Move is in.
moving around.
Copy.
That's great,
Coming
thank you.
down a little bit. Delta's 14. Rotator is at 46.
And a little thorny
One of
head
those orange worms above that thorny head.
rockfish, also kind of
Roger,
hiding down there. That's great.
Just gonna explore geography real quick. That's the direction we're going. Now I'm gonna look a little bit to starboard. Yeah, okay. Alright, the view looks good. Alright, coming back. I'm gonna go for the snap the fish rolling.
You ready?
I think we're good. We can continue up this feature.
Okay.
We've had some good thorny head zooms.
Okay.
I think I'm pretty confident.
Push ahead if you want.
Ready? Try to get through some of this prop wash here. Onward and forward and upward. Here we go.
Seeing more of these little arrow worms or chetignaths in the water column kind of flickering. Show up a little more in the sediment. And there's a bit of a plume. Can really see them.
Be able to get through this dust cloud pretty soon here. There we go.
So after that little ledgy outcrop, I think it's going to flatten out a little bit, judging from the sonar, but maybe what we're starting to see is one of those kind of similar to what we expect to see as we head on up, as alternating steeper cliffy slopes and flatter areas than above them.
All right, a little steeper here, coming out.
Yep. Coming
But,
up with you.
well, this is pretty steep.
Delta 15.
Rock right here that's in front of us.
Wow, it's just covered. There's a lot more of this sediment maybe that's sticking around. There's a nice boot sponge, kind of a tube-shaped one. in the family Rosselidae.
No bridge?
Hard to tell the exact
Roger.
species from imagery, but there are probably six or seven or
It's
more
complete.
different species it could be. Oh, another flatfish.
We'll keep going.
Some encrusting sponge, some of the maybe bright, whitish or yellowish. ROV, could we zoom in on that funny thing on a stalk to the left?
Yeah. Make my way there.
I don't really know what that is, so it's a good reason to get a zoom.
All right, video, come in.
Lights at 30%.
Oh, wow, that's really weird. Huh.
Almost looks like a pineapple.
Yeah. So a sponge of some kind. You can see the osculum at the top, and
Bring the lights up to 60%.
oh, this looks like there's something on it. A little scale worm, maybe.
Try to get closer, Roll.
This is close.
Okay.
And
Just
it
need
looks
to
like
light the
there are
bottom of it.
shrimp on it, kind of hiding out between these bumps. But I'm not. Yeah, it could be a juvenile of one of the larger sponges we're seeing. But this it has a very distinct stalk.
Huh.
Very cool. I think we'll get nice. This really excellent zoom to kind of document some of the identifying features we're seeing, and maybe we'll keep an eye out for more.
You could bring the swing arms up a little bit, Fernando. Maybe like 45 degrees, yeah.
The lowers?
Yeah, lower.
Wow, very cool.
Thanks for video.
Okay, that's a new one for me. I love it. So yeah, because of its size, it could be a smaller individual of some of the larger sponges that we were seeing, yesterday, and maybe further up on this dive.
Okay.
So
Does that
keep an eye
lumpy
out.
look typical of younger sponges, or?
I mean, usually the younger sponges look like mini versions of the larger ones, so it could be maybe one of these
Never seen those worms before, and there are two more.
No,
It's like
it
four.
wouldn't. Those little tubular shaped sponges
Well, and they've
don't
been all over
end
the
up looking
place,
like this.
the
So
pink ones.
no, that one's really cool.
Double
RV, could we look
pictures
at that
now.
little funnel shaped sponge just below to the right? You got it.
You can snap it. Yeah.
Just a snap.
White funnel. Do you need a closer, longer
No,
look,
that's
watch rate, or
that's
is
great.
this a
Just
good
as we
idea?
can get a focus on
Oh,
the.
that's neat.
Yeah, that's great. Thank you.
Understood.
Yeah, I think that looks to me like it could be a little mini Conilasma, one of the glass sponges, maybe. It's very small.
Upper right, I saw something interesting. I'm not sure what
Cool
it
to see
was.
some, smaller individuals. And then lots of those little tube-shaped ones, the Owletta
Is that
sponge.
the nudibranch looking thing?
But it's got legs.
It's one of those
Nudibranch
spiky
with star,
sea cucumbers,
brittle star next to it.
Alright. Slowly tip it
kind of a related species to that California sea cucumber we were talking about yesterday. And some of those little branching structures that were passing by, the white ones are hydrocorals.
Keep coming up.
Yep.
Just gonna take a peek.
There's just so much life on these rocks.
Yeah, there's a lot going on. It's nice that there's a little less sedimentation on the really almost vertical parts, so you can see
Yeah,
a
we're
bit
getting
more about
pretty
what's
steep
going
here.
on.
Yeah.
Lights, fogs are off, 0%.
Alright, I'm just gonna look to starboard, to the northwest. Ooh,
Oh,
What
a
is
couple
that?
of big rockfish.
Lasers on.
they're out there. I just
Yeah, there's
Double
at
flashing
least maybe two.
See 'em?
Oh, they're on them. Lasers are on them, right?
Yep.
There they are. Bingo.
And a little tiny fish. Oh, those are so cute.
We'll let it go. We got
That is huge.
Yeah,
I
possibly
think
a
that'll
yellow
be
eye
long.
rockfish. And a nice big one, too.
I'm going to proceed up. Follow this up. Oh, there's one other fish above me, too.
Yeah, I think
The
it's
striped
on
one.
a ledge.
Yeah, this one looks a little different than the other ones we've been seeing.
We were seeing some of these kind of
getting to 40
banded or
degrees again.
striped ones
Maybe another move would be
a couple
good.
of dives ago.
5 or
There's one species called the
maybe.
red
Yeah, that
banded
should be fine.
rockfish,
Okay.
but I think there are others that sometimes have
Roger, 10
banding
meters,
like this.
215.
So we'll just say genus Sebastes for
job,
now,
Matt.
but there's quite a few. That one looks different too.
He does,
Another
yeah.
move,
They'll
please. 10 meters at 215.
Some of them look
decimal
very
two.
different as juveniles than the adults. They have really different patterns, so
That's
it's
a good
even
copy.
hard to
Thank you,
tell
Bridge.
smaller versions
Going for stick
from the
cam on that
adult
fish.
species.
Be as close as I can. As close as I can get you, still can. Get lasers. Okay. Alright. We got a move underway. I'll keep going up. Fish safe distance back.
Yep.
Beautiful. That rockfish is just kind of hanging out right at the base of one of these larger, structures. And it's just covered in a lot of different kinds of sponges. It's kind of orange and yellow encrusting sponges, definitely different species based on the patterning you can see. And lots of these, the little white branching structures are kind of pinkish that you can see are hydrocorals. They're kind of like little mini versions of the other corals. They don't seem to get as big, but they have really intricate branching, and their skeletons are like really hard, kind of solid structures.
While we're here, see what's on this rock, and then we'll keep going.
Wow.
Hold that for a second and focus around.
You can see an anemone peeking out down there, and just covered in brachiopods and these little tube-shaped sponges. A lot of those branching structures underneath could be hydroids, worm tubes, all sorts of stuff, just covering every inch of that rock. Bad news for the geology. Oh, look, there's a four-armed sea star too.
Yeah, a little bit of exposure on that face. If we could zoom in a little bit on that lower part of that outcrop or face.
Over on the left?
Right.
Yeah, just heading over to the, kind of lower left center. Or, I'm sorry, lower right center.
I think right where that four-armed sea star is.
Thank you. Possibly, yeah.
That's a good spot. Get a
I see some kind of roundish look to maybe the layering.
Come in partial, no driving. Hold that. Is there a specific?
Yeah, that's good. And a little bit to the left of here on more of the open face after everyone gets a good look at the sea star.
Okay, right there.
Yeah, okay.
What
really
sea star?
hard to see any rock.
No, not seeing much rock. Yeah.
Wow. Hardly any.
Keep going. Thanks, video.
Yeah, we can keep going.
I'm thinking it looks like we're going to shortly traverse over a less steep area and then get to a more steep area. So
Right.
we're looking for rock for you, Jamie.
Yeah. Well, this is rocky.
Slope off to starboard.
Yeah, there certainly are lots of rocks here, but
We're a little late to the party.
Yeah.
I think so. But I
Do
don't
you want a
know.
close up of the fish or keep going?
We're good. I think keep going.
Understood. We'll proceed.
Yeah, if we'd been here 12 or 14,000 years ago, we could have seen them freshly scraped by the glaciers and get a real good view of them. But Everything's taken over since then.
Snooze you
Oh,
lose.
that's a sponge.
Could we snap on this sponge up ahead?
Yep.
And also that orange thing could be a big anemone.
This one, this big
Yeah.
ball right here.
What is that?
Stand by.
Like a really large anemone retracted, or a different
that
kind
a
of sponge?
Sea star
Something
feeding
good.
in the bottom left too.
Oh, yeah.
Move
Oh,
it.
totally.
Yellow.
Copy.
There's a lot going on right here.
What is this thing?
Trying to get around the other side here.
Yeah, thank you.
How's
Oh,
that port side
okay.
Look, Bobby.
I think I see
Clear.
an oscule, an opening,
that.
so maybe it is a sponge.
Keep come in video on this.
Lights at 40%.
Oh, no, there we go. Anemone.
Oh, wow.
Wow.
I
Just
did
all
not expect
retracted in
that.
there?
Me neither. Yeah, you see a few of the tentacles poking out, but almost the whole thing is sort of
I'll try to
balled
hold
up
this
and
shot long.
retracted. It's very cool to see this this shrimp associated with it. It looks like it's maybe maybe just feeding on the organic matter that's attached to it.
He's here doing just what we're doing, trying to figure out what this is. And
Yeah.
he just determined it's an anemone.
Also curious, yeah. So
Just
it's a
slowly
huge
backing
anemone
away.
too. Yeah, like I changed my
All
mind.
right, I'm gonna hop over to that sponge up there.
Okay. Laser's clear.
Understood.
Is it
Yeah, I'd
falling?
love to get an idea of what this one is. Again, some of these larger sponges are important habitat forming
Still have the light.
structures.
Yep. Just got these lights. Keep them.
How do you feel, Fernando? Do you want another
And-
5 or 10?
Sure.
Yeah, hard
Yeah, we can
hard
go
to tell what this
five.
one
Oh,
is.
10, that's fine.
10? All right.
It's definitely
yeah,
moving around
should be
quite a
good.
bit in the current.
Another 10 coming.
Copy.
Kind of gives me the impression that it's a bit more. Oh, someone got bingo.
Great job.
Maybe more of a demo sponge. It doesn't have the kind
Yes,
of
can we
rigid
please
skeleton
repeat
that
last move, 10 meters,
I would
215?
expect a glass sponge in that morphology to have. But take a closer look at the
That's
video.
good
There's
copy.
another
Thank you,
boot
Bridge.
sponge. And of course, every inch of this rock face is just covered with something.
Okay.
Move
And
is
we,
in.
we see all these
Copy. Moving.
bivalves, which are, as far as I know, exclusively been brachiopods, and not clams or mussels. Are clams or mussels things we'd see in shallower water? And this is more of a brachiopod depth, or is it more environmental
Gonna turn on
than
one
that?
just to see if I can
That's a
get
good
better
question. I
look at
don't
the
know a lot
tether.
about
Don't need to change
brachiopod
anything video.
distribution. Yeah, I definitely do see like different kinds of scallops, and of course the chemosynthetic
Fog
clams
lights
that we
clear.
know from seep areas. So every different species has their own kind of depth band, and this is not one that I've studied. I'm kind of surprised to see them in such high abundance, but oh, here are a couple of coral skeletons.
I'm gonna go for the darker.
Yeah, could we have a
foreground
quick
one.
zoom on the
Let's come in on Riddle Stars on.
skeleton? Looks like it's covered in brittle stars. Wow.
Is this the skeleton of a black coral, or it just happens to be dark color?
I think it's a skeleton from one of the primnoas, just judging by the size and the shape. And they kind of
Probably
get,
be advised I ranged you out on blue
kind of a golden color that turns a little bit
Steep slope
darker
is
over
about 50 meters away
over time, I think.
on high pack. Don't see it on blue view.
Very cool. So we know that there are different kinds of animals that predate on corals, sea stars, and nudibranchs. So possibly that's what happened, and then the skeleton is all that remains. Wow, this is a huge rockfish.
This is right where I'm going.
Hello.
You know what I'm about to say?
Another one of the,
Yep.
you know, with the red banding.
Sorry, Fernando.
I have a feeling we might be seeing this rockfish's eye.
Yeah. You know what to do.
Did you talk to me? Did you?
Go ahead.
Yeah.
Thank you.
Saying I.
So
Look for your
again,
umbilical
yeah, some of these rockfish have a
bring it out.
lifespan similar to humans, or maybe
Yeah,
even
I
a bit
have it right here.
longer.
See in the bottom count.
We're good.
And I also, wow, on the eye.
Oh,
Okay.
I don't like that.
Guess it doesn't seem to be too bothered.
It's not even blinking.
One thing I also really like that I learned about them is that certain species like to stay in the same area. So divers have documented seeing the same individual rockfish close to the same rock years apart. So revisiting 2 to 4 years the exact same site, and they've seen the exact same individual rockfish. So they have a really high preference for kind of staying in the same area, at least a few species that are identifiable as individuals. Often it's hard to tell. Some have more distinct markings than others.
Should be 180.
There's a nice rock.
D2's current heading.
The really big hydrocoral on it.
We're stopped right now.
And another one of these large sponges.
I think Sirius is still swinging just a hair.
After this.
If you could get a snap on this sponge.
Yep, let's frame it up.
Thank you.
Is there a certain part of the sponge or just the whole thing?
Just like the tissue, those, yeah, just kind of the pattern.
Understood.
If we can get nice and close.
Get a full shot here rolling, and then I'm going to go in on the upper portion.
Okay.
All right, go for upper portion. Boom.
Ooh.
Oh, lights falling, or you're
beautiful.
I'm good with it.
You're good with it. Nice.
So I'll be able to look at this. You can see definitely lines of these spicules, the skeletal structures radiating out kind of from the center, and creating almost like a little bit of a honeycomb pattern. If it's possible to see the attachment point,
Pivot for the attachment point.
just a quick look.
Is that a yes or no? Okay. Sounds good.
All right. I'm going to try to pivot to find the attachment point here.
So also on the rock that's about to be revealed.
Just a quick look could really help with this ID. Oh yeah, so it's pretty different. Kind of has the same general shape of like the conilasma sponges, really large glass sponges we were seeing, but I'm thinking this might be something else.
Is that helpful?
It is helpful. Still don't quite know what it is, but it's great to have this angle. Okay, that's great. Thank you.
Roger. Thanks, Roland.
And I think current's picking up.
Yeah. What's the tides doing?
Okay, so after this, do you want to move to your port, and then I'll rotate with you until we get to 180?
Yeah, you can turn.
Copy. All right. I'll start turning. Then after that, maybe we can get a move in. So that's 185.
So I think we're now kind of just up above that first cliffy looking outcrop. Now my guess is, and guessing's always a bad thing to do in geology, especially with little or no information.
No, I do it all the time.
But we're perhaps looking at kind of sticky little flows, and each one of these cliffs makes up a one of these kind of restricted flow units that are, as we can see in the visual, perhaps several to 5 or 10 meters high, and tend to pinch out to either side, making little bodies
20 meters.
50 to 100 meters across.
Bridge over
perhaps
now.
as little tongues of flow
Can I get a
units.
ship move, please, of 20 meters bearing 180 due south? Good copy. Thank you, Bridge.
I'm just waiting for some tether. Watch it. Nice. Hello, Kusma here.
Assuming it
Is that
is 10:15
Chris?
local, we're at low tide right now.
Did I hear something? Maybe not.
I didn't.
I thought I heard something.
Okay.
I think you heard pilot.
That was probably it.
What's that?
I think you confused our back row.
What did I say?
Hello, Chris Ma here.
Oh, they thought I was Chris Ma.
Yes,
No,
they did.
I was waiting for him to call in.
Hello. He did pipe in with an ID.
Do I
It's
sound
that
like Chris
Sea Star Saramaster.
I had a distinctive voice. I don't think. Sorry for the confusion. I got to work on the doo-doo noise first.
Oh, that was good.
Hello.
Upper left. What is
I'm gonna go for the.
curly thing?
I'm gonna go for the red guy here.
All right.
Sit down close.
All
Make
right.
it pretty.
I'll be getting tethered in just a moment here. Watch lead, but I'll get one more snap for us. You can snap in on this little guy.
Do you guys feel the current has changed?
I don't know.
Bright orange anemone.
My tether's still on that side, so I don't
Yeah,
think it has
I think one of the ones we were
I'm
confusing
wondering if
for
it's
cup
coming,
corals
you know,
in
slowing down, and
our,
that's why
our
visibility
first
is
dive in this
bad.
area.
Tides say that we're at slack, full low
And some
right now, 10:18.
really thinly branching, probably little
So if I don't
bryozoans.
see the tether
See
at
the
all
fan
in
down
your
there,
cameras
kind of
except the aft cam,
alternating
should
red
I
and white banded
keep
fan
it a
of
little
a
bit tighter,
tube worm.
or
Okay. Yeah.
what should
I
we
think
do?
you're doing perfect, Fernando.
A little tiny squid, a couple of
You
them.
just keep like a 14-15 delta, and then if you're at 45 on your camera, I'm just gonna kind of stay in the middle and a little far out. I think that'll be.
Keep it, yeah. Keeping it a little bit tighter is
That
probably
looked
good.
like it.
The football
Maybe.
should
Well,
be doing their job.
I'll
keeping
guess it
up
was a
away from
rockfish, but he looked a little bit gray.
I think we're doing like perfect, Fernando. It's going straight back.
Ooh, there are a couple of
yeah, at least.
Nice.
Oh, I can see it
rockfish
right there in
and
the
rockfish
Yeah,
relatives
the HD
that are
down
grayish.
cam now.
It's a big dead skeleton, huh?
Yeah. Yeah, lots of encrusting fauna.
Frame it up.
Yeah, a quick zoom of this would be cool.
Alright,
I
let's
think
go
it's
for the,
covered in brittle stars
go for the bottom
and
first. Come in.
an anemone. No. Yeah.
Look at all those brittle stars.
Wow. See some shrimp.
I want to come up.
And I
when
think
you're
higher
ready.
up, one of those
Alright, here we go.
gastropods, Calliostoma.
Super dead here, huh? I wonder if we'll see a big live one. That big stringer.
Wow.
Oh.
Yeah, so this was a pretty old, large individual.
I'm clear when you're clear, video. We can keep going.
Could we get the lasers on the base just
Absolutely.
to get a bit of a
Yep.
size estimate?
There you go.
That's great.
All right. This looks exciting over here.
Yeah, covered in those little hydrocorals, I think. Most of these are in the genus Stylaster. But yeah, they're called hydrocorals because they are hydrozoans, so they're more closely related to those little branching, bushy things that we see closer to the rocks than they are to the
Go ahead, Bridge.
octocorals that we've
Roger.
been seeing, like Primnoa.
Ship is stopped.
Copy.
These rocks. I'm gonna go for still cam video too. On this guy.
Here's a little bit of
wow.
a forest we're coming up into.
Yeah, but really interesting, like missing all of the tissue at the bottom.
Yeah.
So something looks like it's maybe
coral.
predating on it from the bottom
Left
up, and
to
I
this
think
one
I see
also another one.
those little
go for,
Calliostoma gastropods,
Silken
little
fish
snails that might be
Lights
the
up.
culprit. Also, a lot of I think Hipasteria
Alright,
sea
let's
stars
close
at the
that
base
gap
of
too.
each of these.
Oh, yes.
And a big
About
nudibranch.
30.
It's kind of a
Free for all.
yeah, it is. Or maybe the Calliostoma are taking advantage of some space that's been cleared
No,
by
you're
something
good.
larger.
Still good.
It's hard to say, but there's these
Yeah, you can zoom in.
beautiful
Go.
corals are really
All the way.
being munched on by all sorts of
We'll
things.
do a twofer here. This one's mostly dead, huh? It's got a little bit left. I think he's good.
Yeah, so really beautiful
I'm gonna go for the top
shrimp
one.
Slowly.
kind of hiding. Whoa.
That tight zoom.
Wow. So you can see the same kinds of polyps that we saw in the
I'll
younger
hold this.
individual. This one's a nice deep orange color, but it looks like the rest of this tissue
come
maybe
down.
isn't long for this world, judging by the fact that it's missing so much near the bottom.
Slow
And these
rotate
polyps can't fully retract. You can imagine being covered in these hard calcium carbonate scales almost, that it limits the amount that they can retract into the tissue versus something like the bubblegum corals we were seeing earlier on an earlier dive, where the tentacles and the polyps can completely retract.
I guess I'll just go to the bottom.
Wow.
No? Okay.
Look at all this.
I might go to the
Left.
gelatinous guy on the left on that.
Is that a
Sure,
nudibranch?
yeah, really big
From
Tritonia
this angle, we can also
nudibranch
see the underside of the
and a
star.
sea star. So I think this coral is being
Fernando, another 15.
attacked,
Oh,
viciously attacked by a couple of things.
It's gonna take me a minute to do that.
Of course, there's some nice new recruitment
Okay.
growing just to the left.
I'm
So
gonna
I
stop,
think
I'm gonna stop for this guy.
Sounds good.
this is a part of the natural cycle.
So how fast do these
Pretty
grow? It's surprising nobody's thought yet to go over and munch on that younger
Please
one.
get, another move.
Yeah,
10
I would
meters,
imagine that it would
bearing
take,
180.
they seem very slow moving, so I think it would take a
Alright,
long
good.
time to go up one. This one here is just starting.
Mm-hmm.
Good copy. Thank you, Bridge.
Missing the sea star on the upper right.
The growth rate, and there's a little new growth at the bottom there,
Oh,
I would
interesting.
say. Yeah, so maybe this nudibranch either missed that part, or it's a little bit of new growth. Sometimes you do see younger corals growing at the base of much older skeletons.
Another keep coming up.
Wow, yeah, this nudibranch is doing a great job of clearing the
How's that
tissue
star?
and a sea star.
And the sea star, wow.
Yeah. And the Calliostoma gastropods.
Corals have a lot of predators.
So I did look into it a little bit of estimates from, I think, radiocarbon dating of,
Interesting how the tips
you
are
know,
all
in order
eaten up.
to do that, obviously you need the
the
base of
snail.
the coral.
Yeah.
Mm-hmm.
And so
thanks, babe.
the estimates I saw kind of varied really widely. One of them said, like, the conclusion I read was under 300 years.
Oh.
And then I think kind of ground
video.
truthing it with
Wanna
other
proceed?
methods, put it in the
back
100-year
out
range
there
for the
and back.
individual coral that they looked at.
Onward and upward here.
Sea stars on every coral.
Yeah, it sure is. Look at that.
Wow. Yeah.
So-
that one there.
So in between these big cliffy exposures, you can see in the bathymetry and in the 3D model, if you're looking at Cam 3, are these more flatter areas that may be underlain by more thinner bedded flows that don't make these big cliffs, instead make more benches.
I think we should be able to get to 15 now. He's farther than 15.
There's one that's been
Wow, completely
eaten.
denuded, I think is
Denuded, okay.
a word I see sometimes.
Denuded
more-
by nudibranchs.
There you
Trying a
go.
little more disturbing now.
There's still a move underway, guys.
Oh, okay.
Yeah. But correct, the next one will be back to 215.
Copy.
That'll send you straight uphill to waypoint 2.
Oh,
Look at
a
the
really
size
big,
of this guy.
yeah, boot sponge. So a couple of different species that these completely kind of clean ones, like devoid of sediment on either surface. There's a few different genera I think that they could be.
Go ahead, Bridge.
They're all in the
Roger.
family Rosselidae.
Lights at 30%.
So we'll call these
We'll just get a full
They've
size.
been
Rosellid
at
sponge.
25 the whole time, sorry.
Ship
Okay,
stopped.
Increase another 20.
There, oh, that's about 50.
Is that good, Ron?
Yep.
I'll just hold this shot. I don't know how much detail we
Copy, nav, ship stopped.
Guess look at the top. Snap the top.
This one has quite a
When
bit
he's done
more
with this one,
of
Copilot,
the
the next move will be
debris,
back at
water column
215.
debris,
Copy 215.
stuck just to the very lip.
Yeah, just around the lip or the outside of this osculum. It's very interesting. This sponge has some kind of mechanism to keep the tissue clear on either side, and I think it has a tuft of longer spicules, those silica-based skeletal elements of the sponge along the top that maybe the cleaning action can't reach. But then other sponges in this group only seem to keep one side clean, usually the inside in rhabdocalyptus, so they're covered in sediment on the outside. And then another one that has sediment on both sides and doesn't seem to mind either.
All right.
So they're interesting,
So
and they
big
can
too.
help us
You want a still shot of this?
identify them. But of course, you need to
Here.
have a
That one.
really good in situ image, a tissue sample, an ID, and then be able to kind of relate that back
Video
to
wants a
features you're
good shot
seeing.
of that
Here's
star,
another, I think, Hippasteria
Okay, do you
sea
want
star
frogs? Oh,
especially the 2 stars. that are
predating
behind
on this
this.
Primnoa pacifica. Or grazing, depends on who you're cheering for.
Taking a coral down in the back.
It's about as close as I can get.
Yeah, it's easy to pick sides, but sea stars need to eat too.
This is true. This is true. You can see a few of these polyps are kind of looking a little more limp and less active than some of the other ones. The ones in the foreground you can see are still,
You got fog
you
lights
know,
on,
at least
right?
mostly
Yep.
extended, and these ones are, I think, as retracted as a
down to
Primnoa
the
polyp
star.
can get.
Roger, going down a star.
So it looks like these ones are out a little bit more, maybe possibly feeding.
Where is it?
Where
There
did you
it
go?
is.
Yeah. Oh, that's wild. Very cool angle. And the little snail, you can see their little eyes.
Very interesting texture on the sea star.
Wow,
Little,
yeah.
It's got like dots.
almost grains of rice, wild rice looking
It does, yeah.
texture.
180.
Oh, let's wait here for a second.
Yeah,
They
let's
look
just
like
wait
they
until-
kind
won't
of are in
affect you,
various
right?
positions of
Alright, we'll wait.
being
Yeah,
open
we'll
or closed, which is interesting. And little kind of blunt spines in
take a look at the
between.
coral
Trying to preemptively
background
get you some tether. But
with the 2 stars that took it down.
Oh, maybe because he's going out to the stars, right?
It's okay, I'm gonna get
The
you closer. And then we'll
pedicellariae of the sea star, perhaps.
Clear with this.
This is a really beautiful, you know, vibrant
another
orange
shot of
color
this
on
planet?
this
yeah, I'm doing this
Primnoa.
Is that good? R3 survey too? You good with that? Or you want more? Alright.
I think I
Well,
can
there
see
you go.
little eyes from some
Thanks, Steve.
shrimp hiding out in there.
Alright.
Hey, this is Chris Ma.
Oh, hello.
Hey, you have some really beautiful video here, and even though we've seen similar types of predation in like the Hawaiian area, I don't know that I've ever actually seen them. I've read about these in the literature. I think the paper by Krieger and Wing used to document these as sort of SPA, but I've never actually seen them myself. And so this has been all very sort of really wonderful video footage. The things that you were looking at earlier, all of the texture, and this is something that keeps on coming up that I haven't had an opportunity to really discuss at length. But when you guys are focusing on the surface and you see all of those, for lack of a better word, the things that look like little lips or little eyeballs or whatever, those are called pedicelaria. And so the surface of these sea stars is covered with them. And it's not entirely clear what they do because they're on the bottom surface as well.
Okay.
And when we saw them in Hawaii, they would open and shut. I mean, they looked like little jaws. And so I'm not entirely clear. It's not entirely clear to me why they do that. Why? So I mean, at first I thought maybe there's some kind of battle going on with the sea star and the stinging cells on the coral. But, obviously there's a whole bunch of these pedicelaria that are facing away from the animal, which would imply that maybe they're used as a defense against predators on the surface. But when we see them, there aren't any little, like, crustaceans or anything around on the surface. So, it's a big mystery to me. I don't know what they're for. I mean, that's why I always relish a close-up look, because hopefully one of these will provide some kind of insight, as to, what is possibly happening. Because, I mean, it's occurred to me, for example, the possibility that, so one of the things that I have seen in other video is that those soelaster that we see so frequently will, while these, will just roll over Hippisteria and just eat them. They have this notoriously complicated behavioral thing where they're that was called in 1967, I think. There's a paper that a whole bunch of jolly marine biologists called the 13-arm hammerlock. And the soelaster sea stars do this to other sea stars where they just kind of wrestle it down and then proceed to eat it, which sounds kind of funny, but is pretty horrifying, one imagines.
Yes,
but I've seen video of, or at least imagery of soelaster feeding on Hippisteria. So maybe they're literally they've just evolved this, this defensive structure to prevent other sea stars from getting on top of them. I don't know, because we don't have any. I've never seen the Hawaiian ones with soelaster present. And Hippisteria is widely occurring. So, I don't know if there's just sort of a leftover thing, if different species have more of a, have more of a reaction, and this is just a carryover. But every time we see them, I get excited, and certainly, like, we've been seeing a lot of these, and every time we get a close-up, there's excellent documentation of these structures. So keep up the good work. And Roland and all of the GFOE people, please, whenever we see these feedings, I mean, I know this would be boring for everyone else, but nothing would make me happier but to watch more Hippisteria eating coral all day. So, there we are. But I see here more Primnoa with the post-Hippisteria handiwork. Anyway, I'll let you go. Thanks for giving me a couple of moments to explain my current love affair with goniasterids. I'll catch up with you guys later. Bye-bye.
That's
Thanks,
great.
Chris.
Yeah, thank you for that.
Sure.
It's always really fascinating. Especially, I love hearing what is stumping other scientists in their field. So now I am motivated to zoom in on Hippisteria too. So we can kind of try to imagine what the function of those features that we can see might be. That's great. I'm good to kind of continue. I think we've seen a few of these Primnoa with a bit of Hippasteria related damage.
Copy.
Cool to get an idea of general counts as we go along, and maybe someone in the future will measure the level of damage to each one. I know that in our annotations we always note when there's a damaged or a dead coral. It's important information for us.
Not officially, but yeah, it's done.
And a follow-up from Chris Ma, most of these look like Hippasteria heathi, or heathi. That's the species of Hippasteria that we're looking at. We've seen a few different species of them so far on this survey.
10?
Yeah, 10.
It's a little Ceramaster who just passed over.
5 on the rotator.
Go ahead, Bridge. Roger that. Can we actually repeat the move, please? 10 meters, 2 and 5.
There's another one, a fairly healthy looking coral with a,
That's a good copy.
big
Thank you, Bridge.
Hippasteria sticking to it. I think some of these other sea stars could be with the really strong, I don't know, kind of outline. To me, look like Ceramaster or cookie stars.
What is this? Oh, it's a fish.
I think that's that sculpin, maybe?
Can you snap it?
Mm-hmm.
There we go. I really like these little fish.
Caught my eye.
There are a couple of ones. The species we were given earlier is the dark fin sculpin. Could be related to that one.
Clear.
Right. We just got to move in.
A couple of small sponges that a lot of them are demo sponges, the tubular-shaped ones, probably in the genus Auletta.
Understood.
But a couple that look like they could be juvenile glass sponges. We saw a couple of the reef-forming cloud sponges yesterday. So we know that they're in the area, and those
We're
could
looking
be
kind of
juveniles.
for a shoulder or kind of a flat spot, at which point we'll saddle over to waypoint 3.
Oh.
Wow, look at all when you look up there, the water column's just full of these little Ketognaths everywhere.
They love playing in the lights.
Oh yeah, they love the lights.
I think they do, yeah. A couple little shrimp swimming by, but lots and lots of Ketognaths or arrowworms.
Got steeper here. I'm going to come up. I can show you how steep it is.
Okay, ranging you back in on multi-beam.
So it looks like we're approaching that kind of vertical feature.
Right, a little steeper part.
Mm-hmm.
Perhaps a thicker flow. Now, in identifying these as volcanic rocks, remember that I'm extrapolating from exposures on shore where volcanic rocks were mapped, but those are maybe a couple kilometers away. So, but these do kind of have the look of kind of tabular.
Go ahead, Bridge. Roger.
Volcanic flow units more than sedimentary rocks. Again, these are Paleozoic aged rocks, probably of Ordovician and Silurian age, or about 400 to 500 million years old, representing kind of a volcanic arc terrain, maybe something like the modern day Aleutian chain. So we can expect a lot of variation, not just volcanic rocks. We could even be having little intervals of sedimentary rocks that were laid down in between the flow units. It's hard to tell from these exposures with all the cover, but we'll certainly try to get a piece of something loose when we come across something that looks appropriate. Yeah, maybe even one of those.
It's always so hard to tell.
Yeah, it is.
You know, in the past.
Do you want me to try to sample one of these rocks? Watch they.
Well, they might be a little bit on the big. That
the left
bigger one is.
Or this little right one would work. Do you want to try it or not?
Nah, let's. Yeah, let's keep looking, although I don't know if we'll see things much better, but we certainly have plenty of exposures in front of us, so.
Proceeding. Got a little
Ooh, beautiful red squid. Wow.
Oh, ink.
A little bit of ink. Cool. So a lot of those squid have been in the genus Gonatus, but we'll definitely have to have our cephalopod experts review.
All right, I'm going to keep going.
Copy.
Co-pilot, I'm wondering if you might get a little bit better visibility if you bring that aux light down so it's not blinding us. I don't know.
That move's complete.
Yeah, that move is complete.
Well, that might not be a bad rock. Well, it's probably a little bit big.
Bridge,
and maybe attached.
That one?
Yeah, that's too big.
It's way too big, yeah.
Same move as the last.
I'm
10
attracted
meters,
to ones where I
215.
can actually see part of it.
Smaller one behind it, maybe. To the right? Well, hard to say.
Yeah, oh,
Good
those
copy.
are some
Thank you,
loose
Bridge.
ones. Maybe, yeah, any of those would be good. Kind of right in the center there, just below the lasers. Oh, and a little sculpin come over to see what we're looking at. I think some of those could be picked up and are loose.
Okay.
This is great. I'm also very interested in a rock sample from around here. There's so much to document and look at. This would be good for both of us.
So ROV, maybe, yeah, that one just to the right of the laser is grabbable, but any of those that look good to you?
Okay. I'll give him a poke. See through this.
Okay, hydraulics are up.
When you see just a single loose rock, there's always the possibility out here that it's a drop stone from that's possibly been dropped from an iceberg as the ice sheet broke up, and any sediment, boulders, sand, gravel, whatnot that it had picked up, then that gets dropped as it melts.
All right, we have wiggle.
But this looks like when you see more than one rock in a place, you suspect they're probably all locally derived.
Can I do a rock box, starboard rock box for this? That work?
Yeah.
All right.
Should try to look at it close.
You want to get extra lights on?
Lights are on at 30.
Come in. Is this a good rock?
That is that we think so.
All right.
How can you tell?
It's hard, has things attached to it, so that'll take a while to clean
It
off.
sure will.
Let's do starboard.
Copy.
Pilot, can you hear
I'm
video?
excited about it. We haven't had a good rock in a couple dives.
There we go.
I'm seeing,
know
I
when
think
you're
I
ready
see
for,
little skeleton
box
shrimp, those little caprellids. We'll see if they stick around for the ascent.
Can you tilt? There's the bottom of the rock. It's the
Do you think it
rock.
would fit in the bio box?
There we go. Yeah.
Yeah, I could fit in the bio.
Maybe, if you're okay with that.
No, that's fine.
Yeah, then we can kind of keep a hold of
I can zoom out video.
some
Go
of
for
the
stuff.
encrusting fauna that's there.
Okay.
It's a little solitary hydroid in
You
the
can zoom out
front.
video.
I think I saw one of the chitons that we were looking for as well.
Lights are clear.
Mr. Fernando, are you ready for drawer out? Wait, my craft's probably in the way of the drawer, so just give me 1 second.
Drawer out?
Yeah, 1 second.
Oh, it's up by you? Okay.
I'm going to go for bio box if you want.
Hold on then.
If you want to open up both sides. Alright, you can open the drawer.
Okay, hold on.
How's that ship moved in?
And of course, the
Let's wrap it up.
We're going
trachiopods
starboard, right?
attached to it, potentially in the genus
Pushing drawer out.
Go bridge.
Laqueus.
Roger.
Thanks for that identification. from Chris Ma. We've been seeing brachiopods everywhere, and not something that I've ever seen in great amounts, certainly not in the abundances that we have here. So we have a few references for identification. One of the scientists said we have collected a zoo.
Oh my! Already.
And that's not far off.
Starboard
I wonder
outboard.
how many different file are in there. Okay.
ID EX2306-D11-02G in the starboard outboard bio box.
You got a waypoint.
Copy 02G.
All right, here we go.
Was that outboard or inboard?
Starboard, outboard. I did say inboard at one point. Roll. Okay, here we go. We're taking off. Got more tether. I'm going up this steep section here.
Oh, wow.
Ship is stopped.
All right. Well, my day is done. We've got a rock.
Ah.
Coming up.
Okay, flatfish to the right.
Rock. It's about.
I mean, I'm also pretty happy. There's lots of biology on that rock. That'll keep us busy for quite some time. I think it's your first rock in 3 dives, isn't it?
That is.
Wow.
Yes.
Well, I hope it's a good rock.
Looks good to me. There may be some small Primnoa branching at the bottom of this feature.
I'm gonna snap it.
Oh, thanks. Yeah, actually. Great. Yeah, confirm. That's a Primnoa. Thank you.
All right. Onward and upward. Team. Here we go.
Ooh, can we zoom in on those corals right above the lasers? I think we have a cup coral, a big one. Yes, I think that's Desmophyllum.
Big cup corals. Those are big. All right, let me get close.
And if they are, I'd like to collect one if that's possible.
So you think it's a suction?
No, they're hard corals. We might have to break one off at the stem.
Understood.
Because I don't think we can collect that rock. But let's get a good zoom in on them first.
Understood.
Thank you.
All right, Vinny, coming in.
Wow, so somehow in the midst of all of this sediment and debris that's flowing through the marine snow, these cup corals have found an attachment point. These are Desmophyllum dianthus. They're related to Lophelia, the reef-forming cold water coral. You can see the skeleton of this is a hard calcium carbonate skeleton with all these different little divisions, these little plate-like structures, and then you can see it's covered in a thin layer of tissue and little tentacles coming out. So these are scleractinians, a stony coral.
Oh, neat.
And they are big, really big for a cup coral. Some of the largest that I've seen.
So I'm thinking, take the arm, grab them, bring them in front of the suction nozzle. And
the deep sea surveys that I've been on.
Watch lead pilot?
Yeah, watch lead.
Okay, if I grab them with the manipulator, can I present it to the suction nozzle and then suck it up, or are these stiff?
They're very hard, like stony corals. So I think you might have to try to use like one of the claws to snap it at the base, if that's possible, and then maybe feather it or use the suction to drop it in. I don't think it'll go. I doubt it will go in the suction.
Let's get lasers on.
They're sure out there.
I mean, I don't have to do much, just enough for you to open a box.
And hopefully there's a place for it to land on if it breaks off and not tumble down the cliff face.
Good point. We are very steep here.
Okay, so we're ready to change the monitors.
Just put your toes up against the wall. Craft it.
My starboard toes up.
Yeah, you got it. Once you come off the stow, I can take over.
Okay.
So we have a good reason for sampling these. There's, you know, these are really rare to be able to collect, and there's
current.
some important questions, genetic questions that we have about how these corals are related to each other and other corals in, you know, in the same group or the same family or genus. And so we'll be taking tissue samples of these for DNA analysis that could help us answer some really important questions.
I think I might grab it outside the cutters and then like twist and break so it's like squished. Yeah. You want to zoom in on both cameras, video?
I don't know about the cutters. The hose might give you better grip, though.
I don't think it'll cut.
Okay.
I think it'll have to be a snap, and if we could...
Right.
I know it's a hard ask to try to get one and not damage the other, but let's try. Wow. That is big.
I'm going to try to get it like right here. Kinda.
As close to the base,
wow.
I think, as you can. Oh.
Oh, I could get the one down there.
Yeah,
now.
could we maybe either try the feathering or the suction sampler to pick it up?
Yeah.
Yeah.
That was, look how stiff that was.
You should save
oh,
what's in
you
your
could
claw
keep that
already.
actually as a, oh.
I think I'll just go for the one that's on the ground here, that one.
Where are you going?
I think I might try to suction sample it up.
Full wide both cameras.
Or I could, it's pretty stiff, I could cradle it and bring it to the suction end effector too.
Okay.
Yeah, the suction might be the best to just preserve the skeleton as best as we can.
Okay. I'm gonna get the nozzle. So here.
Okay. I can draw out. Let me know.
Yeah, hold on 1 second.
Copy.
I'm gonna switch camera 12 to jar. You wanna get jar 1 ready?
Yeah, it's ready.
Okay.
You ready?
Alright. Bobby's flying.
Coming out. Dry lock off.
Alright. Drawer coming out slowly.
Backing off. In auto depth. Holding here.
Great.
Alright.
Alright.
Drawer's out. Okay, let me know. You
Drawer
can look at that camera as well for reference.
Yeah, you can draw it.
Okay, pushing you back.
Okay. Alright, suction. Suction, where do you want to start? 500 or higher?
Yeah, set your toe down.
Jar light on.
What was that, Petey?
Jar light.
Jar light.
Yeah, you can see where my toe was.
Yep.
Jar light.
Gonna put it right back.
Suction.
Yeah, you can go like 500 or 600.
Copy. Right there?
Yeah,
That's 500.
It's 500, copy. Alright, you want to zoom in on how many these and get it a little more lined up?
Sorry. This is right there.
There we go. You want to give me 1,000?
Copy.
Give me a little tilt down.
That's 1,000.
Sorry. You want your box back?
Alright, I gotta get that. No, you keep flying. Alright, here we go.
Do you want this view over there?
No, you're
The
good.
main one?
You're good.
Okay.
I just need to get the... There we go. Are you inhaling?
Yeah. 1,000 right now.
Great. Stop.
Can we maybe wiggle it over the bio box and try to drop it?
Yeah.
All right. Hold on. Let's come out on HD2.
Pour it in, we're going to blow it in.
Yep. I'm going to pull off.
Maybe you want to hold a small suction.
I think it's in there. I think it's going to be wedged. I'm actually concerned that it'll not come out of anything.
Okay, port inner.
Oh, I see it.
Yeah.
Okay, hold on.
We're gonna try to blow it in a bio box here without breaking the hose
All right,
out.
coming out with the drawer.
What am I doing?
Are you clear from everything? Okay, pushing out.
Hold it. Some hose. Bring it around.
Great view from the aux cam.
Yeah, for sure. How am I looking, Bobby? Did I turn a little bit? First right. Left. First right. There it is. Is that going in the right place?
Yeah. Yep. Just had to start slow and ramp up
Okay.
slowly.
Let me know.
Hold that there, Levi.
Blow it in, Mr. Fernando.
Okay, that's
Give it a pulse.
500 to start.
Giving a little up.
Boom.
Giving a
A
little
bit
up.
more.
Giving a little up.
There you
Hold
go.
it up.
All right.
Very
Drawer
nice.
stowing. Oh, sorry.
Keep the drawer out so I can stow this.
What did I just see? Port.
Port inboard.
Inboard. Thank you.
Okay.
Maybe connectivity study. And
Alright, I'm secure.
Okay.
oh, actually, I know this.
Jar 1 has sediment in it.
You're floating.
That's sample ID EX2306-D11-03B.
Sure.
And the port inboard bio box.
Bobby Moore with the assist.
Desmophyllum dianthus.
That's definitely my favorite sample.
I don't know if you need to clean it out. You just need to note that it was used and maybe not use it.
He can probably blow it out. Let's see what he's got. We'll try.
Oh yeah, it's got a lot of sediment.
Well, actually, we're going to try to clean out jar 1. There was some sediment in it. We're just blowing it out, see if we can reuse it. But if you
Can we
prefer us not
leave
to-
it in there for now, and then if we need the jar, we'll do that? But
I
there
think
could
it might already
be.
It might have already. Most of it's already kind of blown out when we blew out the.
Oh, okay.
Uh,
Just there might be something interesting in there.
it's mostly cleaned out now. I think. I'm just looking at the camera.
Yeah.
Can we zoom on that orangey
Yeah, you
encrusting
come in
sponge
here.
while we're here? Oh, cool.
Interesting.
Yeah, really different
Sorry, video. Floating here.
kind of
There you go.
morphology from some of the other sponges. I
Don't
hope
zoom
we
in
get a
anymore.
piece of it on your rock, actually. You never know what we're going to find on that.
Is that a good enough look?
That's
Watson?
great, thank
All
you.
right, we'll proceed.
Yeah.
All right, I'm back with you, pilot. Delta 16. Rotator at 52. Lights are still 50% video. Gonna kill him.
Seeing the dorsal fins of a rockfish kind of hiding in between these crevices. And lots of these little tiny
tether, as far as I
hydrocorals.
can tell, it's looking good.
Copy.
Could we get a snap of that yellow foldy encrusting sponge there?
Yeah, we can snap in video. My toes up against the wall, come in. Yep.
Cool. Wow, again, a really different morphology from the encrusting sponge that we just saw. These are really hard to sample aside from grabbing the rock that it's growing on. So we'll collect this this close-up for reference, and I think we're good. Thank you.
Okay.
There's a rock under there. Where'd that come from?
Oh, and another different species of cup coral right beside it. The Caryophilia, I think.
All right. Proceeding up, up, up. straight up and down here. Thanks for helping me, Bobby. That was fun.
Yeah, that was good.
Would you prefer flying or crafting?
I don't know.
Is it easy doing, like, manipulating the craft at an angle from the TV? I don't know how.
That's true.
It makes sense to keep with, like, the pilot is flying mentality, but there's not really much flying happening. And like Fernando said, I think you have the better awareness for the craft.
Yeah.
Especially since all I was doing was holding position, you know, off the wall for the drawer, etc. There was no real fancy flying.
Wow. I think another cookie star and another vicious coral predator, the Hipposteria. Maybe taking a bit of a break. Oh, there's a big, looks like some of the Solasters. So the larger sea stars that predate on
lasers on that
other
sea
stars like Hipposteria that Chris Ma was mentioning earlier. So now we're seeing evidence of
like
the reason why they might need to be worried.
10 meters, 215?
Yeah.
Okay.
I'm going to get close to this this one here. Let's get up there.
Let's bring the lights up at
Bridge, RV nav.
40%.
Really impressed with the size of this one.
A move of 10 meters bearing 215 degrees at decimal
really
2
big
knots.
examples.
Take it up another 20, please.
I'm pretty sure from the genus Solaster,
Good copy. Thank you, Bridge.
also called sun stars.
Yeah.
Lasers clear.
Wow. So yeah, definitely not seeing the same kind of pedicularia, which is a word I just learned that we saw on some of the other sea stars that we were zooming in on. And maybe this one is a larger, sea star predator, and doesn't need them.
Yeah, I was just getting my.
Although
lights
the exact function
stay
is still a
centered.
mystery. Each of the arms kind of entwined with some of those tube-shaped sponges. And there's always so much to see on these zooms. I'm seeing little, tiny, tiny baby primnoas.
Oh, yeah.
Some of these white hydrocorals, probably in the genus Stylaster, that kind of lumpy encrusting
Video's
sponge.
clear.
Several other kinds of encrusting sponges, and maybe an encrusting ascidian or tunicate, I think. Of course, lots of those brittle stars.
Is that pink? Same as the or is that a different hydrocoral?
Just judging by the branching pattern, I would say that it's a bit different. You know, it's less planar than the white one, kind of more bushy.
We're kind of in a forest of the white ones.
We are, yeah. It's pretty fantastic. And again, these are, really hard skeletons on these little hydrocorals, but it also makes them really brittle, so they're very difficult to sample unless you get a rock that they are growing on. I love these little guys. Ooh, this looks cool. Look at that.
This little, is that an overhang?
Oh,
a big slab.
Wow.
You want to look under the overhang?
Sure, give me some extra lights.
Let's get about 60% lights.
Looks like if you can't port, you could also see under.
Okay, apparently most of the Solasters in this area, there are many different species, so it would be hard to tell just from video. Some of them might be starfish predators, but most are predators on ophiuroids, sea cucumbers, other kinds of echinoderms.
Lasers on.
So that could be what those two were looking for. We zoomed in on earlier. This is very cool.
Looks like a couple, a slab comprised of maybe a couple flows, maybe another similar piece next to it, but these are tossed around. Loose boulders.
The eliminated section here, yeah. Hold that. There's a big rock.
Wow, yeah, so this is a nice close-up of those little hydrocorals as well. And I think some cerianthid anemones maybe, some regular anemones, big chitin right there, the huge foot.
Lower left. Yeah.
And lots of these pink and white hydrocorals, probably in the genus Stylaster, although there are a couple of others.
Look at all this stuff.
And I think those look more like colonial tunicates to me, the white encrusting things with lots of little dots in them. Easily mistaken for a sponge, but I think they're tunicates. And of course, lots and lots of these brachiopods as well.
I think we'll keep going, Ron.
Upper right. Show that
big
section.
rockfish hanging out. Using this overhang as a bit of a shelter from the current, from the looks of it.
I'm sorry, light's clear.
Okay.
So you see a pile of disorganized boulders like that. You might see something like that along a shoreline, but I think we're here too deep for this to have been a shoreline in the recent past. But we are not very far from the plate boundary and the Queen Charlotte Fault, which is a very active structure. It's in this area. It's where the Pacific Plate is moving
multi-beams back at 50 range.
In a right lateral sense, and a strike slip sense sideways next to the North American Plate, which we're on, but
sure.
It's located, I can't remember what
bridge
looked like this morning, 50 miles or so, I think, from our location. So the Queen Charlotte, which accommodates about 55 millimeters per year of offset on it, which is a pretty good rate. Generates some large earthquakes. In the last 100 years, there have been more than seven magnitude 7 or larger earthquakes. So we're starting to get into the realm where when you see piles of rocks like we just did, you might think they possibly got jarred loose in one of those big earthquakes.
Oh. Here we're getting a nice look at that kind of the sponge I think that we saw earlier in the dive with all these little projections coming up and out, maybe almost branched in some places, covered in brittle stars. This is a demosponge of some kind.
Flatter spot. Yeah. Okay. Davey agrees.
Very cool. See lots of little crustaceans attached.
Yeah, you're seeing something in multi-beam. 20 meters out, 25 meters out.
Yeah. Thanks, Dave.
Watch leads nav.
Watch lead here.
Yes, you can see on HiPAC that we're approaching waypoint 2. I was told by the previous nav that at that point we'd kind of like to start heading towards waypoint 3, maybe along the wall or a shoulder or kind of a flat spot that we see. Is that still the case, A, and B? I imagine we'll just kind of wait for your call out to start making that move towards our left.
All right, yeah, that sounds, I think that'll be a good plan, and we'll sort of have a chance to look at the top of this kind of a bench as we move over. Yeah.
Are these rocks any good?
Roger that. Then we'll continue uphill until we hear from you to start going alongside.
Sure, sounds good.
Yeah, and this might be a good spot if you want to start
Rich.
making your way to the south towards waypoint 3.
Roger, watch leads. I'm going to zoom in on multi-beam, which I think you can see back there. About 20 meters out, we have a feature. So we can either kind of stay on this side of the feature or...
Sorry, I don't see where you're looking. Oh.
Talking sonar.
This is on multi-beam sonar.
The orange one.
If you have that pulled up back there.
Bringing it up. Yeah, here we go. Oh, yeah. Oh, yeah.
So there's kind of the rubbly that we see now, and then this flatter spot up ahead, about 20 meters out. Would you rather we kind of wait for that or start heading south now?
Yeah, that's a nice face. We should probably go see what that is.
Roger that. So we'll continue uphill for about 20 more meters and then head southeast. Thank you.
Maybe we can call in another move, because that one finished like about a minute ago, right?
Yeah, that's right.
Okay. Yeah, I'm still kind of far from him.
Okay, another 10.
Copy.
Bridge, ROV nav. Same move, please.
Stringy branch coral center screen.
Thank you.
Could we zoom in on this squirrel, please?
Absolutely. Just getting some more tether now.
Just looks a little bit different from what we've been seeing.
Okay. I come in.
I think it's a black coral.
Laser's clear.
Lasers are off now.
Oh yeah, for sure.
Mm.
So it's kind of hard. This one's really caught up in the current here. It makes it a little bit difficult to see the branching pattern, but you can see the dark black skeleton underneath of the orange colored tissue, the polyps and tentacles, those kind of strands that are dangling in the current are the tentacles for these corals. And probably safe to say it could be a Bathypathes
I wouldn't push farther
kind of
than this.
caught up.
Roger.
We saw a lot of the Bathypathes alaskensis 2 dives ago.
Very different.
Quite a bit of this. And I am seeing the long single, stem coming up and then branching, so I think that's probably a good guess.
Let's look at the lowest branch.
That's great. Thanks for that.
Lowest branch.
I don't think it's going to show us any additional
So these,
detail.
the polyps are kind of divided into 3, sections with 6 tentacles in total. You can see a little bit of a mouth in the middle of them. Ooh, wow.
Big
Look at that one.
Lasers on.
Go ahead.
Feels a little precarious.
See what's underneath that.
Wow, yeah. Oh, we'll have to take a look.
There he is.
Yeah.
Yeah.
A couple of really big rockfish.
The extra lights.
Yeah, bring the lights up.
Wow,
like
just
70%.
covered in those, lots more of those tube-shaped sponges, possibly Auletta. I think I'm seeing juvenile Primnoa pacifica corals, lots of hydrocorals.
All right, we're going to keep going. We're going to keep coming up.
Yeah, a few even just some Primnoas that are just like a little stick shape
ranging in on
with
sonar.
polyps attached.
What was that?
Shrimp all over it, and maybe some, well, definitely some brachiopods, and I think I'm seeing some serianthid tubes. A big sea star, I think.
All right, pilot. And co-pilot kind of in that range of starting.
Seems to be a real
alongside.
flaky aspect to
Okay,
the
so at this point, I
flow
use
face we're
heading
coming
and
up
I
to.
just move sideways. Is that the plan?
That's the
Or
plan.
should I
if
move
you're
like 45 and then move?
Cool. A really big. Could we just take
We
a
can,
quick look
do
at
a
that big
45.
sponge?
Oh yeah.
Lasers on.
Oh, just turn them off. Add it back
So
on.
maybe turn to 180 and we'll
Ooh,
move along it.
wow.
I'm gonna frame that up.
Look
yeah,
at
that's
that.
the idea.
Yeah.
Co-pilot. Let me check with watch
Just a
lead,
rockfish
see
in
if they
almost
wanna come
every
up this or
crevice and then
along.
just kind of hanging out on the vertical face of the sun.
Frame up the sponge, really.
Watch leads nav.
Yes.
So as you see, we made it to that
Lasers
feature.
clear.
The question for you is whether we kind of stay at the bottom and move alongside, or come up to the top and then move southeast. along.
Lasers clear.
Yeah, let's start to move southeast now.
At the base, okay?
Yeah, and
Roger that.
get a little sense of what's down here. Yeah, before we head up.
You got it.
It's a great zoom. Thanks for that.
Yeah, co-pilot. So we're going to stay at the bottom here, and we'll start moving towards waypoint 3.
All right.
Which
Lasers back on.
from you bearing is 160, but we can do that in steps as you're comfortable.
160.
Yeah, 150 meters, 160.
All right.
For pilot, it'll just be to his left, 130.
Okay.
Alright. You can move going that way, I think.
Yep. They want to stay at the bottom and saddle along.
Okay. All right. I think I'm stable.
You can turn your heading to cut it, Fernando.
A little bit. Okay. So turning right now.
So you can just put like, see this hard return? Put that like, you know, at your 45.
Okay, right there. That's 185. I have it there.
And then he calls
I think I'm, yeah,
50 or
I would
130
be
or whatever.
ready for the move.
Yeah. So like Levi just said, I'm thinking like I'm almost thinking like 135.
Okay.
You can go 150 if you want. That'll bring you in a little bit closer.
Either way is fine.
Whatever. Do your choice.
Really get the sense of how steep some of these are. You don't, even on the 3D perspective, you don't get that. It doesn't
Let's
have
just
the
start with,
resolution to show these individual boulders and little cliffy faces.
So we'll be kind
How
of
do you feel about 10
following
meters at
along this
135?
this steep feature, just to get a good
135.
look at it.
Yeah, it should be fine.
Maybe see
We'll see how close I get.
see
But
how
yeah.
close we
Pilot,
can
you
get.
okay with that?
Yeah.
135.
You want to stow my starboard upper for me for that?
Copy. Your upper, your lower, both?
Starboard, upper stow.
Copy.
I'm just gonna,
Well, now he's over you. He backed out, I guess.
I backed out just so I could have room for the swing arm to swing.
Roger.
I
Lots
brought
more
it super
little
close.
shrimp swimming by. Of course, those
think I
little
might just do 5
arrowworms or
meters,
Keating
Fernando.
nests kind of
Okay.
flickering
Since we don't have good
around.
visual on the wall.
Sounds good.
I'll take it back.
Okay. Stowing your upper.
Again, if you ever
I
get
have
a
visibility
good close
for
view
the
of those
bridge
arrowworms,
right
or if
there.
you want
Let's
to
go, let's go.
look them up
Let's go.
to see what they
Forward,
look like up close, they have really
There it is.
vicious little
47.
teeth on
Yes,
them.
can I please get a ship move? 5 meters bearing 135 degrees at decimal 2. Roger that.
So
And
these
the
little
speed
filter
is variable. System
feeders,
may
the
not get up
small
that high
sponges are
in a
seem to
short 5
really
meter
enjoy
move.
this vertical feature.
Yeah, 0.1 if that's all it ramps to. Thanks.
So sponges have little ciliated cells. They kind of have little tails on them inside of the tissue, and they'll move water and kind of slowly pump it through and filter out the little bits of
Just so
detritus that they
starboard, lower
use
one.
as a source of
Starboard.
food. Look at this. That is very cool. This kind of red banded rockfish just hanging out at the
Lasers on.
intersection between this,
Roger.
this big rock and this flatter area.
Are you going to zoom in? Or Tyler? Fernando.
Lots and lots of sponges and little branching bryozoans. You can see, a pelagic sea star. One of those swimming sea stars. Looks like it's settling in for a pretty soft landing.
I wonder what prompted him to jump.
I bumped the wall, maybe that did it.
So you see I think those cracks in front of us in the face are those vertical joints that
Seems like I'm pretty far away from
created those
Yeah,
slabby
we have a movement.
boulders we saw a little further down as those break loose and fall over.
Waiting for some more tether watch. They might grab this fish.
Smaller rockfish just wedged right in between
the one
those.
with the sea star on.
Yeah, you figured that out.
And that I wondered he did land right on the rockfish.
Oh,
He's gonna get a ride.
Funny. What are the
maybe
odds?
next move,
What are the odds?
He
Trying to
didn't
get a
seem to
little bit
wake up the
closer,
rockfish.
maybe
No, a
150.
rockfish does not seem to mind one
We'll
bit.
see where that puts me.
There's a couple of other things riding on this rockfish, little crustaceans of some kind.
Roger.
I think the sea star
This
is
is a
upside down.
beautiful close-up of the pattern of those scales. And the spines and the dorsal fin. What are the odds? That's pretty cool.
Show the sea star.
Okay.
Pilot. Go ahead.
Yeah, I think this
Roger.
sea star is in for a little more traveling than it probably normally experiences today.
Is he upside down? I think
yeah,
so.
I think I'm seeing some tube feet sticking up. So
he's trying to figure out how to get
Looks
upright.
like it
Yeah.
goes,
I don't think it'll
130,
take very
I would
long
say.
for this one to get
We can
flipped
confirm
some
that.
kind of way.
Video. Alright, Bobby. I can show what you want. Here, it looks like the pan and tilt mechanism is at 0.
Lasers on.
Pretty close to 0. Zoom out video. Alright, Bobby, here you go.
Maybe turn it to port.
Huh.
Or we can do just 90, yeah.
Pretty cool gastropod, kind of a different looking shell that we got a
There you
zoom
go.
of when we were looking at
250.
that rockfish, kind of a longer
215,
one
Okay.
than those that we've been
125,
seeing.
Definitely more than 125. I think 150 would be good, just to get a little bit closer.
Roger.
Very cool.
Alright, proceeding.
What do you want at 150? 5
Oh.
meters?
Are you okay with 10 meters?
Yeah.
Yeah, I think let's 10 meters.
All right.
Yeah. And hopefully
10 meters, 150.
I'll be at a good distance, and then we can
It's
do
pretty
a long
impressive
so
seeing all of the encrusting fauna. You imagine that,
Bridge, Robbie Nav.
you know, the
New ship move, please. 10
the
meters,
odds of
10.
settling
bearing
on a place like a little
150
crevice in
degrees,
this rock
a
where
decimal 2.
a juvenile sponge or a hydrocoral or other kinds of corals could get enough of a toehold to grow.
Good copy. Thank you, Bridge.
It's probably pretty sparse. So there's a sea star eating something. Wow. Love a close-up if we can.
Roger.
If it's a Hippasteria, I want to see more of those pedicelaria. Oh, I think it's maybe a cookie star, though. So the Ceramaster, which I've been told eat sponges, so it could be predating on, or grazing on maybe some sponge material.
They probably want to see the other side, huh?
Yep. Lights at 30%.
Your arms are out.
What are the odds we could pan over and take a look at what it's eating? Not pan, I guess it'd be a
pivot
move.
over. Yeah, we can do it in a minute. But I'm going to store my port up for in a second. I gotta get away before you do that.
So you describe these things as viciously attacking whatever they're eating, even if they move imperceptibly slowly. But I wonder how often do they need to eat?
So all the way up.
Oh, interesting. I wonder if Chris could provide us with some information. Yeah, how fast are they actually moving and eating away at that coral material? You know, things seem to move very slowly, grow very slowly in general. In you know, the time scales of things seem to be really extended.
It's proud.
Like the skate eggs that we saw,
all the way out.
The skate egg singular that we saw the other day could take, I think, upwards of 4 years to completely, you know, finish growing inside and hatch out of that egg casing.
Wow.
Yeah, so that
video come in?
we do see things move at and grow at a really slow pace in the deeper waters.
Pretty obscured here.
Wow.
Wow, it's very precariously balanced, but it looks pretty solid.
I could buy that it's wrapped around one of these little
right
there.
Sponges, but
thanks, video.
Yeah, you can see its little tube feet kind of suctioning on to the surface of the rock, balancing on those two larger arms.
Yeah, I think
that's cool. Thanks for that.
Okay, tilting back up.
Go bridge. Roger. Move complete.
Sounds good. Okay, so that's 45 degrees, Levi. I think we're at a good distance from the wall.
It's definitely slower going when we cut walls like this.
Yeah.
A little more tricky. You want a close up of this star watch later or not?
Yeah, that'd be great. Thank you. This looks a little bit different from the sea stars we've been seeing.
Get a real good view of it. It's different.
Understood, Ron.
Color.
They're experiencing a little bit of a lag, I think, in general. Some of the folks tuning in are shore-based scientists, but
lights are
you can get some
30%.
Good imagery and then maybe an identification. Yeah, the color pattern looks a bit different from also from the sea stars we've been seeing.
Tight focus.
Yeah, I don't remember any pinkish ones.
No, it's actually a really beautiful color. Very cool. Some of the, you know, kind of ball-shaped projections coming out of the dermal layer, and a really strong kind of margin. It was like marginal plates.
One more tight.
Okay. Whatever you need, Ron.
Got a cool observation. Oh, here we go. This one looks like Geofferaster swifty.
Okay, video.
So or a Geofferaster. Very cool. Thanks for that.
And enough exposed rock, you can almost get a sense of the texture. As virtually everything we've seen on this expedition, fine-grained looking dark rock. Did see a little bit of texture in it, though, sort of a horizontal looking texture. Perhaps flow bending, perhaps, or just reflecting the flow structure within the rock.
I have an answer to some of our questions about feeding rates for those sea stars.
Fog lights clear.
So years ago, there was an observation of a Goniasterid feeding on bubblegum coral, 3 individuals.
Our estimate from
They came back a year later, and those same 3 sea stars were still there. So, as with a lot of things in the deep sea, a very slow process.
Wow.
Maybe why we saw a little bit of new coral growth underneath of the area that had been stripped away by the sea star.
What do you think, Fernando? 125, 135. Where do you want to?
Let's do. I mean,
Interesting that we
So it
saw
would be
yellow
155 from, because you were looking at 240,
Geoffroaster
right?
Yeah, that latest
Swiftii,
strike was
maybe
was
like
sea stars
140-ish.
yesterday
The first one was 125.
compared
so
to this
somewhere
pink
between
one.
There.
Yeah.
Just an interesting
maybe like
observation on the different color morphs.
Yeah, 145,
145. All right.
Yeah, I think that should be good.
How much distance?
I think 10 meters is fine.
Roger.
So those
Pilot?
round round
Okay.
spots, looking closer, they're the basal discs of some of the
10 or
larger
15.
Primnoa.
15 is good.
Bridge. Already
There's
down.
another nice hydrocoral.
Can I please get a ship move?
Wow, look at all the
1.5
little.
meters bearing
Oh, that's
145
interesting.
degrees at
Can
decimal
you
2.
pan up a little bit, please? That little ball with little polyps sticking out, or tentacles almost. I would have thought that was a sponge, but looking
Good copy. Thanks,
closer,
Bridge.
it's oh, maybe it is just has little encrusting polyps on top of it. I'm not sure. Way more than 8 tentacles, so it looks like something else. Huh. Interesting. Thanks for that. I don't know what that is.
Maximum copy. Pretty small then.
Looks like maybe a couple of barnacles off to the left.
Lasers as we come out.
Great. Wow. Yeah, every time you zoom in, you see something different. Just covered. Wow.
Video's clear.
And yeah, some of those circular kind of, spots that you see on the rocks, you can see a little branch coming out of them. So that tells me they're the base of a large coral like a Primnoa. And yeah, just covered in these little tube-shaped sponges. They seem to really be thriving in this area.
I'm going to go for the anemone next. Watch it. You come in.
Whoa. That's an excellent close-up of this corallomorph, I think. Judging by the little white balls at the ends of these tentacles.
Frosted
a little
tips.
bit different from the anemones that we've been seeing. Look at the color gradient too, from the dark purple to almost transparent is very beautiful.
And the change in texture from kind of those lines in the purple to the lumpy texture of the tentacles where they're translucent.
Yeah, it's beautiful. Oh, and there's one of those crabs at the base. I almost didn't even notice. Just kind of tucked in there at the bottom, and then another one behind. So this is often an association we see with a couple of different kinds of anemones. There's often a crab hanging out at the base. And sometimes symbiotic shrimp that also associate with these.
Fluff at the end of the stick, yeah.
I think those are, I think we had an ID from somebody in the science chat, tubularia.
Yeah, more. Is that it, Ron?
That's good. Those bring up lights at 30%.
Yeah, tubularia maybe.
That's the best we can do.
These little solitary hydroids that seem to have a really long stalk.
Video's clear.
Okay. Thanks, Ron.
There we go. Thanks to Lara for that ID. Tubularia.
Go ahead, Bridge. Roger.
Lights are clear.
Okay. I passed.
Move complete.
I passed you a little bit. Pilot. Still rotator is 58.
That's a big.
Ooh, big one.
Huh?
Wow.
Splashing.
Oh, this is also covered with something. Can we zoom in on this?
Yep.
On whatever this is.
Incrustation.
Unless they're really small sponges. What are they? Wow.
I'm gonna get you close, Ron.
Yep. Lights at
Could
40%.
be anemones also. Let's
take
take a
another
close.
20.
I'll stop guessing. We'll take a closer look.
Or take zoom in, Will, I should say. Whatever you want. I'm holding here.
Okay, yeah. These well, yeah, look like I would say zoanthid anemones, so kind of colonial anemones. Kind of remind me of the stoloniferous corals we've been seeing, where they're connected by a thin layer of tissue to each other, versus the solitary anemones that we've been seeing the much larger versions of. Very cool. It's a kind of a new sighting for this area.
All right, there's more
Thanks.
to the right.
Ooh. This is helpful, getting a really good close-up. Of course, a lot of these you have to dissect them and look at the mesenteries, I think, to figure out different species. A lot of the identifying features are internal.
Thanks, Will.
So we'll stick with zoanthid for now.
Full wide. Lights are clear.
And you see, yeah, quite a few of these really large rockfish kind of looming in the background.
You see the ox one, Bobby?
Yeah, that's very, very close.
Survey 2, if you see ox 1. You want it?
They're recording it now. What happens if we bring up the Og's lights with the Og's light camera?
Kind of blows it out, Ron.
Okay.
Gets you a win.
Hello.
All right, he's trying to decide if we're bothering him enough for him to have to move.
Hello. All right. I'm gonna back away from the
Yeah,
water.
for most of my work, I don't usually pay much attention to the fish. I'm looking at the invertebrates that they're blocking. I guess it's kind of like trying to look at your rocks and all of this invertebrate fauna getting in the way.
Yeah.
Everyone has their thing, but I've definitely grown to appreciate them.
Can we get another strike here?
Oh, nice.
Sure.
Little partings, almost shaly partings, and that makes me
So you're at
think those are either
waypoint 3.
flow banding or perhaps that's a little
Let me check
interval
the
of,
watch leads what they want to do
of
again, but
sedimentary
at this point
rock
we start
or
heading uphill.
Great.
Perhaps tophaceous sediments that are part of the sequence.
Watch leads, nav?
Yeah, watch lead.
Yeah, we're looking for some direction here. We're at waypoint 3 now. Would you like to continue alongside or back uphill?
Yeah.
What's your preference?
Well, yeah, let's kind of wrap around these outcrops. I could keep going a little bit here.
Roger. We'll keep following then.
Give me that 15 that direction. Or, can I even go 190, right? Or am I mistaken?
Yeah, we don't need to strike it quite as much.
Right, Verona? Can we move it directly towards D2? Is that okay?
Oh, sure.
Or is that a little too close to this? Is that a fish?
Oh,
yes, it is.
If you see an octopus, let me know, Will.
So they want to keep going along.
I think we do like one.
Yeah,
I'd
just
go
a little bit.
I would just go slightly port of what you are.
180.
Yeah.
Alright, give me 15. I'll try to get my face back into something good here.
Actually, I'd go a little bit more.
Okay.
170.
Alright. Great.
You want, what, 15?
170.
I'm getting some more tether watch. Did you see something here you want to zoom in on?
Actually, yeah, if you want to zoom in on the rock face,
Tell me
that's
there's 170.
a little bit clear there to the left of the lasers. We might Just have a little look at the rock.
Bridge,
Charlie Nav.
We'll just look at this rock.
Can I get a ship move, please,
Hold
of
that.
10 meters bearing 170 at decimal 2.
Try to see what this rock looks like.
Good copy. Thank you, Bridge.
You get a sense of, again, it's again dark and fine-grained, but see a little bit of horizontal streakiness, a little bit of banding. And on the assumption that we're looking at a volcanic rock that might reflect the flow banding from when it was coming erupting down the side of some slope.
Cool. That's a big rock.
Nice big boulder. You can see it's more or less in place. You can kind of see how it the fractures and the joining in it, as they become more and more expressed, will break the rock up into what we later find as boulders like this as they start to come loose by whatever process around here, perhaps earthquakes on the Queen Charlotte Fault that have knocked that loose. Yeah, who's this?
That's squid parts.
Little squid parts.
Huh. I wonder what it could have cut the squid in half. Thanks, video.
It's good surface on that rock.
Alright, I'm going to have to stay above this. I don't want to get the tether caught on that bad boy. Alright, Fernando.
Okay, coming up.
I'm going to move it this way.
Alright.
More fish. There's two of them. There it is.
Go ahead, Bridge. Roger. Move complete.
Obviously lots of good habitat and happy hiding places for the rockfish.
I'm going to go for this coral next.
Little shrimp up in the water column.
Actually. Pretty dead. What's the strike to the next waypoint, Bobby?
That's similar to my heading, right?
Yeah, next waypoint is right in line 190 to Sirius.
Yeah, I think we can kind of keep on this heading as we head for the next waypoint. And we're going to crossing like what looks to be a little bit of a flat spot, although it's clearly pretty steep off to the left.
I think come left, see what's on the other side of that, and then we'll bring Sirius towards you. You ready for that, Fernando?
Yep.
15?
Yeah, 15 should be good.
15190?
Yeah, 190.
Bridge, are we nav? Ship move, please, of 15 meters, 15, bearing 190 degrees at decimal 2. That's a good copy. Thank you, Bridge.
Come in video?
Move in.
Wait for some tether. Yeah, hold that.
Okay, yep.
So a nice little forest of these hydrocorals, if I'm not mistaken, on this vertical face.
Theo, what's that called on the zoom in? Oh, it was a tube worm that retracted its business. You know what I mean? Did you see that?
Put those.
These little tentacles. It was over left.
Huh.
Over there somewhere.
What's that? I don't know what that bulgy thing is there. I guess
You
just
say hold? You
looks
zoom
like
out a
maybe
little
one
bit?
of those bigger
Hold that.
tube corals.
I saw like a little tube go. Yeah, see it going, one of those. There's one of those. Just one of those things, yeah. I wonder if it's catching food from all the water rushing by it.
Yes, I would suggest that the featherworm is.
Featherworm? Is that what they're called?
I believe so. Colloquial.
Yeah, this is a great view of a tube worm showing its feeding appendages out there catching this marine snow drifting by. It's a really good look.
Right. We have a new commentator. Do you want to introduce yourself, Emily?
Hi, everyone. I'm Emily Ash. I'm a marine biologist, but I am working at NOAA Research Headquarters right now doing communications. So if you follow NOAA Research on any social media, I'm the one behind the scenes.
But you have a little bit of background in biology.
Yes, yeah. I've done a lot of research in mangrove ecosystems. Coastal zone management is what I have my master's in from UMiami. I've done some shark research. This has been on my bucket list for a very long time, so it's great to be able to be aboard Okeanos Explorer and be part of the deep sea exploration. I've been following along with these live streams since I was a very little girl, so living the childhood dream right now.
That's great, and Emily is also becoming very good at removing all the biology that encrusts the rock samples that we're bringing out.
Yes.
I know already she's going to be pretty busy.
Yeah, I am now the master rock scraper. Today's samples will be my biggest challenge.
Coming up over this rock rondo.
Copy. Delta's 15, rotator 70 degrees. Coming up with you.
Co-pilot, your bearing to waypoint 2 is now more 205.
So is this one of the first expeditions you've been on?
This is my first expedition I have been on. Very exciting, hopefully.
Right now.
So
more to come.
What are your impressions? Is it kind of what you imagined, or?
Oh, it's everything I've wished for and more. There's so many cool things.
The long days.
Oh, they're my favorite.
And the difficult to sleep nights, or are you doing pretty well?
Honestly, I like it. Getting rocked back to sleep, to fall asleep, yeah. It's very nice. Let's see. Ooh, another rockfish.
Bob, you want to introduce front row? No.
And Jen Ashoff has checked in with the chat with a very detailed description of the rock units here.
Why don't you take it, pilot?
Thought give you something to do.
Let me see. I saw the local name of the group.
Ooh, it looks like we have another soul.
I usually
soulfish.
trigger EX
Center screen.
Oh, yes.
for introductions.
You can come in, video. Go for eyeballs.
That's great.
Yeah, likely the whales group. These would be Cambrian to Proterozoic, so that would be about 560 million years and older, or 500 million years and older.
Excellent.
Okay. I'll try to
Sequence of volcanic rocks and flows along with interbedded mudstones and shales that have been metamorphosed to fairly high grades. So we're getting some thinly foliated here, mostly nonetheless pretty massive rocks.
go for the shell item.
That's a nice view of the gastropod right there. Very nice.
Got the parasite on. Going for the ride along. How you doing, Brownie?
I'm doing good. Doing great.
Yeah.
I could,
Looks like you're lined up to waypoint 4.
Yep.
So we're just gonna go with this. Okay. Next video.
Has anyone checked in with a call on those flatfish? Are they sole or are they likely or are they halibut?
Hold on. Zoom out. I'll get you there. It's gotta be around here somewhere. There it is. Want extra lights?
Yes, a lot of it.
that is 50%.
Take another 20. There we go.
Take more zoom video. I'll try to hold it here. Hold that. As you can see, I'm fighting a little bit of current. Alright, go ahead. Or zoom if you don't.
Let me adjust my focus.
Okay.
Wow.
Make somebody a good desktop.
Go for the top part. Go in. Hold that.
There's my focus.
There you go.
Sorry, Ron.
Clear the screen.
Sorry. Alright, let it go.
Full wide.
Thanks. Alright, he's lined up. 204. Just got to turn port to port.
Lights clear.
We could probably get 10 in, right?
Yep, I'm ready when you are.
Oh, this is our back on. That's a big one.
Roger, 10 coming in. Copy.
He's like tall, right?
Some more rockfish. Had a little eel pout swim by there.
Bridge, ROV nav. Ship move, please. 10 meters, bearing 205. That's right, thank you.
And another one.
Let's go ahead and keep the lasers on.
Okay.
See a little bit of the foliation here and the rocks to the right.
Kind of got to wait for some tether rolling. You know, just take this fish while I wait for tether?
Sure. You should have tether.
I'm way out there, I think.
It's only 45 degrees.
Yeah, but look how small B2 is on your camera. Feels like I'm way out. Alright, Bobby? Yeah, see it? Still on there? I think I'm way out. Oh, sorry.
We're in a little area where the marine snow is falling upwards. I don't know if that's we've caused that or if that's just a local current as the currents are running around these boulders and steep faces.
Full screen.
Okay.
Kind of slowing down there. Maybe it was us.
Fill the screen.
Roger, fill the screen.
Clear.
Okay. That kind of looks like brains, doesn't it? The encrusting sponge. Gonna snap it? Yeah, I'm seeing some. Okay, let it go.
It's a nice sponge.
Echo Bridge.
Encrusting sponge.
Alright, proceeding up this ridge here.
Move complete.
Copy, move complete.
Yeah. I'm getting closer. And contact, come in. He's got a big foot, doesn't he? Foot to shell ratio.
That's great. A nice gastropod. You can see its mantle really nicely, its body under its shell. I'm not a gastropod expert, so if anybody knows what this is in the chat.
Yeah, I was going to say, did that all come out of that shell, or
Yeah.
is he on something? Wow.
Does that all retract in there, or does it stay out?
It can retract in there. They have this this thing called an operculum that's kind of like a trap door. So, but this is a great little view of this guy.
Thanks, Theo.
But
Roger.
yeah. Fun fact about gastropods and these sea snails, and snails in general, is their feeding structure is a radula, which is basically a tiny tongue-like feature that scrapes away material that they're feeding on, and it's covered in these tiny, tiny little teeth. And it's quite disturbing if you look at it under a microscope. You wouldn't expect such an intense tooth-covered tongue from your average snail.
Would not want to have a pet snail with teeth on it.
Are we still moving, Nav? We're not, right? They call
Correct.
down.
They're stopped.
Copy.
I'm at 238.
So we're currently at a depth of about 340 meters. So we've come up, maybe a little bit more than 100 meters since the start of the dive up the side of this cliff, when we're actually getting near the top. Climbing up one of the, at this point, we're about to ascend, if we're not starting to, at the base of it, already climb up one of these last big ledgy cliffs that we see in the bathymetry.
Watch lead nav.
Watch lead.
Yeah, just to confirm before I hand off to the lunch watch, we're at 0. Our last sample was 03 bio, and we're continuing uphill at this point to waypoint 5, correct?
That sounds good to me, yeah.
All right, thank you.
I'm going to go for the dark red guy, Ron, right below. See it? Where is it?
Oh, I thought you were
Looks
going
like we
for
have a
the
tiny little
water
squid that
column.
just went past at the top of the
enemy, I
left.
think.
I'm going for this dark red thing. Oh, it's a little baby, what's it going to call it? Alright. Nudibranch, what are they called?
Nudibranch?
Yeah,
A
it is
nudibranch or
It's a little one.
a little sea cucumber. Or a little
Yeah.
nudibranch. Very cute. It's
I have
very
a,
small compared to the other ones we've been seeing. It's
It's a little one, for sure.
yeah, I don't know how to tell those apart.
Did you guys want to zoom in on
Yep.
anything else?
Christopher Moss says that is a nudibranch. It's one of the predatory ones. Very cool.
Tritonia.
Oh, you can see the not tubers, what you call it, feather worms? Flying away. We're going to do a pilot change up here. I'll get back in a minute.
Sounds good. So we're having a little pilot change here, so we're just going to take in the view for a minute or two.
There's so much life all in one spot. So much diversity. Everything from your echinoderms with your sea stars to cnidarians with your coral and anemones, all the way down to mollusks with your sea snails. So much all in one place.
Juvenile Primnoa is being called out in the chat.
Looks like there's one of those larger predatory nudibranchs almost at center screen now on that rock base.
All right, this is Merlin back in the biology chair. What did I miss while I was out at lunch? Oh, that's you. There we go.
We saw a smaller version of the predatory nudibranchs that we've been seeing, the tritoni.
Oh, those big pink ones.
But this guy was a little guy.
Okay.
We did briefly pass over a white nudibranch I don't think we've seen yet.
Very cool.
But
right,
just
Chris,
more of these
got a couple of
rockfishes,
requests from you.
some eel pout.
Go for it.
Let's put
I
by.
love those little guys.
Let's
Yeah.
put shilling cam up there.
I see some nice juvenile primnoas
Okay.
on this rock.
Yonder, too short.
Well, now you're in
Sure.
the geology chair.
Yep.
Well, need some help
kinda like
besides
to keep
just
wire
rock.
cam up
Well,
You might be
and instead of serious
might be a little
cam,
slow in the rock commentary while Jamie's
just
away.
a little bit.
That's all right.
let's get wire cam up and
We've been
monitor,
seeing lots of the same things, unless
whatever it is, 11, 13.
unless he's requested a sample in this area. I think we'll just stick with rock.
Oh, over here?
Yeah.
Yep.
And let's get CPHD up instead of app cam.
Some more of that
Is it
kind of
useless?
almost
No.
branching
The
white
Nova,
Dema
the one
sponge
with
that
the
we've
LED
we've
ring.
had a couple of nice close-ups
I've got
of.
CPHD to the right of it. You want it to the left?
The terrain in this area is incredible.
Yeah, I want the wide view.
Just everything covered in a
the wide
thick
view
layer
app cam.
of
Oh,
this
the aft cam. Gotcha. Not the-
detritus that
Yep. Understood.
settled
I don't know
marine
if I was
snow.
clear what I asked
The
for.
digital aft. Yeah.
Yeah,
I
what would
gotcha.
be causing these these mucus webs we're seeing on the rock faces?
Ooh, good question
think
that I
move that
don't
one
know the
left
answer
and put
to.
the
I
digital
guess there's
down
quite a few
in the middle.
mucusy animals that we've encountered on
is
the
true.
survey. We saw a hagfish a couple of dives ago.
There. Now they're all up there.
Know that some of the sea stars, there's one that's
bud.
conspicuously referred to as the slime star.
That's true.
That seems
All
to
right.
like to slime things.
So
but
I've
it
got
could
you at
also be
4.5.
from
I love the new router setup.
farther away.
Do you want me to
or
keep
higher
you at
up in the
4.5
water
so you
column,
can see
and
where
it's
you're
just kinda
centered, or
settled,
do you
so
want me
some
to
of,
keep
like,
you
the
centered and I'll call out
larvaceans
the angle?
create those really
on the cam?
beautiful
We can
mucus
What's
nets
your
that are
preference?
filled with bits of
I'll do whatever you
detritus,
want.
and they
yeah, just follow me.
sink.
And if I get more than 30,
They're abandoned
5 or
and
40,
remade
then
a couple of times a day.
tell me to quit.
So those will
popping off.
also
If I
sink and
down
kind of get
below
tangled up in
75
some of
or greater than
these
75, tell me
little
we need to scoot.
tubes and branches
I'll
that
follow
are
you
coming
then
off
with
of
the
the
cam.
rocks.
How about that?
I think some of those long tubes of the tubularians we were seeing earlier, kind of big solitary
Coming towards the
hydroids that make really long stalks.
center screen. Below laser left.
Stand by. Let me get in there. Come wide.
See it?
Yep.
And a nice close-up. I always appreciate that, so we can just get a little bit of a feel for how much life is covering these rocks. Every time we zoom in, I see something new.
You gonna want some fogs on that?
Yeah, lights at
ROV
about
watch lead.
40.
Copy.
Could we pan
Fogs on.
down to that kind of red fuzzy thing to the lower right?
Sure can. Do you want to see the star first while I've got it centered?
Sure, yeah.
Okay, stand by for white star. Go ahead, Roland. I've got it squared up and toes planted, more or less. Oop, toes drifting.
Ooh, just saw a fan from one of those tube worms snap shut. This looks like a really small Ceramaster, one of the cookie stars. And I still stand by the cookie star common name. I don't know about this ravioli star. I think everybody knows my stance on that by now, but I'll just
for
repeat
awareness,
it.
you got your lower stud, your upper stud, but you still have your shilling out.
Yeah, I think maybe a juvenile Saramaster.
It's your call. I think they're
and
flattening
to the
out a
left
little
of
bit,
it,
so we
that
can, if you want.
encrusting white
Okay.
glob, I think is a colonial tunicate. Kind of looks like a encrusting sponge, but I've been given those samples to look at under the dissecting scope, and they don't have any of the spicules that we would see. Oh, cool little eggs down there. Could we zoom in on whatever those are at the bottom? Maybe some little...
Oh, very cool.
Yeah, little eggs, perhaps.
Is that what we're looking at to the right? Or did I miss what you were asking for?
Oh, very cool. Yeah, I don't fully know what those are.
Center screen right now?
Yeah.
What's this in the middle of them?
I think that's a scale worm. On the top, a polynoid scale worm probably, but then on top of this encrusting sponge there are these little globs, and I'm not totally sure what they are now.
Want to put the uppers out?
Yeah,
it's always good to capture a nice full zoom of anything that we don't know, so that we can review the imagery later and try to give it an ID. That's great, thanks.
Okay, did you want to look at the pinkish red thing?
I do still want to look at the pinkish red thing.
Okay, when you come out, Roland, I'll just
thinking
back
it
off
might
and
be
shift down.
maybe a nudibranch is my guess from way back here, but shouldn't guess. We have the ability to go and look.
We're going to reset. Resetting. Uppers are out. Good. Okay. All right.
All deployed again.
Fogs are still up. We'll leave them that way, right? Okay.
That dog's secure.
Lots of these white hydrocorals.
Go ahead, Roland.
They're really abundant in this area. Ooh. Very cool. Maybe, yeah, kind of seeing some of the features, those branched appendages coming off the back. That reminds me of some of the nudibranchs that we've sampled earlier in this survey. Definitely different from the Tritonia that we've been seeing.
This,
so maybe a couple different kinds of nudibranchs here.
A bunch of the
Could we get the lasers when we pan back, just to get a quick size reference?
Okay, lasers are, I'm going to paint them.
Oh, also, can we zoom in on the other red stuff just to the lower right of this nudibranch?
Is that what you were talking about?
Yep. No, but now that I see it, I want to know what it is. Okay, I think some of those little solitary hydroids as well. Wow, look at the zoom.
Oops, stand by, lost
Very
my
cool. No, that's fine. Oh, another nudibranch down at the lower right. Yep. Wish there was something that could let people at home know what I'm pointing at. It would really help a lot. And lots of those kind of spiral pyramid-shaped shelled snails, the Calliostoma.
Okay.
Very cool. Yeah, I think what we're looking at are a different kind of solitary hydroid. They don't have the really long, thin stalk. Ooh, wow. Oh, yeah, look like little flowers, and they have those little tentacles coming out from around the sides. And I think that also looks to me like a carnivorous sponge, that white thing, kind of tall, thin white thing in the middle. Looks like an asbestopluma, maybe. Little baby anemones. There's so much to look at when you zoom in. Maybe cereanthids. I see one of them has a tube over here.
Slow pulling out.
And of course, lots and lots of encrusting sponges. Some of these look like calcareous sponges, which are a group that I don't know as much about, but they're based with a skeleton of calcium carbonate, kind of like the corals, instead of silica like the rest of the sponges.
210, 205?
205.
Copy.
Very cool. Thanks for that zoom.
I may creep a little bit over this.
It's in the starboard rail cam. Did something gooey get stuck on the lens?
Videos.
I think we're
It
good
does
to
look like, yeah, some kind of seaweed or
continue along this feature.
something
Oh,
like
another
that.
cool
slime.
egg. Maybe like a little egg trail attached to that little tube.
Full wide.
Full wide, copy. Shall we move along?
Lights are clear.
Lights are clear.
Copy.
Kill fog.
Fog's off.
Yeah, would pilots like another move?
I think so.
205?
Yeah. Looks like we're good spot.
How long? You want a 10-meter move? 15,
20?
ROV watch
Maybe
lead.
let's do
Go ahead,
15.
watch lead.
Could I request a sample here?
Guys,
How
hold
are
off
we
on that.
situated?
We're just about to call in a
Just
ship
about to
move,
move?
so we will hold that.
Okay, yeah.
What do you see?
I've had a request for some Cereanthids, and we just saw some in that Zoom.
Okay.
We're hearing that.
I hear that name a lot, but you're going to have to help me.
Oh
What
yeah,
exactly
we'll
are we
zoom
going
back in
for?
and I'll point it out.
Okay. Is this going to be a slurp, most likely?
Yes, a suction for sure.
Then you get anchor.
We'll just kind of try to scrape it off. Because
There's
it's on a hard
anemometers
rock,
down
there's not
left
as
from
much
those
room for
red
it to
guys.
run.
Maybe a bunch
Sometimes
of
when I've tried sampling Cereanthids in soft sediment, they just go, they disappear into the
John, I'm pretty sure we had
into
an
the mud,
imported
and you never
sample
see
in
them
jar
again.
1.
That's
So I
affirmative.
think
Yeah,
we'll
go ahead and
just
start it.
bypass that, yeah.
Jar 2, yep.
Yep.
We'll circle back there if we need to at some point.
Yep.
Got it spun up. HPU.
Hydraulics on.
Ooh, your first sample collection.
You want cameras?
No. Yeah, sure.
Oop.
Why not? You can go ahead
Oh,
and
there's
roll
a different
Okay.
fish than the ones we've been
Interesting.
seeing. That is interesting. Almost looks a little more cod-like.
Draw out easy. Draw out. Draw out fast is fine.
It's full.
4-6 is good.
So we have a bit of speculation about what the species might be here, but they're a very understudied group, and I think little is known about the Cereanthids and the hydroids that we might be seeing. So
or you want me to follow you out to the tip?
getting a
Just gonna
sample
get up out
to
of the way, so I
study
can
it in detail in the lab be really useful to have from this area.
Okay, Roland, I know you were watching what we
Yep.
floated away from. Get me back there if you don't mind.
Think just up left from that star you see center screen, straight up from lasers.
Yeah.
And then diagonally up to your right, past the pink. And now immediately right of lasers.
Okay, let me settle in, and Copilot can you make sure I don't run the slurp into something. Go ahead and turn it off, please.
So there's a few things I'm interested in
Turn off
on this
all.
rock face.
What do you want me to turn off?
Oh. No, not, I'm looking at hydraulic motor, sorry. Make sure that suction pump's not running.
Probably
Correct.
get a couple of
just don't
bonus
have suction.
brachiopods
Looking
and
at the
brittle
wrong thing out
stars
of
if
the
I had to
corner of my eye.
guess.
That works.
They are
Try
everywhere.
to sneak in
It'd
there.
be really nice to confirm the species.
To the
They
right of
look a
that
little bit
Buda
different
brake.
from the larger
Are we
dark
slurping?
brown ones that we
Yeah.
saw.
So it's where those red
in
things were,
previous
the
dives.
last things we
The red
looked
thing
at?
is what we're going to go for?
No, it's a different red thing in that zone.
Oh, all right.
So we'll just settle
I'm gonna
down
just
and then get a
toe in
nice
and we'll
tight
zoom in.
zoom.
It may be the ones
Try to
actually
pick out
at the
the
bottom edge of the screen, but the
organisms
ones
we're
I were
looking
describing
for.
was next to the...
Okay, you want to go partial?
Yep.
You can walk into it. Okay, science, you want to give a little guidance?
Okay, I'm scanning around trying to find what we were looking at before. Maybe up by the nudibranch, I think is where I saw it. Huh, maybe not.
So
Where did
go
they
down
go?
to the reds lower right.
Maybe scan to the right and to the left, looking for Cereanthid anemones. So they are like a little anemone with a big tube. There we go,
Right
yeah.
there.
Oh, no, that might be a, well, might be, or is that a worm? Oh yeah, there we go. You got it. So
Just that one
actually,
little teeny,
I'm interested
teeny,
in
teeny
all
one?
of this stuff, that little sponge and the hydroid that's attached, but the main target is that little tube with the tentacles coming out of it.
Okay, I'm going to go in. You want to go ahead at like 300 to 500 RPM?
Yeah, I can get it going now.
And someone needs to get the bucket cam, slurp jar cam up. Can you
Wow,
do
and
that,
it is
Nav?
small. Good catch.
We'll pulse it as needed so we don't damage him. I'll
Okay.
go in there, and you can stay partial rolling if you don't mind.
Yep, you want
Might get a
any
bonus
like
Keating
on
nath
HDT?
while we're at it. They're just everywhere in the water column
where
here.
You were not quite that tight, please. And pilot, I'm starting to get close, so go ahead and crank it.
Okay, suction.
Take them all. Yeah, there's a lot
tighter,
of interest in
Colin,
this space. So you might have to kind of scrape it a little bit. It's
Suction at 700. Get you more.
just like you're vacuuming out your car.
That's 1,000.
Go in there. That pink thing, is that what I'm...
I think, well, I don't see it anymore. I think you probably got it. But if we could just do a little scraping around here, there's lots that we're interested in. Love to get a
See it in the
brachiopod.
jar.
Thank you. And that sponge just to the left, and some of these little hydroids would be a great collection.
Got some things in the jar
Are you
there.
okay? Should we back off on the suction, you think? Or is it okay, guys?
I think it's okay.
I'm not looking at the jar, I'm just...
They don't seem to be getting damaged.
Okay, good. All right, I'm trying to get that pink and the little miniature bivalve looking. There it goes. All right, what else? That little sponge to...
Yes, please.
Next to the string. Okay, all right. Come on.
Oh, there's a scale worm in there too. In the jar.
There we go. I think we just got.
Yes. Oh, and what about that other sponge? Yeah. You think, is it within reach?
It's in the jar.
While we're here.
Yeah. Beautiful. Probably have 5 different species of sponges just in that little slurp.
Anything else?
Oh, see below the brittle star that little polyp,
red,
reddish. Yeah.
Okay.
See if you can get that.
You okay, guys?
Yep. Samples look good in the jar.
Samples look good. Thank you, Nav. Let me see if I can
They're
tickle,
just swirling around.
tickle that little pink thing.
Oh, we might get a tubularia, a bonus.
He just went to hide. Let's see if he...
Yeah.
You can't... You can roam, but you cannot hide.
Whoop. Okay.
I got him. I think I got him. Did I get
And
him?
then what about one of
that,
these
Brittlestar?
white hydrocorals?
We're going to get the arm of that Brittlestar because we just hurt him, and we're just going to... That way we hurt him on purpose instead of by accident. How about that? Oh. No, he doesn't want to let me take him.
Yeah, they're pretty, they're covered in spines. They get really stuck.
All right.
See that white hydrocoral, the branching coral? They're pretty brittle, but if we could snap off a bit, that would really help while we're here.
Okay, Roland, you with me?
Yep.
Maybe a little tighter? Okay, hold.
This has been on our collection
There's a
list for a
piece.
while. That's great.
That's enough?
Yeah.
Okay.
Oh, a little,
crab.
yeah, a little decorator crab underneath. I think we'll leave that one.
All right.
Still watching for the last items to come through.
That's great. Very happy with that. How many different phyla are in that one sample jar?
Okay, last sample
We'll have
just
to
went
count
in.
them and let you know.
That'll be a fun jar to process.
Yes, yes. We're very grateful for your skills in the lab. It's been making things work go really smoothly.
I don't think there's any swimmers.
So
but
of
I
course,
can't.
every different group
No,
of
the first jar
organisms
is
has
just
to
dirt.
be
Oh,
carefully
okay.
separated
I wouldn't worry
out,
about
assigned a
Jar
tentative
6 you
ID,
wanna
photographed, and
Thank you,
and
Will.
preserved.
What are these clear things we've been seeing swimming in the water column?
Oh,
a nice squid.
Yeah. Are they kind of like ball shaped?
Cone, like stick-like. They've been kind of everywhere.
Stick-like.
I'll
The little,
post, like the very clear ones everywhere.
yeah, arrowworms probably, or Keating naths.
Interesting.
Yeah, yeah. I've seen them lots in a series of mesopelagic or midwater surveys that I've done off of the East Coast, but they're everywhere. Yeah, Keating naths, also called arrowworms, and they have they're pretty much translucent, very small, and have 2 sets of really vicious-looking little teeth that kind of light up with a bit of iridescence. They're very cool.
Ooh, very interesting.
And they are everywhere. Might be attracted to the lights
The
here.
lights, yeah. I cannot say I am a worm fan.
Say box 2. Ox 2.
Oh, they're so small.
Sounds good.
That's okay. We have other worm fans here. There's something for everybody.
Watch lead now.
Watch lead here.
Can you confirm that sample was 04 bio?
Okay, sure.
Yes, 04B.
Good copy. And do we want to avoid going back over that suction jar in the future? Or are you worried
Yeah.
about anything coming out?
Yeah,
Okay.
that'd be nice to have
We'll
it
go ahead and mark it as a
discreet.
potential swimmer. Thank you.
Thank you.
Yep.
And that's suction jar. Which, sorry, which jar? 2?
That was canister 2, yes.
Oh, sorry, maybe I missed one while I was out at lunch.
Number 1, they attempted to suck a sample into, but it ended up just being dirt, and they ended up blowing it into the bio box. But we're going to, for now, mark that as contaminated, and we can circle back to it if we need to.
It would look like
Deal.
it was seated pretty
Sounds good. Okay, yeah, I'm all caught up.
Yeah. Oh, inside?
Yeah.
Little swimmer.
That was great. I think that is the biggest number of bio from a single.
I suppose
sample
since Jamie's
location.
here, should probably log that. Let's try it.
I'm good on hydraulics. You got it clear, right?
Yep, hydraulics secure.
Okay, swing arms are still looking sweet.
Nice sample pilots. Nice samples.
Good work together,
Yeah.
guys.
A lot going in there.
Camera magic.
Are you good with the jar? Do you want the still back up?
Maybe still cam. Still cam would be good. I'm gonna do a scan on it. Yep. Multi-beam while I'm a little high here. Let's see what's out there. Are we good? Watch lead on getting a move in.
Yeah, I'm good to continue.
All right.
15 meters at 205. Still good, pilots?
I mean, I could come over a little bit to square up, but I think if Todd's happy where he's at, that works for me.
205. All right.
Ooh, and that's a great squid right there.
20 meter moves.
Can we get
Oh,
that
wow.
down very well?
Maybe early on.
I love seeing it when they're.
Still camera.
Yes, I just did. Yeah.
Bridge RV now.
Coming on.
Partial zoom.
Oh yeah, let's follow this
Yes,
squid
we'd
for
like
a
to put in
second.
ship move if now's a
See
good
what
time.
lights roll or no?
Yes.
Plugs up.
Range. 15
Beautiful.
meters, bearing
I love it when they
205
tuck up their
degrees at
Yeah.
decimal
their
two
tentacles
knots,
like
please.
that, their arms,
So you getting away from me?
Tilt
using
up.
that fin
Stand
to
by.
kind of fight the current.
Good copy. Thank you, Bridge.
So a lot of these are in the genus Gonatus, but that's just for
Well, if I can
context. I
zoom to
wouldn't
get that
hazard a
mouth,
guess at
he's got
its
it right at us.
identification. I don't often catch squid
Try to go in.
in the
Was that the guy
work
that
that I
was
do
in the,
looking at the
by the
seafloor.
hose?
This is very cool.
We've been seeing a good amount of them today.
We have, yeah.
You swim a
Oh,
little
I
faster
love
than me,
it.
Debbie, so
It's really making
close.
some progress.
Yeah. Yeah, a little bit easier of a current today than in our dive
Okay.
site
My left
yesterday.
toe. Are we clear on
Wow.
left toe?
Oh,
That was
About to
great.
maybe bumping just
They're
a
full
Beautiful.
wide.
Coming up. I'm still chasing him.
good.
There he is. Have we gotten any stills on him?
Yeah, they were snapping.
Good. I think I might have to let him go.
Wow, look at all this. Huh.
That's the direction I'm going.
Oh, there's a nice big Primnoa
So I'll let him float.
for the moment,
Coming back to say hi.
unassaulted by sea stars,
No.
but a lot of its
More
friends are.
air than
Whoa!
that. Oh,
Whoa! Okay,
Here we are.
there we go. This is kind of
Look what he showed
a
you.
little bit of the abundance that I was hoping
Where'd you
for,
do the
that the
Yeah,
species
beautiful
distribution
pirouette
model
item.
Be clear
was
to
kind of
fall.
pointing to. I'd love to get some good shots of this
another
little localized coral thicket. Oh, yeah, another little tiny one, maybe one of the glass shrimp almost.
Oh, we on tether.
Wow,
Hard to see.
lots of material for these hippest area. I'm not worried about the coral so much in this area. One of those really big nudibranchs on a huge Primnoa. Could we get the lasers on this?
Lasers are on.
There we go. Wow.
These are some great rocks, too. They seem a
my
little bit more
face here.
exposed than
Oh,
the ones
right.
we've been seeing.
There are rocks here, yes.
Yeah.
Look, the currents come
Yeah, I
completely
think the
around.
having the exposed
absolutely.
rock seems to help for
It's dynamic down
settlement of these larger
Do you have
Yeah.
the ability
corals.
to pirouette around
I'm
it?
thinking I will, but I do have a lot of
This,
current
that
in my
nudibranch?
face.
Yeah,
As
there's
soon as
two.
I came up and over this.
Two really big ones. There's
So do
one
these
down
nudibranch
there.
that we see on these coral, are they
just try to
feeding
walk
on them?
around.
Or are they
I-
just getting up
So I have a
above
fun challenge
the
for you.
rocks?
What
I
is the
believe
strong
they
current like
also
that?
feed
As soon as my
on
side
corals,
goes
but
broad, it's just gonna be
someone in the
interesting.
science chat,
Lights
please
up
correct me if I'm
40%.
wrong, but I've
Copy.
definitely
Plug
seen them
I have full confidence in you two.
on corals that have been look damaged
great.
and with no sea stars in sight, but
Great stills too.
it's possible. I'm not seeing a lot of damage underneath of that one in particular.
Look at this
Maybe
rock.
they are
Look in
just
the,
taking advantage of the
manip
extra
cam.
height.
Look how
Oh, this is
fine
beautiful.
feature
Yeah, this is really great.
Wow.
Very linear there on the ridge.
All right, I'm working hard on last to go sideways and keep it smooth.
So you can see why we're interested in these corals, especially the larger kind of arborescent or tree-shaped corals. They provide a really intricate habitat, a little bit of an extension into the water column for some of these other animals. Okay, this one has at least 3 really big sea stars on it.
As far as I can tell, you look good. I can't really see you.
Can you
rear
back
it off
off as you go around?
So there have been studies done on coral bycatch and other samples looking at the number of associated species. If you start picking them and counting the kinds of organisms that are living in there, they can be hundreds on an individual. So often you'll see lots of different kinds of crustaceans, shrimp, and amphipods. We're seeing those mollusks again. Look at all the...
It's interesting to see such healthy ones right next to some other coral that aren't looking so great with a lot of exposed
A couple of them are having
stalks.
a real hard time, but still lots of living tissue.
Yeah.
Oh, wow.
Very good.
Okay, I'm going to let that float off screen, and we'll just come back around again.
Good reference, though.
We were doing a little bit of camera magic science just to get that shot.
Sure, sounds great.
There's a bit of current coming to my face now that we've come up on this flat area.
Yeah, I can see that.
I'm going to come back to the coral now.
So the current is picking up. We're just going to reposition to try to get a good look at this. It's really abundant coral area.
The underside of the star would be best.
Definitely. Those looks like a hat. I'm thinking those are maybe demo sponges. They're pretty floppy for any of the cone-shaped glass sponges that I know.
You have the DVL on as well. I can secure that if you'd like.
It looks like it's working now. Unless we see interference, we can maybe secure it.
All right, you're sliding back.
Okay, I'm going to push forward again.
Yeah, so that's me looking at you, 7.5.
7.5, copy.
Good picture and multi-beam.
Good picture, multi-beam. Yeah.
Come down a little bit.
Look at the current. Look at what's changed.
I checked. I don't
Thanks for that.
Thanks
for joining us.
So let's see. Your slack tide was like 10 and 4 today, so we got a little.
Okay, so we're seeing some dead skeletons, pretty big ones of Primnoa, and then just a huge living colony on the top of this really steep rock feature.
Coming up to it.
And of course, lots of Hippasteria happily feeding.
Current might increase is what I'm getting at, right?
It's we're in the middle of high tide coming in right now.
High tide flood, yeah.
So we're fighting some pretty serious current conditions as we try to get a little bit closer to these beautiful Primnoa colonies.
Wow.
Look at that.
And the best stars I saw were the ones in the upper right, but I don't know which direction you need to face to be safe.
Again, some funny comments in the science chat depending on if you're on Team Coral or Team Sea Star.
Just go down on the left side of them then.
But again, these are really slow, slow processes. So Chris Ma was saying earlier that they followed the same individual sea stars a year later. They were the same 3 on the same coral. So it takes them a long time to kind of move their way up and completely denude a colony of the tissue.
And I can give you that magnitude. Stand by.
And the snap-on nudibranch
and these Tritonia nudibranchs, you've seen quite a few really big ones.
Go ahead.
At 250 meters depth tide, it's not showing much.
No? Okay.
Just decimal 2 to the north. Surface is
beautiful.
About decimal 5 to the north.
So yeah, we're seeing a couple of the tips. There's a section there where there's some tissue missing. And these
can you just leave it partial and let me walk?
Caliostoma snails.
This you want more wider than this?
I don't know. I'm okay with this if you can.
Yeah,
I can see each individual polyp of the Primnoa hanging in the water column.
You can tell that from the color?
Yeah.
sort of taking advantage of a little bit of
Fogs are still 45.
space that's unobstructed by other fauna on the top of this rock and growing out into the current to gain access to food. I don't think access to food is hard to come by in this particular area. Just is full of marine snow. Oh, cool! Look at that barnacle maybe to the lower left.
Yeah, surface
Something
current's
sticking out.
.5, Todd. Looks like it's kind of coming into the cove here, as we might expect.
If you could zoom on that barnacle, it looks like it's doing something cool.
Center lower screen.
Yes.
Stand by.
Just closed up.
Okay, rolling.
So you see there's like a, wow,
Bottom left corner.
a little bit of a empty patch where this barnacle has found a place to settle.
Pulling out.
Come on back. I'm getting too close.
And of course, lots of brittle stars.
All right, if you want
Those
to try to snap
gastropods, the Calliostoma. Cool. Shrimp everywhere. Amphipods. And this barnacle also thinking about reaching out to feed.
Yeah, it keeps reaching out and then going back in. Is he just trying to see if he's got anything that time?
I'm not sure what that barnacle's up to. It's very dynamic. Wow.
Very large
snails.
I'm gonna walk it left. How are we looking left on the rail cam? Oh wow.
You got some corals off to your left, yep. Little ridge.
Full wide.
Stick my toe against the rock and just image this gently without having to fly it.
So we see lots of the, dead skeletons kind of to the left and the lower part of the screen, and some really big
Hovering
living
right above
colonies
one
along the
toe,
ridge
yep,
of
there
this
you go.
rock.
Oh wow.
Just beautiful.
Crazy terrain. Look at that.
Still great imagery even at this.
So again, this
backing
is
up, it still looks really
commonly known as the Pacific red tree coral,
usable stuff.
or Primnoa pacifica.
Okay. Why don't
If you could just kind of scan this area to get an idea of how far it goes.
Skids.
Go ahead, roll that.
Maybe to the left. But I know we're fighting the current the whole way. Seems to have picked up quite a bit since I came back from lunch.
Yeah, they're in a
Wow.
optimum spot to catch the
Are the lasers back on?
current in and out every day.
That sea star just clinging to the coral there. It's a big guy.
A couple anemones down there.
Paint those sea stars. There's the one with the underside we've been looking for.
Yeah,
Well, nice boulder pile. When I first got back, you could see that foliation or schistosity. Wow, that's hard to say.
Whew, yeah.
Right after lunch, I guess.
Another one of those words you read a lot but don't often say out loud. I have that problem.
Right. But a little bit different from the blockier outcrops we were seeing up on the cliff face, and makes me suspect we might be seeing more of that type of rock up on as we get up onto this little more flat area we're headed up to.
Okay, so we're kind of at the top of this feature, which is cool to note. That's where we're seeing the Primnoa. We're at about 330 meters depth. This is kind of right in the zone that they like to be. Yeah, it'd be cool to get a close-up of this
Okay, rolling.
of
we'll
this sea star, although it looks like it might be taking a break.
Hopefully holding on.
Just digesting.
In a few months, he'll go back to eating, it sounds like, by
Yeah.
that timeline we got earlier.
Mm-hmm. And I do see more of the pedestal area on the underside of this sea star. And now I'm going to wonder for a long time what they might be used for. You can see this branch of a Primnoa skeleton just completely removed of all living tissue, starting to get encrusted on maybe by some sediment and some kind of a tube-dwelling invertebrate.
And I forget what these are called.
Pedicelaria.
Pedicelaria.
Yeah, I can see a bit of the tube feet, and I wonder if that's a bit of the stomach. I know some of them kind of prolapse their stomachs out in order to feed. Beautiful, and there's some living coral tissue just down to the lower left.
We're looking at the soft tissue.
Little, all kinds of little amphipods. Little bits of detritus, I would think, kind of caught up in the branches. So they often have this kind of golden color, and then they kind of get encrusted with things and turn like a darker color over time.
Slow pull out.
Lasers are on.
Beautiful. Those are some really nice, healthy-looking polyps to the left. I know they won't be that way for long, but, well, maybe another couple of years.
We're all
tilt up.
Kind of in between geological time and normal biological time. There's a much slower rate in the deeper waters of the deep sea.
There you go. Okay, laser's now clear.
Could we take a Niskin here?
Sure can.
Survey 2?
You know, I was just thinking that.
Oh, good. Yeah, especially in an area of such high Primnoa abundance. I think it's a good record to have. And I did enter that one.
Okay, so we're on 5.
Yes.
Let's fill the screen with just the star again.
Unless I missed one.
Sorry, Nate, I didn't catch that. Say again.
Yeah, sorry, RV, I'm not sure if you heard me.
Yes.
Okay.
Niskin.
Great, thanks.
Setting up for a Niskin. This will be Niskin 2.
Yes.
Nav is ready?
Yeah, just making sure Nate's good. Copy. All right, we're going to go ahead and reset now.
Pulling for a pull. Fill the screen with
We're
this.
Fire Niskin in 3, 2, 1.
Good fire.
Confirm Niskin 2 fire.
ID EX2306-D11-05W, Niskin Bottle 2.
That finishes that shot. If you're still stable, you see right of the star in the background, the little balls.
Balls? What balls?
On the coral.
How far out?
Straight right of the star.
Just to the right?
Where the bright spot is.
Let me see if I can rock it without losing my toe. Stand by.
Holding.
If I lose my toe, I gotta punch out.
Now you're centered.
Right there?
Yep.
Go ahead, and I'm not sure what you're looking at.
The pink balls.
Oh, interesting. Yeah, that another one of those Calliostoma snails and a whole tuft of these, I think they're tubularias, the kind of solitary, mostly solitary, although these look like they might be kind of connected and branched hydroids at the end of these long tubes. Very cool.
Is that max zoom?
That is not.
Just looking at the mollusk.
Yeah.
Now it's max zoom.
That's great. We did have a request of mollusk feeding. And lots of little amphipods, I think, crawling all over these little tubularias.
Slight tilt down.
I think I'm seeing some, oh, maybe amphipod tubes as well. Saw some of those earlier. They're kind of mud-encrusted tubes that these amphipods make.
That's good. All right, video's clear. Very nice perch. And a very strong current.
Beautiful. Can we pan to the left while we're here? I thought I saw something cool at the base of one of these corals. Oh, wow, there's a rockfish just peeking out from in between those slabs. And up a little bit. Oh, no, over, yeah. So just to the left of that big sea star, at the base of that kind of healthy looking coral at the back on the left, seeing something pinkish.
Pink at the base of the coral past the star.
It's a bit far out there, but we can snap out there and see if we can
Yeah, just a snap, just to get an idea. Can't tell if it's a fish or a part of a nudibranch.
Go ahead, Rolly.
Aha, fish, thanks.
Oh yeah, good eye.
That's great. So maybe using the base of this coral for a little bit of a hiding place wedged in between the shelf of this rock. Thank you.
All right.
Wow.
Well, it looks like each one of those corals has its own sea star.
Yes, at least one.
At least, yep.
So could we scan, like, is it possible to go to the left and around this feature just to get an idea of the extent of the coral distribution here?
On the edge of my
okay.
tether. I can go a little bit further. Co-pilot, you want to follow me and bark at me when I get a little too tight?
You got it.
And then we'll have to come back and then push forward.
That's great.
Just gonna keep you
just as much documentation as we can get of this area. It's fantastic. A real, Primnoa thicket, even if it is under siege. Still lots of really beautiful, healthy corals.
I see a little slack. Oh, that wasn't tether.
That
was
was just debris. I can drop down a little bit, see if I can get a little bit better visibility.
Seems good with all these rocks poking around.
I do not have good
wow.
visual tether.
This is good. There is something through the rear.
Teeny tiny little squid. Floating, swimming through.
Yep. Something reflecting.
Cute.
Well, stay away from that.
I think it's coral.
Yeah.
Beautiful. Third time's a charm. I'm so glad that we, caught sight of some of these really large corals on our third dive in this area. Documented a few recruitment areas, probably with some juvenile corals, but nothing like these.
Good.
That's great. Thank
I'm
you.
gonna turn my head back. I came over to look at you.
Okay, I'm gonna spin right and walk left.
Yep.
And maybe just.
I have to come back over 15 degrees.
I'm looking downhill that way though, so.
Yep.
So we're getting an idea of a gonadid shrimp, squid. Some of those little squid that we've been seeing, definitely gonadids, but to be sure that it's gonadus, you need to be able to see some of the tentacle structure.
That's where I did the pirouette, so I don't need to go there again. Shall we dial one in? I think there was some interest in this sponge earlier as we were approaching, and then the corals blew us away and we decided to look at those, right?
Yeah, I am interested in this sponge as well. Still trying to figure out if it's on my list of species of interest, but I don't think so. Oh, possibly. Actually, do you think we could get a piece of this one?
But of course.
Thank you. Yeah. Should be, you know, spongy. Not hard and brittle like some of the other ones. So I think we should be able to tear a piece. It also looks like it maybe drifted down here.
I can clear them or keep them up if you're happy.
Maybe not.
It looks like with them off and...
Yeah,
Okay.
like we
Plugs
could turn
on.
them off. That's fine.
There you go. Plugs.
Oh, hi, Caitlin.
Hello.
All right, I'll spin them up.
Hydraulics or up.
Okay. And...
How are you planning to collect this one?
No, just grab a piece of it, but where and how much? HD2 up there?
Anything from kind of the lip, so I can get a good look at both sides, is also always helpful. And kind of the transition between the two.
Okay. Want to center, kind of square that up, Chris, if you don't mind?
Sure can.
If you ever want Augs 1 over here, pilot, let me know.
Aux 1. Yeah. Thank you.
Okay.
Let's put lasers on it real quick. And see how big it is. I mean, I could try to tear a piece off, but I'm concerned I'm going to pull the whole thing up. You want to go tighter on HD2?
Okay.
I mean, how big is it?
I think
it's
it
probably
should
too big for the bio box, right? To get the whole thing.
I mean, it's pretty flexible. You can stuff it down in there probably.
Yeah.
If you got the whole thing.
But then what will we do with
What's
it?
your preference?
I think, can we zoom in on the base just to see? I wonder if it fell down here. It looks like it's kind of toppled a bit.
HD2 zoom.
See that base right there?
Oh, I see. No, okay. Maybe just a piece then.
Okay.
If we can kind of rip out a piece like what we do. Yeah.
If you came in like that.
I think from the left or
Is he anchored to that, or is that just his foot?
Looks like, yeah, the basal attachment has grown pretty solidly on that rock, and that rock doesn't look like it's moved recently, so I think it's just kind of slowing in the current. It's a really large individual.
Just so I don't obscure my
I'm used to seeing them more upright. This one does look like it's kind of bowled over to me. There are a few different demo sponges that take on, that have this kind of shape.
Bios, you have one.
One of them is on my target list, so I think it would be helpful for a collection.
Both sides, okay.
Yeah, the one on the starboard is a rock covered in biology.
Okay.
And I'm not quite sure what's in the port inner. Don't know if they'll float. I don't think so.
Oh, the cup coral. That's okay to mix those two if you want.
I just wanted to make sure, yeah, it wouldn't float out of the box if we opened it.
Cup coral should be there pretty solidly.
Yeah, it's pretty
so you have
paper-like.
You have your choice of starboard inner or port outer, Todd.
Yeah, I think that's good, man.
Kind of scrape toward
maybe a little less.
A little less.
Yeah, and we can just snip. Yeah.
Just moving a little bit.
Stand by. I have a little bit too much forward thrust on. Stand by, stand by, stand by. I'm just having to push into the current.
Yeah.
But maybe more just straight down and less forward.
Yep.
Wow, there's really a lot of current pushing me backwards. Reset, guys. Stand by. Doors.
Yeah, that's fine.
And as soon as I put a little bit of downward force with the arm,
yeah,
pushed me. It just let me float forward. Just more down and less forward. Let's see if I hold there. Okay,
that's good.
Want to tilt up on HD2 for me?
Yep.
Hopefully I still have that same amount of range of motion. Boy, that Aux 1 cam is really nice.
I'm gonna put it over here just in case you want it.
Yeah, that might be closer to. Thank you, John.
nips are also very good views as well here.
There's too many good camera views.
I know.
Huh.
Good problem to have.
That would be great.
I don't
Just
think I
outside
have
the
much
cutters,
of it.
yeah.
Flopped out of it. Gonna tear?
Should. Of course, it's always a risk when we don't know what kind of sponge it is,
Yeah, I
how
think
it will
it's tearing there.
behave.
Nice.
There
Got
we
it.
go.
That's
Great.
it.
Got a clawful.
Okay, guys, where are we going with it?
That's your choice.
You've got
Starboard or port.
Yeah, starboard inner or port outer, both available.
Which one do you like?
Either one. Just your dealer's choice. All right, co-pilot.
Port for you.
Port. We're on port. Okay.
Yep. Coming out easy.
We're going to go port outer.
Yep.
I like that new camera view.
Yeah, it does give you a nice view. There you go.
Is it falling? It's falling,
Yep,
right?
that's falling.
Beautiful.
Tell me when you're clear.
Clear.
Beautiful.
Sure,
Thanks for that collection. It's excellent.
Nice job, Todd.
Thanks, Chris.
All right.
Yeah, that whole thing would have been a bit much to try to stash in the
Yeah.
drawer.
Agree.
Okay, arm clear.
Copy. Jogs coming off. Outback secure.
Got HT. I'll take HT2 briefly.
Yep.
Watch lead to confirm that was 06B, correct?
Yep, that's what I have.
Good copy, thank you.
All right.
We left most of it there to live another day.
Mm-hmm.
Lifting off.
Copy.
And I think we were about to push forward, weren't we?
Yeah, we can get another 15 in if you're ready to adventure more.
Yes.
15 at 205.
Yeah, I think that still looks good.
Copy that and work.
Thanks, John.
Bridge, RV nav.
Follow you out.
Okay, so it looks like we're continuing to follow this kind
We'd
of
like another ship move, please. Same
almost,
as the last. 15 meters
what
range.
do you call it, a
bearing
ridge-like
205
feature, not
degrees
quite.
At decimal 2 knots.
It's another sea
Good
urchin.
copy, thank you.
Oh, and some of these little very linear silvery fish. Unfortunately, all of our fish biologists are off for Labor Day, so we don't have a lot of great fish IDs. You know, a lot of invertebrate biologists represented in the science chat and here in the biology seat, so we'll just have to kind of capture the observations for analysis later.
Sweet, thanks.
That's the high frequency.
More Primnoa skeletons and a little bit of living
900.
tissue.
900, yeah, it's
Third
900.
line down,
Do you want
head.
the 22?
I don't.
No,
2250, no?
I think where you were at, it was
900.
zoomed in really far. Yeah.
You want me to range out a bit?
Maybe like 10 meters.
And you see those little poorly kind of rounded off ledges in the outcrop, which I would guess at this point would be the schistosity and the layering of this rock, and similar to those boulders that we saw here, a little more in place.
We're definitely cooking with gas now. The current is in full force.
I can get your starboard lower up if you want to saddle to it. That's your choice.
Oh wow, looks like we're coming up to more Primnoa.
Might just keep an eye on it, Chris.
So we see a lot of the kind of dead skeletons or partly dead, definitely being stripped of their tissue by sea stars, the Hibasteria, some of these Calliostoma, snails. Could we actually zoom in on the snails again?
Sure thing.
Yeah, just another another observation of the snails kind of eating.
Okay, fogs are off. Call it if you need it, Caitlin.
Okay, I think we're bright.
Okay, go for it.
Enough already.
I'll try to hold you there. It's a little.
Sounds good. Yeah, like
tenuous.
you let me know if I need to come out.
As you can see in this
Beautiful.
image,
Wow.
Cranking along.
Yeah, the current's just kind of ripping through here.
You want a tight zoom on the one in the middle?
Yes.
Okay, go in whenever you need to.
Alrighty.
But forever, well, I see plenty of particles. I was going to say, not quite as turbid as it's been, but not true.
Well, we can actually see D2 from Sirius today, even though we're 14 meters up. And yesterday at 14 meter delta depth, you couldn't even
Right.
see the outline.
We were barely seeing the lights, yeah.
So I think it is significantly clearer today.
The reading is down in the 1.3s. Yesterday, I think we had a 1.6.
And the turbidity?
Yeah. I think I'm seeing maybe some of those little amphipod tubes. Definitely a lot of these Calliostoma gastropods.
Feel free to drive me around.
Kind
Okay.
of
Can you tilt up?
pyramid-shaped shell in profile. Still lots of living tissue of the Primnoa.
Affirmative.
Little amphipods everywhere. Of course, these larger, probably pandalid shrimp.
You still got, you got room. I'm looking at you 6'5".
Thank you. Move complete, Bob.
Okay.
Complete copy.
I think, if anything,
Beautiful, and the kind of golden-colored skeleton of this coral, where it's been recently had the tissue removed.
Oof.
Look at all this life.
Yeah, I just kept it on for your lower start.
Really big Hibasteria on the other side. That's great. Thanks for that.
Are you wide?
Now, as we zoom out, I'm sort of looking around at the boulders. This wouldn't be a bad place to get a sample of this more apparently more foliated rock if there's something nearby, and I've got a couple rock boxes still waiting for me. Don't see anything immediately. Maybe one of these, yeah, and one of those in the foreground or coming into the middle.
Do you see one in particular, watch lead?
Yeah, either the one that's kind of straight below where the lasers would be, or perhaps the one that's over in the left corner, lower left corner. Might be a good size and something that's easily grabbable.
That might be okay.
That little thing?
Yeah, the little thing.
Right there. Wait, where?
Oh.
One.
Or even that one with the dark face is another good one too. Yeah, let's see if you can get that one. Have a look. Looks like it's not too big.
You want to zoom in on it before we deploy the arm?
Yeah, then we can have a
Stand by, Caitlin.
Apologies. I'm coming full wide.
Okay, now you can go in.
Okay. Coming in.
Is that too small?
Yeah, no, that'll be all
I
right.
think they're looking at the darker one, right? Yeah. There's a
And
lot
it's
of
got plenty of stuff on it
associates
that
on it.
that's a little bit.
Wow, there's lots of stuff on it actually. Something over there, I don't know what it is.
And is it possible to just zoom out just a little bit and see what this one to the left of him looks like? I think that looks like the same thing. Yeah. Yeah, that looks like a good one.
The one that we were looking at?
Sure.
You can stay there, Caitlin.
Okay, I'm coming back partial.
Okay. That's good.
Both rock boxes are open.
Both rock boxes, so we'll go starboard.
Copy.
Let me bring the grip force up. Grip force 6.
Thought about the 2-for-1 there.
I know. You want to zoom on that while it's in our face?
Sure, yeah.
Maybe.
I think I might need some fog songs.
Copy. Fog's coming up.
Oh, perfect.
And I can wing in board Todd whenever you're ready.
Thank you, Chris. That's perfect. I think that the more upward angle of one of the fogs is really helping. Yeah. There's a star trying to escape.
Mm-hmm.
Right. He's moving pretty quick for a sea star. Or is he a brittle star?
A brittle star, I'd say.
Oh, that's why he's moving so fast.
Yep. And I think,
Woo!
Makes us escape.
Yeah, that's a good plan. Smart little brittle star. Looks like there are a couple more still attached, so we'll see what we get.
Should we look underneath? Oh, perfect.
Oh wow, a little bit of everything. Even the bottom.
It's like a treasure trove.
Now, that is not the vehicle's thrusters doing that. That's the current.
Whoa.
Whipping along.
Wow.
Just ripping through here. Okay.
Shall we?
All right.
Yeah.
I'm going to come full wide.
Okay, wing it inboard.
Yeah.
There you go.
Dropping.
Come back out.
I missed where we put that.
Starboard rock box.
You good on fog scale?
Yes, I am.
Okay.
Arm is stowed.
Copy. Hydraulic secure.
Was that in the port or starboard side?
Starboard.
Starboard.
Lifting off.
Sample ID EX2306-D11-07G. Starboard forward rock box.
I'm looking at you 7.5 there.
Push out.
Can we pan a little bit to the right just to get a quick view of some of these large primnoas? Beautiful. See some more even further in the background. But we are getting really pushed around by the currents, picked up quite a bit in this area. Wow, that one is huge. You just get the lasers on that one to the right.
Can try.
Can try. Yeah, it's okay if it's a little
Full
too hairy.
broadside to the current right now. Let's see if I can hold that.
Wow. Oh, there's K2, I think.
Oh, there they are.
Wow.
Looks good.
Beautiful. Thank you for that. Oh, look at that.
Half a meter wide.
Wow.
Maybe a little more.
Yeah, they're pretty giant.
Okay,
Okay, thank
got
you.
my heels in there. Come up and spin quickly. Get back on track before I.
It's pretty much every primnoa you see a very happy looking hip area.
back on track.
Lots of those little silvery fish.
I'm looking at you 4-5
along the bottom here too.
Yeah, that was. I don't really want to do that again. That was kind of sketchy.
Yep.
With this current.
Wow, even more primnoas coming out
You want
from the
15?
shadows.
Yeah. Yeah, I'm pretty far out there, right?
Yeah.
What is that a fish at the base of this one?
For your reference.
Yeah, I think it's a rockfish. It's kind of the back.
Oh, yeah.
Target bearing, the waypoint
Or
5 is 218.
lying down.
Almost.
Maybe, I don't know.
Flatfish.
Oh yeah.
Bearing, would you like the ship move?
These big Pacific Dover sole, I believe.
Todd, he's at 200. I think you're pretty far out there, Todd.
Yeah, I'm way out there.
Let's do another 205.
Okay, well, I'm going to settle in, and then we got to let Sirius catch up with us.
15 meters?
Yeah, please.
Got a minute
Inward.
to zoom on this, and there's a
Bridge,
sponge
driving now?
next to his tail as well.
Yes, we'd like another ship move, please. Same as the last. 15 meters range.
Okay, Caitlin.
Bearing
All
205 degrees
Okay,
at
coming
decimal
in.
2 knots.
I'm going to come down, Todd, just try to get a little bit more light on you.
Okay.
Gonna hold this shot.
Copy, thanks, Bridge.
This one's a little bit grumpy looking to me.
We've got his mouth in this view.
Yeah.
Looks like there's a bit of damage to this one actually. Look at the top there.
Oh,
Maybe
let's
you
zoom
got
in on damage.
bitten by something. Wow, ouch.
He's a survivor. Or she.
Wow.
Looking at starboard rail cam.
Yep.
It's a nice scene.
That's next zoom.
If you can come partial, please.
Coming out.
I'm shifting. I need to lock it down, and I'll reset to get his face.
Love if we're here for zooms after this. I would take a look at that really nice, healthy-looking primnoa beside us.
Sure thing.
Thank you.
Okay, Caitlin.
Okay, going in on face.
What's our surface current up to, John?
Wow.
It's actually
You can really see the torsion in this one. The mouth kind of twisted sideways, and the whole rest of the body
Tilt down as you
with
come in.
the eyes kind of in the same place.
That's good.
Did we say slack was 1600?
Wow. Whoa.
The high
Look
tide,
at the
you said?
range
No,
of
slack
those
tide. Slack
eyeballs.
tide.
I didn't realize how much the whole thing could move.
This morning during the pre-dive, doing the drift.
10:18 was low tide. 4:28 p.m. is high tide.
Yeah, so you still got 3.5 hours.
Oh, it's moving.
Yeah, we're right in the
After
middle.
scanning around, he's decided
Yeah.
it's time to get out of here.
Just kind of done with this close-up. Wow, look at that. You can see the spacing between the teeth is kind of big. Whatever
Nice shot
took
of
a
the
chunk
damage
out of that one.
Yeah. Can I zoom in
Todd,
on
so you
damage?
guys good for the moment?
Zoom in on damage while you can.
If it settles.
Go ahead.
Ow.
Yeah.
That guy has a boo-boo.
Looks like it's healing
guy.
though.
That's unfortunate. Wow. Okay, and video is clear.
Okay, and I heard a request for this coral upper left. I'll scan left.
Go ahead, Rich. Copy, thank you.
Yeah,
You
another
got
one of
time
those
now, Todd?
interesting kind
Yeah,
of
we're settled.
small
Should we
stalked sponges as well. I think there are a couple of species that I've seen in my guide that have that morphology. A couple of different Demo
For your
sponges.
reference, you have starboard inner bio box and 3 suction jars.
Getting a little bit more sedimentation here. You see the sediment piled up on just the one half of that rock, but not enough to discourage the coral.
Mm-hmm. Yeah, I think
He's
once it.
stuck to the more vertical face that's not getting sedimented.
Mm-hmm.
I'm gonna get in real tight. Get
Yeah,
a
and
real
I'd say
close-up
once
on
they
this
get to a
still
certain size, the sediment at the base probably bothers them less. But yeah, this one up in the foreground, kind of pristine. It looks like it hasn't been predated on by any of these
clear lasers?
sea stars. So it looks perfectly intact. It's just beautiful.
Zoom once settled?
Stand by. Thought I had it, and then I did not. You got it. Go ahead.
Okay. Holding here.
Wow. Yeah, really healthy looking, kind of fully extended polyps of this Pacific red tree coral, Primnoa pacifica.
Up or down, Caitlin?
I think I can see little eyes of shrimp
playing with the still cam?
kind of peeking out.
Yeah, got a nice close up on this guy.
Yeah, Nate's getting some shots.
Yeah, this one's beautiful. Very healthy looking colony.
Fogs are off.
Maybe we'll fog it up before we finish.
Do you think that being anchored out here on this kind of solitary rock surrounded by sediment is maybe a little bit of protection from the predatory sea stars?
I'd wonder. I could imagine one of those sea stars, you know, starting to crawl over the soft sediment and getting swept away because there's not as much to hold on to.
Right.
Right.
Come back, coral.
So if they're used to kind of associating with these bigger rocks, then maybe
I wanted to do a focus pull, but
yeah, they would need that to kind of grip. So maybe it's on a little bit of an island in the midst of all of this. It could be kind of acting as a little moat.
A moat, sediment moat.
Yeah, to defend against these coral grazers. Little amphipod, I think. Crustacean attached to the end of this branch. Wow. And then all the, of course, all of the polyps and the coral behind this one. There's just so many here. Really big individuals, providing habitat and a little bit of extra complexity and structure. Some sort of slow zones, kind of creating little eddies for smaller fish and invertebrates to hide and live.
Okay, I think video's clear.
Okay. Do you want to try little fogs for a still cam shot or not?
Sure. Like let's try that.
Coming up.
Ooh.
Nate, you want to talk to us, drive us up, down, left, right, more light, less light? I can't. Feel free to talk in our ear.
Just collecting some still imagery of this really beautiful coral specimen.
I'm going to zoom in too, pilot.
Go ahead. I can't hear Nate. Is he talking to us?
Yeah,
feel free to drive us if you want us to do something to get a nice shot of this. Okay.
You happy with the light, Nate? Okay.
It's really pretty. I like the out of focus coral in the foreground and the starfish hanging on.
Yeah.
Okay,
gives you just a bit of a feel for the kind of abundance that we have in this area.
Coming wide?
I think so. Okay.
This is fantastic. This is what I was hoping to see when I saw the coral prediction model just light up in this area. So I'm very happy to see all of these Primnoa.
Off to your kind of push right a little bit.
Yeah, I mean, whatever your local high is, I think I can always come back around.
I'll just go up and over this. And Caitlin, if you see something, you want any specific directions, feel free to bark.
So what kind of things go into these models for predicting species distribution? Is it potential substrates or, as well as current?
Yeah,
whatnot?
Definitely like a combo. So the first thing that they will bring in is all of those bycatch records. So I think the one that I looked at used about 20 years of
Should we get a shot of the underside
coral
of the sea star
bycatch data from the trawl surveys. So every time a trawl touched the seafloor and came up with a bit of coral, it was documented weight, I think, or counted, identified to the best of their ability. So some of them are lumped, and then those records were ingested into this model, along with, I think I saw,
wish I could tell you.
Depth for sure
So
is a
dark.
really, big predictor in coral distribution.
I think it might just be one of these
Temperature,
steps.
and
Or the little
I think I
outcroppings.
saw something like current.
Big corals. Yeah.
I'm not sure about substrate, if they brought in any kind of
Yeah, 'cause
mapping
it's
like
so
that.
see-through-y.
Actually, I could check. I was just looking at this ocean color, so different measurements of the seawater maximum tidal current.
Holy moly.
Whoa, look at this one. Yeah, those seem like the main factors that were sort of ingested by that model to give us this prediction. And look at this huge coral. If we could get the lasers on it for a nice measurement,
Try to sweep
especially at the base. Wow. So the base alone is over 10 centimeters wide, closer to 20.
Wow.
Part of it. Just huge. And we can really clearly see which way the current is going, and it does look to me like these are kind of growing in a way that orients them to catch the current and catch all the little particles that are coming out. There's a nice acanthascus sponge at the bottom that looks pretty pristine as well.
That's a nice
But
shot,
look
Todd.
at this one. Just when I think I see the biggest, healthiest looking one, then we come and see this.
I'm going to get in close.
Just beautiful.
Fogs.
You can see all of
Yes.
the particles
Okay.
kind of going
Fogs taken
through
up.
and around these branches. And you can imagine each of those tiny little polyps, reaching out into the water column and grabbing bits of detritus and all of the organic matter that it feeds on. There are
So
lots
big.
of, some really pristine looking ones up here. Just beautiful.
Okay. Let that go. Climb this hill.
Oh, I can see a fishtail in there. There's some kind of fish that's hiding out.
Inside the coral. Yeah.
Okay. Fog secure.
Okay.
I wasn't really expecting a steep little hill right here, based on the bathymetry. I think this is a local little lump.
Yeah.
Look at this rail cams, they don't seem like they keep going.
Yeah, we are coming up to another little, although it looks pretty flat on the high-pack bathymetry, another little ledge, a little slightly steeper area, not as steep as what we saw earlier, but will be a little bit of a rise.
There's a big sea pen straight ahead, kind of at the top. And more Primnoa, I think just the bases of them kind of coming into view.
Are you interested in the sea pen?
Yeah, I'd love a close-up, please.
Okay. Can you get settled in, Caitlin?
I think we've seen a little bit of this one or a similar species. It's Helipteris or Balticina, I think is the newer name for that genus. So a few different species. This could be Helipteris californica. There's a pretty distinct one a different species that I don't think this is, but then there are one or two unnamed species from this area. But samples of those exist already in the museum collections to my knowledge, so we'll just get some nice imagery, especially close-ups of the polyps are always very helpful. You can see little spines at the bases, should be a pair of them, I think, that can help us kind of give a
sounds good.
an image-based ID. And of course it's getting whipped around in the current. That's great. I think that's yeah, I have a good look at the spines. This is enough for us to assign it a morphotype, maybe a tentative ID. Thank you.
Okay, it sounds like science is clear.
I'm going to go all the way to the bottom just in case there's something really cool, since we're already parked.
I'm going to zoom out just so we can keep the base in view.
Nice view of that brachiopod and some brittle star arms. Lots of little tubes from worms, probably. The stalks of those tubularias.
Gonna zoom in.
Little sponge.
Can we tilt up to the top one?
The little, what do you call in a brachiopod, little fingers?
Oh yeah, there's definitely some tissue on the top here. You mean these coming out?
No, more in the internally.
Well, I haven't looked at
tentacles.
I almost call them cilia.
Yeah.
But that's just me. I'd call it cilia. That's what it looks like to me. But yeah, little tiny hair-like structures. Probably helps with filtering out food that it's interested in, is my guess.
That's full wife.
A lophophore. There we go.
Lophophore. I always thought of that as more of a singular thing, but that's from way back in my college years, taking paleontology.
Oh, okay.
Right.
I was like, what biology?
I got you 4
Ah,
5
paleontology.
Where am I at? 4 5. Should we make another move?
Wow, another giant Primnoa. A couple of damaged or predated ones, but still just huge.
No problem. Bridge,
huge specimens.
Yes,
that
we'd like
big
another
one right in the
please,
middle
same as
there.
the last. Range 15 meters, bearing 205 degrees at decimal 2 knots.
Wow.
Good copy, thank you.
So we've reached the top of this, so 100, 150 meter or so high rocky face we came up, and we're now up on the summit at a depth of about 320, 325 meters, which is where we'll be, I think, for the rest of the dive.
I mean, I'm okay with this. I'm okay with being in and around here for the rest of the dive. Look at all these beautiful Primnoas.
There's still a couple ridges up here, but they're only a few meters high. And
what time does Lars have meetings? Just wondering if you wanted a chance to get in the pilot seat a little bit.
RV, can we zoom in a little bit? There might be a bit of fishing line.
Oh, where do you see that?
Just a straight line, kind of. Well, actually, could you turn the lasers on and I can direct? So it's like the line in between the corals, just above the lasers.
Going left, right?
Yeah, still a little bit above, and it's going left to right, right there. Just passed over them. I'm not sure. Could just be another kind of rocky feature, but it looked a little too straight to me. If you pan to the left. Oh, maybe not.
Okay, rolling.
No, the closer I look, the less I see it. Could just be
sandy spot.
Yeah, some debris kind of following a crack in this rock slab. Thanks for the zoom.
Sure.
Yeah, I believe he has one at 2:30.
Wow.
I'm going to try to join him for that right when we get off shift.
Look at these just giant Primnoas.
Is he listening in the workshop?
Look at that one.
Workshop, co-pilot.
Give him an opportunity.
Hey, is Lars down there? Yeah, I just didn't. Todd was wondering if he wanted to get some bottom time here while he's got some time.
I think I see more coming out of the shadows, so
those corals
another area of high abundance.
They were monstrous.
These Primnoa pacifica. There's a little ctenophore floating into view. Well, pretty big one actually. Yeah, I just love to follow this along as much as we can and document its extent.
Jump on the iris.
Sure, scientists will be interested in kind of calculating the size and the overall health of the corals in this area.
I
sounded like it.
I didn't hear it.
That's what I'd be looking at if I was annotating this video anyway.
Watch lead now.
Watch lead here.
Are you okay with us just following this ridge for now?
Yeah, sure.
Absolutely,
yes.
yeah.
Are you?
Roger
yeah.
that.
Yeah, I mean, if this ridge is where the corals are, then I'd love to follow it.
Nice and lit up in blue view there.
Yeah, that's pretty cool.
Looks like red rocks.
That guy, not very healthy one. Gave him a little tickle, but we're okay. I think the current has backed off a little bit.
Dive Super's here. Yep. I think it's generally coming from your couple points off your port bow.
Good back, good bow. Sounds good. Thank you.
Yep.
Just gonna push that forward.
So we just acquired this little ridge here. I think we just guidance is to just follow this along. Local high here.
Okay, follow the ridge.
Yep.
Okay.
And I can turn my head. We can start going that direction. We need to.
Yeah.
I'm looking almost straight down at you, but we just got done with the ship move, so.
Copy that. I'll push forward here a little bit. Delta 14. That's good. It's quite interesting, huh?
Yeah, I'm seeing some really big corals. Lots of sea stars.
Lars, you missed a skate earlier.
I saw it.
Oh,
I heard you get excited, so I tuned in a little bit more than normal.
Good, good. I'm thinking a long nose skate.
Excellent. Did we ever find out what the egg was that we saw a few dives back with the stripes?
You know, I haven't had time to look it up.
Gotcha.
If somebody sends me the still of that, then I could probably narrow it down.
Gotcha.
Wow, look at all of this.
BBL on.
Okay, sounds good.
So again, we're seeing lots of these Hippasteria sea stars, but even more abundance or number of these Primnoa corals, which is fantastic. We'll follow along this ridge and kind of try to document the extent.
Yeah, no,
We've had some really beautiful close-up imagery.
In that way, sort of
Now I just kind of like to see if we can kind of count them, get a feel for how many are here.
Be aware whenever they.
Every time I think we've come to the end, then I start to see more coming out of the shadows. This is fantastic. I'm happy to follow this ridge.
And then just keep going, keep on going.
Just getting around this light from the push forward.
There you go now. Push past skids, okay?
Yes, go ahead, Dave.
That's the kind of flight. Just fly over them.
Wow.
Yeah, I'm not sure. I can't tell from the sonar. From the perspective view, it looks like we might have a little bit higher to go to the top. There might be one more little ledge to climb if the if what I'm seeing is right.
And do you think those are corals pinging, those blobs, or are they rocks? Could they be corals?
I think they could well be.
Wow.
Keep out and come dead center. Looks like we have a little bit more over here. Look at it. Start coming back down.
So that's 4-5 there. We can get another 15 if you want to push out.
Copy that.
Looks like your blue view is showing
Right
more stuff off to the right.
Bridge, RV
Wow. So yeah, these large corals
yes,
just kind of dominating that area. They're just everywhere, and it looks like there might be more coming up ahead. Starting to get little signatures that in the sonar that normally I would have thought were rocks, but they could be these corals.
Don't let you slide back. Just cleared it.
Oh yeah, so here we are. Now we've moved. I'm not used to the scale of being this zoomed in to the 3D model, but yeah, now we are. It does show us right on top of that ridge, right on the tip top.
Oh, another flatfish. Less sure about the idea on that one.
Big coral on the top.
We'll leave it to the fish biologists.
What's over here first, and then,
whoa. I'm also seeing lots of these hydrocorals. We were able to get a sample of one earlier, which is very helpful. ROV, could we zoom in on that white just below the lasers to the left a little bit?
Sure.
Kind of a blob. I just wasn't quite sure what I was looking at.
Quick zoom here, video.
Yeah, just a snap.
You ready?
Yep.
Go for it.
Oh, okay. Yeah, a hydrocoral from a different angle. Thank you.
Okay. Pilot's clear when video's clear.
Something left of the rock there. So the fish?
Yeah, looks like a little fish. Couple urchins. What if? So do you want that coral video, or you want the bigger ones behind it?
Bigger ones.
Okay. I assume so.
Whoa. Well, there's our first Liponema, I think, the pom-pom anemone that we saw lots of
past the pom-pom
a few dives ago. We've seen a little bit.
Snap? Yeah.
Just a lighter colored one. That's cool. Oh, it's really beautiful. Kind of a pale lavender color. Liponema anemone. Oh, and some little tiny cup corals too. Very cool. Every time you zoom in, you see something different.
Just a great
and look at these really big primnoas.
Do you prefer I just keep you at 4-5, or do you want me to keep you centered?
4-5 is good for me.
There's another one, maybe a meter across.
Wow.
I'll keep you solid at 4-5.
definitely some of the larger individuals that I've seen.
So we've ascended the steepest contours, but if we believe bathymetry, looks like there's a little bit more altitude to our west.
To our starboard?
Yep.
Can we square up on the big one on the left center? See how the white is coming up? Take a look at that.
From the bottom, all the way to the left.
No.
Please pardon the interruption.
Right below
Your
the
conference contains less than 3 participants at this time and will be...
Copy, thank you.
Good to know.
Okay, go ahead and do
It's
a
like,
snap.
did I do that?
can
What
go
button did I
down if
push?
we need to.
I never know, yeah.
Okay, so seeing those little hydrocorals at the base and some of the shrimp.
This is what you were talking about?
That's it,
I think some hydroids, those little kind of really small branching tree-like structures. I see yeah, now that we're closer up, little shrimp on the coral, on the branches. Some small anemones growing on some exposed tissue.
Just look at it right at the base. Just take it all the way down.
Beautiful. So that's that part of the older coral skeleton at the base. Doesn't have any polyps on it. It's kind of like the trunk of a tree. But it's still covered in, looks like living tissue.
That's perfect.
This one's a really beautiful, kind of pristine, healthy looking
and lasers clear.
Primnoa coral.
Just fly past these on the left if you
We'll
can.
pause here to get a bit of imagery of this one. It's just spectacular. You can see the marine snow floating in the against the dark background.
A little bit more.
Kind of a wintry scene.
It is, yeah. It has been reminding me of actual snow quite a bit, especially just at the so thick in this part.
The current keeps pushing you back.
And the corals with their branches extended, almost making, practically like a wall for the water to slow down when it encounters the branches and the little polyps in between, and slows down the water and the debris that it's carrying so the coral can feed.
Right, Nav, you said higher is more towards starboard?
More to starboard if we believe bathymetry here. Okay. It's not a whole lot more elevation, but a little bit.
Okay.
Yeah, from the 3D perspective, it looks like it'll be flat. It'll be fairly flat for the next 50 meters or so, and then there's another one of these little ridges similar to when we went over, and one slightly smaller one on the way, maybe 20 or 30 meters out.
RV, could we get the lasers on some of these? Just would love to be collecting approximate height measurements of some of these corals as we pass by. So a reminder for anyone watching who's not familiar, those laser dots are exactly 10 centimeters apart.
Get a little bit closer to,
so you can
light them
use them to make estimates of the size of the things that we're seeing in the video.
That's a good distance.
A few more of those, probably Strongylocentrus genus of urchin. Sea urchins at the bottom. And of course, evidence that a Hippasteria has probably been here, judging by the condition of that coral in the middle. And also there's some really beautiful, healthy colonies.
Do we know what the range of these corals are?
I do. Where's my guide?
An object bottom center screen.
I think it's the soul. You want to go ahead and come in? Step in. Ooh, see if you can.
Oh, another one of those sea cucumbers. The one's related to the giant California sea cucumber.
Are that his droppings that are right behind him? That's an interesting bunch of pellets.
Like it. Yeah, I'd say so. So that's Apos Dicopus is the genus, which is kind of a fun one to say. And Leucothele, or Leucothele, I think is the species on that one. Of course, the giant California sea cucumber being another species in the same genus.
That one rounded boulder heading, we're just passing on the left, strikes me as maybe being a drop stone from an iceberg, since we see nothing else like it out here.
It's kind of odd having to bounce up over these corals and come back down.
Yeah, they're pretty tall.
Yeah, definitely. Alright, I'll push forward here.
So I'm still just trying to get a feel for how far south we see this species. Certainly we get lots of it in BC waters, and I know the type specimen, so the original coral that was used to describe the species in the first place was from off of Japan. So both sides of the Pacific, but as far as how north and how far south it goes, off the top of my head, I can't remember. Definitely, just a bit south of us in BC. We see them quite a bit on a couple of our seamount study sites.
Maybe another move if we're comfortable. Co-pilot now.
Sure. What bearing you guys want?
What's your local high there, Lars? We're on top, I think, is what John was saying. So
yeah,
we've run out of dive track.
Copy. I think we can keep heading west, maybe. Would be getting up to that 320.
Yeah, I think west is
this little ravine here.
roughly where we'll get to that next little ridge. Where might be a little bit of downhill before then,
30
though.
meters out, you start to kind of pick something up on multi-beam, maybe.
Go for that.
So you want to move due west?
Yeah. Let's go west. I'll kind of settle over there.
Roger that. You want 20 meters?
Yes.
In work.
Do you think we could pause for a Niskin here?
Sure.
Just while we're close to such an abundant Primnoa area. It's always
put that ship move in.
Always good.
Okay. Bridge RV now. Yes, we'd like another ship move, please. Range 20 meters, bearing 270 degrees at decimal 2 knots, please.
Copy.
I have us on Niskin bottle 3.
Copy, thank you.
Same.
A little drop off here.
Yep. Copy. John, whenever you're
Ready.
Okay. All right, watch leads. We're going to go ahead and reset and fire Niskin here. And 3, 2, 1. Fire. Third Niskin. Confirm.
Perfect. Thank you.
EX2306-D11-08W.
Yeah, no viz on you, Lars.
Okay. Could you take the blue view and bring it down a little bit? So it's just where we're going to be dipping down into this little area. Look how flat that is.
Yep.
Yeah, it's just this ridge right here, and then it just flattens right out.
If we don't see anything out there, we can come back to the ridge.
Yep.
There's going to be a little lump out there.
Copy that, and we'll find it if there is one. Alright, co-pilot, I'm just going to saddle over here, try and keep my head into the current, and then drop down below.
Okay. I can line up once you get whatever bearing you want.
Copy.
Maybe about 2 meters high. But you're going to have to go down a little bit before you get there. Down maybe 3 meters, looks like.
It's pretty wild, though.
And you got something here, about 25 meters out.
Perfect.
You want to try to head on that?
Yeah. Good, coming back down.
Coming down with you.
Okay, yeah. It looks like we got something 20 meters out. 220 maybe. We'll wait for that ship move to catch up.
I see co-pilot, you're lined up at 205. That last ship move is 270. Be
Copy.
advised.
Yeah, as soon as you're ready, Lars, and can saddle over, we'll go ahead and get you on there.
If you want to start coming over, I'll saddle.
Yep.
I might leave my head at like a 220 into the current here as we do so.
Yep. Thanks, John.
Yeah.
Look at the view behind us.
Oh, wow. Things sure change when you get out in the mud.
Yeah. So we can see behind us there's a big boulder covered in those from NOAA. A couple of different sea stars associated with the soft sediment.
We're just going to get
another flatfish, possibly a Dover sole. Pacific Dover sole. Nope, there's another one.
It's your call, Lars.
Yeah.
Stop on it.
If you can hold a 250 for now, I'll just keep going.
Yep.
Center screen.
Something looks a little different about that one.
We can slowly work our way over to that 270.
Little, maybe it's just the angle. It looked a little longer and slender.
It's going to be about 10 meters out from you.
Copy that.
Paint up for the blue view, see if I get a better shot.
Yeah, the ID guide that I have is kind of limited to halibut and 2 different kinds of sole. So it might not apply. Oh yeah, definitely. Could be just a younger version, those 2 side by side. They definitely look a little bit different.
About 8 meters out.
Yeah, we got some rocks, coral.
Oh wow. This one looks like the coral got so big it might have toppled that rock by catching the current. Yeah.
Now, would he be able to get back upright, or is his stock too stiff?
It looks like they're trying. See the tips there are growing.
Are starting to grow out.
To grow vertical,
suggest he's otherwise stuck like this.
Yeah. Yeah, I wouldn't expect the part that's already grown to be able to change, but then the new growth can go up. Of course, as soon as it gets a little bit heavy, I think it would start to sag, but actually, yeah, the ends of that coral look pretty healthy.
Okay.
We have a ship move, should be underway, I think, unless it just
It's another thorny head rockfish, I think.
I was at 2142.
A couple of them here.
Oh no, 2141. Yeah, you're parked, guys.
Okay.
Was that a 20-meter move at 270, Todd?
Yes.
Okay.
I'm looking at it. That's what this looks like right here.
Okay, we can go ahead and get another move in then.
Where to, guys?
20 meters at 215 is where I'm pointing, going up here.
Yep.
Maybe a 220.
Yeah, 220, I think. 20 meters, Todd? Did you get that?
Back to the southwest.
Yeah.
Yep.
Please.
Yeah, we just had to jump over one of those ridges.
Copy. 20 meters, 220. Bridge, nav. If you're available for a move, we'd like to move 20 meters, bearing 215 at decimal 2 knots.
Never mind, those 2 white things I
this
thought were different.
flatfish side by side.
Different angles.
Oh, yeah, it's
And different colors.
Good copy. Thank you,
Yeah.
Bridge.
So I think one, could be kind of a
And get in close. So
a juvenile
still
version
get
of the
good
other, or
shots.
they could be different species. The coloration on them do vary quite a bit. You have here, I can show you a few different pictures.
The 220 Todd.
And these are all Dover soles. Interesting.
Dover sole, yeah. So this is still my guess.
All right, yeah.
Unless somebody tells me differently. Yeah, it's kind of interesting to compare these two different, kind of morphologies. So, and another fish.
And
this
then
one
we
still don't know.
Okay, if you want to go ahead and come in. Have we got good video of the fish we don't know?
No, we have not.
Not great that I can recall.
He swung away.
Maybe catch another one. This is cool though to compare the two morphologies.
Coming up.
Kind of yellowish. The range on those eyeballs is very cool. Very cool to see.
So I kind of get the feeling I can see how they move their eyeballs around. Their eyeballs are completely surrounded by soft tissue. And so it's really almost more like pulling the skin around and the eyeballs come with it. I always wondered how the skull deformed itself to move an eyeball around.
Allow that, yeah. Well, there must be, some kind of a basal region for the, ligaments to attach to.
Right. Yeah.
Put down, feel the screen.
And what's that really bright looking
spot?
The golf ball? Yeah.
I think that's actually a colonial Ascidian. So there's some very spongy looking tunicates that kind of grow encrusting in encrusting forms like that. And that's what it reminds me of. Could be a sponge, but I'm just from the samples that I've looked at in the past, I've had those come across my microscope and I've always been surprised. Look at it. Oh, this is not a sponge at all. So yeah, unfortunately, all I have to go on is kind of different color patterns, so the slightly different overall shape to the fish, but then kind of looking at those white dots, but color patterns on flatfish are apparently very variable.
Now this one's next to a tennis ball.
Yeah. Well, I think that one is a sponge.
Okay.
For sure. Or, well, could be the back of a brachiopod, I guess.
Okay, pilot's clear when video is.
Video's clear.
And lots of this really delicately branching pink hydrocorals. Just everywhere, wow. And some big primnoas up to the right.
This feels a lot better.
Yeah, if we see that fish again, it would be nice to get another zoom on it. I'm not sure that we captured it.
I think we got it right up
Oh,
here.
great, yeah.
They want to
Oh, there's two.
Before they get
Couple, yeah.
All right, go ahead and snap video.
Ooh. So I think I also maybe avoided this because I don't know exactly what they are. They're not in any ID guide that I have, and I don't think I've heard any fish biologists tuning in today.
Oh, Michael says maybe an Alaska pollock.
That's a pollock?
Guys, you should be settling out.
Not that one.
Okay, good. I was about to say, it's like I've seen pollock in the Atlantic. They're a little bit bigger.
Copy that.
We thought maybe a herring earlier, but
it does look very trout-like.
I got you at 8.5, Lars.
And trout, okay. Oh, and another one of those stalked yellow sponges. It's always cool to see.
Small fish just behind the...
Look at all these really big primnoas. Yeah, just a really small version of that one. Huh.
I don't suppose they could be juvenile salmon.
You're asking... You're asking somebody that mostly studies sponges and corals, so... I'd have a really hard time. I get a feel for the ones that are in my study sites, and unfortunately beyond that I'm not very helpful.
I know my only experience is perhaps trying to catch them.
Wow. You know, swimming to kind of stay in the same place. There's still quite a bit of current here.
Laser's clear.
This is
3 shot.
Beautiful. Thanks for that.
Yeah, I'm just
Someone will be able to ID that fish from this excellent video. Fortunately, not I.
Straight damage. 9-0.
Copy that. I'll push forward. Push forward after this coral.
Wow.
Lars, how's the current
Again,
look?
another contender for a size record on this dive. That Primnose is giant.
Trying to wrap my head around it.
Ugh.
Oh, you want to look at that seed pin stick to the right?
Copy it. Single stock.
Yep.
Surface current 0.6 of a knot. Just east of north.
Put the lasers at the base of it as you go in.
Lasers are on. Going for the base.
Yeah, I think this is another one of those sea pens. Halipteris, maybe? Which I keep on forgetting is the old name. Balticina is the new name. It's been recently reassigned.
Start at the base and move up.
Yep.
Cool, so there's a kind of a larger swollen end on that branch called the peduncle of the coral that's anchored into the sea floor, kind of inflated with water to keep it there. Oh, we've had an ID on those little fish, Eulachon. I'm glad somebody's out there that can help us. So you can see each individual polyp of this sea pen, kind of like a different version of the polyps we were seeing on those really big corals. They're both octocorals, so if you were to count those little tentacles coming out, you'd see 8 of them. But the Penitulacea are in their own group, and they all kind of have the same form of one long central, pen-like stem with the polyps coming off of it.
No, I thought that was a good
Often
speed.
see them associated with soft sediment with modifications to bury the end or the peduncle in the softer sediment. Wow, look at all these really huge primnoas. Like a bit of kelp degrading and caught up in the branches of that one. A really big pom-pom anemone, that Liponema we've seen earlier. A couple of the Tritonias, and lots of these little lacy hydrocorals in the pink and white, probably separate species.
Look at that,
most likely Stylaster.
This vantage point, just sort of rotating to your right.
Left coral rotating to right.
Yeah.
I can secure your starboard lower if you want.
Sort of pan your, yeah. That lower section of it is very different from the upper section. Like almost like two different corals.
Wow, just beautiful.
Okay, ready for zoom?
Go ahead.
Seeing a little bit more predation on the bottom of some of
no,
these.
just between
One of those
two different kinds.
nudibranchs.
Right there.
Some kind of thinner, sparser looking branches in this forward one, which is really interesting next to the kind of typical version. Ah, I see the foot or an arm of one of those big Hippasteria sea stars.
Oh yeah, on the other side.
Yeah.
Yep.
I
Giveaway.
knew they were in here somewhere.
He was hiding, but
yeah.
Or left, and I'm going to zoom in on the polyps on the left corner.
So it's really interesting. I often wondered if kind of the thinner, sparser branches wasn't a different, maybe there's a variant of this coral that I haven't seen myself, but it looks like it's all attached. So probably the same one, just with the tentacles a little bit droopier, maybe.
Right there is closed and open. And I'll pull out.
Very interesting. It's maybe just different stages of growth or feeding. We have a collection request. Is this an okay spot if we were looking for a Primnoa piece?
Yeah, I think we're pretty comfortable. Ship stationary. You're looking down at me 60.
Yep.
What exactly are we looking for?
Just a snip of any Primnoa you like, as long as it's got some nice tissue at the end.
And that's what we have directly ahead of us?
Yep.
Okay.
These red kind of tree-shaped corals.
Video's clear.
Well, I suppose kind of orangey pink, actually.
You got starboard outer left, so it doesn't really work for your setup, unfortunately.
Starboard outer.
Unless you come in straight at it.
It's the only thing that's left.
Yeah.
Okay.
You could probably put it right in between your toes, and then just come across the side of it.
Okay. Set up as is what you're saying about.
Well,
I think if
you turn and put it in between your toes, come directly at it, then you'll be able to potentially get the drawer out.
Gotcha.
Or we can just pull back and get the drawer out now, or cut it, come back off, get the drawer out. You know what I'm
I
saying?
think it could mix with starboard outboard if you wanted it to, but it looks like starboard inboard is also free.
Okay.
But I think it's just a rock, so it shouldn't get mixed up.
Like a little shelf just below it, you might be able to get your toes on it. That would be able to drawer out.
Okay. I like that idea. Worst case, this is also something that we can get a good draw lock on.
Yeah.
We can always pull out if we need to.
Yep.
Might not be a terrible option to pull out the shilling cam for this.
Yeah, if you get nice and steady, let me try.
That is tall.
So again, we took a small piece of Primnoa on an earlier dive. This one will be really helpful to have for comparison. There's something very interesting going on with the population genetics of this large habitat-forming coral, so it's of extreme interest to scientists to be able to take a look and compare as many samples as possible from this area to others from around the Pacific.
I'm going to put your augs over here, pilot, so you have that view.
Good, thank you.
And of course, any slight morphological differences that we might see that kind of correspond with those are always helpful when trying to decide, you know, what the extent of the species is. With that information, we can have a better idea of what kinds of you know, vulnerabilities they may have.
So we can keep that in.
It's always helpful to have as much information on their distribution as possible.
Yeah, there you are.
Perfect. Thank you.
Yeah, I got it on the drawer.
Good.
Stand by.
Always. Right, watch lead. Where this is at kind of an odd angle to be able to open up the drawer once we get it. We're going to set up our shilling cam to kind of give me a sense of depth a little bit out and forward so I can see exactly how far in I'll be cutting.
Sure, is that the little arm cam?
Yeah, so our we have 2 hydraulic arms.
Yes.
The second arm has a camera on it that we can manipulate a little bit.
Oh, sweet, I see it.
That's a great view.
Yeah, and then if you could just get it so it comes up a little bit. Go I think that's
Oh, now I see the screen that it's coming out on. I love it.
Tell me how tight you want it. You're going right for the front, right?
I'm going for the top is what I was thinking.
The top, copy.
And just, you know, a small snip is all we need for our purposes.
Okay.
Thank you.
It's touchy.
It's actually pretty close to what I was looking for.
Yeah. I want a copy of that arm cam footage from here. This is very cool.
Alright, that's probably as max top I'm going to get you.
That's good. Can you just bring me either shoulder outboard or forearm roll just slightly? Just to lift it up a little. The other way, yeah.
To come all the way around.
Okay, that's fine. Just leave it there. Is I think good enough.
Okay.
Right. If you could bring me back over.
Yep.
I'll bring my main camera up.
Sorry. Still had it.
Ooh, flatfish flying.
All right. See a pretty big flatfish flying through in the background.
Arms live.
Just sliding back a little bit there, Lars.
Okay. It's probably the additional.
Yeah, you got a wind vane out there now.
Okay, so we just want a little off the top, correct? Watch this.
Yes.
Or so.
Yeah, maybe. Yeah, that's great.
Okay.
Thank you.
Close and draw.
And they're pretty tough, so anything that's not, you know, snipped might kind of hang on.
Okay.
If that helps to know.
Yes.
Sample.
Beautiful.
Okay.
starboard inner here, pilot?
Yeah. I think I might have to pull back though. Do you think you have enough room, co-pilot?
Yeah, it looks like we're in pretty decent shape here.
Okay. Hold it here. Go ahead and bring the door out, please.
Coming out easy. Ah. I just fired a Niskin by accident.
Copy. Nav, can you just note?
Yeah, I'll note that. Bottle 4.
Yeah, bottle 4. Oh.
I don't mind a bonus Niskin in this area.
We got starboard inner. Is open?
Yeah.
Oh, I thought we were doing outer. Sweet. All right. I think I can get in from there. Coming out. Coming out of grip lock.
Sticky. There you go.
Whew. Beautiful.
All right, George coming in easy.
Please, thank you.
And watch leader, are we going to count that Niskin as a sample, or do you want to?
Yeah, I was just going to ask you maybe you could make the Niskin sample 9 to happen first, and then biology sample 10.
Roger that.
Okay, arms secure.
You told me just in time.
Hydraulics coming off.
So we just did a Niskin?
Hydraulics secure.
Okay, if you wouldn't mind just stowing that shilling real quick.
Yeah, let me get D2 back out here. Or Art. What is this?
Okay, I can.
Well, actually, let me look at the arm before I put it away.
Okay, so I have the Niskin as sample ID EX2306 D1109W, Niskin bottle 4 at 325 meters.
Nev, did you copy the video? Copy.
Nav copies.
I don't know what our angle typically is where we set this. Is it looking at the left
starboard
box or past it?
I'm not quite sure how you guys have been leaving it. I assumed it was just stowed, but it wouldn't hurt to have it just inside of that, excuse me, that port rock box, just so we still get a little bit of video without having to worry about it being an obstacle.
And the Primnoa EX sample ID EX2306 D11-10B in the inboard bio box, starboard inboard bio box.
10B, copy, thanks.
Okay.
Beautiful, thanks for that.
All set, co-pilot?
Yeah, we're good to go.
Nice work. Okay, coming off bottom.
Nice sample, pilots. All bio boxes have been filled. Excellent. Unless you want to double up.
Well, filled once.
Filled once, there you go. And we have one remaining Niskin.
Perfect. Done our Niskin duties today. It's really easy to get distracted and forget.
I don't think Nate was able to get a frame grab on
I could find one.
Unfortunately.
Yeah, I think.
So they might not be able to use it.
Okay. Do they require frame grabs for them to be official? Is that the...
Yeah, I think they have to have imagery to go along with it.
Copy. Put your jar cam back up, Pilot. Or sorry, your still cam.
Thank you.
And you still have all those canisters left.
Correct. 3 canisters that are untouched, and if we need to go back to number 1, we can use that as well.
Another one of these ridges. It's pretty clear which way the current's running through here.
Yeah.
Yeah, I think I have an idea too. Wow. So this is the abundance of primnoa that I was really hoping to see. It's very exciting to see that this model has done a really good job at predicting where we might find an abundance of these corals.
See if it's similar.
And in this high resolution bathymetry, you can even make out these little mounds that are no more than a meter or so high that they're on, as opposed to the flatter, more sedimented areas in between where they're still present, but a little more few and far between.
Huh? Yeah, I haven't been keeping a close eye on this because I've been so focused on the corals, but that is a really interesting observation to note that they're, less on the kind of sides of these cliffs where I was kind of thinking they might be more on these little bumps.
Getting close to that 320 contour.
Yeah, that could be the start of whatever mound is over here.
Watch leads, I don't know if I'll have enough room to duck down into this little hollow, but I will.
Oh, that's okay.
Right across to, if you're looking at this sonar.
Yes.
We're getting a pretty hard return directly across, maybe 20 meters out.
Yeah.
Okay, yep.
So we'll push for that.
Sounds good.
How stretched out are you guys?
So we might have a little bit of
I've got you 4, 5,
little bit of
6,
space from the bottom as we kind of traverse this dip.
Yeah, we'll probably
Then we're approaching a wall-like feature, so we should see more shortly.
260, copy. Let me know when you guys are ready for one.
Yeah.
Ready. How long?
20 meters.
20 meters, 260, and work. Bridge RV nav.
I'm going to send you up Mars 260.
We'd like another ship move, please. Range 20 meters,
Can,
bearing 260.
really easily see which way the current is flowing, carrying all of this marine snow. Still seeing little Ketignas and
good copy. Thank you,
shrimp swimming by.
Copy, coming down with you.
I want to keep that coral in view to help me see where the bottom is.
Ship move and work, Pat.
Copy that. I won't stretch too far out past this until we get that ship move underway.
I can come down just a hair so you reacquire bottom.
Copy.
More of those little silvery fish. I think they are Eulachon. So it's interesting. I was sent a little bit of info about them. They're anadromous fish, which means that they move between fresh and salt water. They're a kind of smelt, which is one that I'm a bit more familiar with in some of the small rivers in PEI.
On that move anytime, Copilot.
Copy.
found from Northern California all the way up to here in Southwest or Southern Alaska.
Is it 260, John?
260, aye.
Copy. You good if I center up on 260, Elijah?
Yes. Okay.
Wow, another huge Primnoa and an almost equally huge Hippasteria.
We have a little bit of time here, video, while we're waiting for the ship to catch up.
I'll come down here just to give you a little bit more leash.
So we have a bit of time waiting for a ship move. We could take a good close look at what we see growing here.
All right, I'm lined up on 260.
Copy that. Nice. Okay, video. That should have a toe in.
Wow. So you can see even some, you know, dead and damaged polyps, those kind of whiter ones that are full of debris. Look like they're
where the sea star's been eaten away.
where, yeah, maybe damaged by this sea star. And then some really beautiful kind of pinkish, you know, living and healthy looking polyps around it. And a very sharp transition between where the sea star's arms can reach and where the coral is happy. See that really beautiful kind of golden skeleton of the Primnoa underneath. Some little shrimp. Every now and then I can pick out the eyes, and then I see there's a shrimp attached to it. Otherwise, they're very well blended in.
If you like video. Understood.
And a really good view of the Pedicelaria. Again, Chris Ma tuned in earlier to tell us a little bit about these structures, and they're kind of unknown in function. Not all sea stars seem to have them, from what I can tell, and they can kind of open and close. So they're a little bit mysterious, maybe for protection from bigger sea stars that like to try to predate on them. But they're on both sides, on the top and the bottom. And it's just speculation. Scientists apparently still don't know.
Pilot's clear.
I'm seeing lots of brittle star arms and lots and lots of hydrocorals at the base of this really large Primnoa.
Okay, I'm going to double check with Fernando. I think we may just delete that one.
Sounds good.
And then tell everybody that the next one will be 9, so we don't have to change everyone down the line.
Okay.
But I'll confirm that.
We do it. We have the next biosample labeled as 10 at the moment, so yeah.
Okay. Oh, we did it. Okay. Yeah, we did the Niskin and then the bio. My bad.
Yeah, that happens.
Lars, you had the very rare opportunity of having the great art shade your cameras.
I noticed.
That's an extremely rare opportunity.
You did a great job.
Oh, I never doubt that.
Some more Primnoa over here, and hydrocoral and a little sea urchin. Maybe a bit of a dead skeleton, some, I think a piece of kelp maybe. We've been seeing some kind of stray pieces blowing around. It's my guess, unless, yeah. I was like, unless it's a rock. No, I'm pretty sure it's kelp.
It's maybe up against a rock.
Mm-hmm. A really big Liponema anemone off to the right.
Red there, bottom right. You see that? Tiny, just slipping off screen.
What is it?
I don't know. Small. Just below that rock. Yeah.
Shrimp.
Is that what it is?
red one?
Yeah.
Yeah, to the left of that little bitty sea star. That'd be a good subject. We haven't gotten any nice close-up.
Can you maybe zoom in on that dead coral skeleton with the tissue kind of around it? See at the base of those two rocks?
Oh, I see, yep.
Yeah. Just a branch, and I can't tell if that's coral tissue or something else kind of growing around it.
Yep. Coming down.
Yeah. Okay. Oh yeah, it does still have some tissue intact, so it looks like that snapped right off from somewhere, a bigger colony. Huh. Very cool. All right, thanks for that.
No problem. So there was something red.
Yeah, that's a shrimp. I think Roland was talking about that little sea star right there.
Yeah, it's right to the left of that sea star.
Go ahead and come in.
Left corner.
Left of sea star.
Little shrimp.
Bottom left corner. Striped arms.
Oh, gotcha. Thank you.
Beautiful.
Looks like a little pill bug right next to its
the some of these I think were called the candy stripe shrimp. Not sure if this is the species that someone in the science chat was referring to earlier, but definitely see the really stark, distinct striping on the legs and a little bit on the carapace.
Yeah, it came over outside into that current. Yep.
And probably a Henricea sea star beside it. We've had a... Oh, look at that snail. I almost didn't see it.
Why?
Wow, it can really move.
That is fast.
Yeah, that was fast for a snail.
I thought at first I thought it was something
rolling.
Yeah, whipping around in the current. But there's another one to the right.
Oh yeah! Wow. Yeah,
they're very well disguised.
No kidding. This covering of debris made them nearly impossible to pick out, you know, from the rocks beside it. Just it wasn't until we saw that one really move.
Or are those rocks?
Or are they? Now I don't know.
Actually, there's a green.
It could be.
Right,
we've seen urchins covered in all kinds of debris. We've seen decorator crabs, I believe, with a little bit of
left of that white
tissue on the top.
Left of the snail.
Okay.
Clear.
Right.
I think they just piped your meeting.
Oh, understood.
And a little tiny, cute little fish. Right at the bottom, maybe something like a snail fish.
Edge, center.
So we have Fusatriton oregonensis, I think is the tentative for the ID for that gastropod we were looking at just now. So we're just doing a quick shift switch with our ROV team. We'll just pause to enjoy the scenery at 325 meters depth here in the in this part of Cordova Bay, just about to approach another one of these kind of lower, less steep wall-like features, I think. Excited to check them out for possible Primnoa colonies.
Yeah, science called the fish.
Yes, love to take a look at this little fish.
There's a big one on your
Oh, they're so cute. And a little tiny anemone beside it. Wow.
Oh, that looks like, we haven't seen one of those.
No. Kind of looks like a little snailfish. Yeah, I don't think it's an eel pout. I don't think it's a grenadier or rat tail. It looks like maybe a little snailfish to me, but...
Sorry, watch leads. We
no, that's
front
fine.
row. We were talking on the fish we saw on our rail camera that looked different. So that may have been some of the confusion there.
I was like, the fish that's right in front of us.
Very clearly not a rat tail.
I mean, it's got a long whippy tail.
Center it up a little bit.
I don't know. We haven't seen any rat tails on this dive yet, so it'd be very
Did you
interesting.
see where it went?
Yeah, I think he swam off the starboard.
Cute. We did see a skate though.
His nose was so flat.
Yeah. We're on top here, so we've run out of dive track.
Wow.
You guys are just going to have to
Rubbing.
figure it out. It's pretty flat. I'll do a quick pan on
Let's get lasers across it.
Mini Zeus here so you can get a view.
Wow.
Oh, there he
That's
is, off to our
beautiful.
starboard again.
Yep.
Let's
That does
see.
not look like an eel
Where?
pout. On the starboard rail,
Is
he's
that
just going off.
a
Let's come
skate
wide.
maybe? A chimera
Lasers
maybe?
across
a
the
small
skate.
fish.
Let's go take a look then.
No, we're
we
going
need.
to take a look, roll, and we'll come back.
Yes, please.
You'll need the lasers.
Ooh, really
you
big
went. There
brittle
you go.
star. But yeah, I'm also interested and invested in this fish ID now. Where did it go? Just saw a little glimpse of it. Oh, I think it might be, oh, let's take a look.
Do you see it, Watchley?
I do. Could be a chimera.
Where can you direct me? I'm not seeing it yet.
Well, I just see it in the other camera. Sorry, I'm cheating.
Oh, it's still off to starboard. Yeah.
Is that starboard? Okay.
Roger. We're going quick go.
I'm going to pull up all of my fish guides.
Is that him?
And yeah.
Swimming away from us fast.
Yep, we're going fast. We're just going for an ID here.
He landed.
Ooh. Oh, it's so hard to tell without the face, but it looks
Yep, I'll come
roughly
around.
like the chimera.
Oh, he's turning on me. We're going to go back the other way. Keep it wide, rolling.
Lasers clear.
Laser's clear.
Let's see.
Okay, let's go in. Tilt me up just a hair.
Whoa, beautiful. Yeah, that's
We're
definitely
gonna have to let
my.
it go pretty soon.
Oh, these are beautiful images. I'm just pulling it up now, but yeah, it looks like a chimera fish to me. It's not even in my guide. They also call these
I'm gonna have to let it go.
ratfish sometimes, or I see a ghost shark. It's a cartilaginous fish.
He's gonna see what's going on with this soul.
He's deciding to work with us here and stop.
Cute.
Because I had some big corals I had to stop for.
Thanks. We break for corals on this vessel. Wow, beautiful. Yeah, we definitely haven't seen one of these. I'm pretty sure Caitlin mentioned this this fish exactly as being very excited about. So I hope she's watching. Wow, okay. So yeah, this species definitely isn't in any of the guides that I have, but one of the chimeras. So I guess
he's like in a little eddy.
Oh, just beautiful. Closest living relatives are sharks and rays,
We see
though their last
under
common
his rear.
ancestor with them lived
Yeah,
nearly
you can go
400
in.
million years
Try-
ago.
seems to be
Species are largely
complying.
confined to deep water. Very cool. Just
Look at his
beautiful,
front
really
mouth.
unique. And look at the like iridescence in the lateral line.
Following. Stand by.
And the markings along the face under the eyes is so something I haven't really seen in any other kind of fish. Oh, they're so cute.
He's coming in towards us.
Wow, look at this.
Okay, going for still. Going for still, Cam.
Oh
still,
my
Cam.
gosh. There's, oh, wow.
That's full
Really bright. Okay. All right. Maybe taking a bit of an aggressive stance. You really wanted
we're
to
gonna
Tired of
back
us
out.
coming after him.
That's great.
And back into view.
Sure.
For,
Suspect he
Sirius.
had more to do with the current blowing him in than
Wow.
attacking us.
Thanks for chasing that one down. That was a very
45
cool
pound.
catch.
Roger, coming back to you.
I never would have seen that coming out of the starboard cam, so that was great.
Good eye, Chris. Way to watch for him. Oh, Chris jumped ship.
And doubly made Caitlin's day, I think.
All right, pilot or co-pilot. I'm coming back to you.
Copy.
Gonna find a clear spot here and then Come back down.
Lasers back on.
Big corals.
Another note about the Chimera, they're very abundant in Puget Sound along with six-gill sharks.
Lasers back on.
Yep, sorry.
Very cool.
Lasers are on.
Sorry, Chris, we got a request for zooming on that brittle star, and I just, we saw it ages ago. Definitely got distracted with that charismatic fish. But if we see any more, I think he was talking about
large,
those bright red brittle stars. We'll definitely take a closer look next time.
Coral rip.
And more of these huge Primnoa colonies. Could we get the lasers on this one just to get an idea of the size?
Lasers are on.
Lasers on.
There they are. Thank you. Wow.
I know we've seen a lot of these,
Yeah.
actually, so we won't spend a lot of time. We're just grabbing a little footage of the nice background here.
Sure, I'm happy with that. I could look at these all day.
This seems like one of the taller ones we've seen.
Mm-hmm.
I'm going to push you in video.
Yeah, definitely.
Same
I
for
think
still.
each of these probably grow a little bit differently
hold
depending
there,
on
video.
the specific currents that they're exposed to.
Yep.
Trying to brighten up the inside.
And for
without
some reason,
washing
this
the
one
backside out.
decided that, you know, up
All right,
was
let's
a
get,
little bit
I
more
think, a little fog.
advantageous
30.
than growing out.
It's 38. Turn it down.
No, it's fine.
Beautiful. Just the really deep orange color and the, you know, kind of black
Let me know when you're
water
good,
column
I'll
shot
tilt
in the
up for video.
background. These are really stunning.
Roger. Tilting up for you, Roland.
Okay.
See lots of these little arrowworms kind of flickering around in the foreground and the background in the water column. And just a really
Pull slow out.
impressive abundance
Keep
of
it
all
at
of
the top
this
edge of the screen.
marine snow.
Keep it towards the top, so tilting down.
Yep.
Yep. Working on it.
Wow, these are so beautiful.
That's full wide.
Roger. Yeah, I tried to get as very close as I
Hold
can.
there. Get his lasers one more time.
And our fish is back. He's right on the other side of this coral.
Video.
Oh, okay.
Yeah, so if we, there
Oh,
he is.
there he is. Yeah.
Off to starboard. All right, let's let this go.
Yeah, you're still full wide.
Yep.
Wow. And according to Elena Jorgensen in our science chat, there probably is a male chimera.
Fog lights are
Not
clear.
sure how you tell the difference, but it's a good observation to have.
Nav, do you have another move for us?
So whatever you guys think slope would be here. Last move is 270 directly west. Or whatever you think blue view and sonar is going to lead you.
It's hard to tell. It looks like a little higher on the at 270.
Wow, we're seeing a couple of different things now. One of a really big
270.
boot sponge or Rosellid sponge.
Right now.
A couple of those large sea pens, the
That
Balticina.
tends to be uphill, kind of 270.
Liponema
Copy that.
Direction.
pom-pom anemones,
I can set
and
her up.
a few really large colonies of
We're gonna set her
Primnoa
up,
pacifica.
Roland.
Some of them have
Get ready
fallen
for our
over.
next move.
You gentlemen interested in
It may be a
20 meters?
different species of rockfish
Sounds
than we've
good to
been
me.
seeing. We've seen some white dots in the pattern on these ones.
Copy that. Let me know when you want it. It'll come quick.
I think I'm ready.
Co-pilot
Co-pilot's ready.
Let's do it.
Heading up over this bump.
Wow.
A whole bunch more over here on port side.
Beautiful. Yeah, every kind of ridge, area of exposed rock, we keep on seeing more of these really massive Primnoa corals. Look at
wow.
this.
This is the best forest yet.
Yeah, this is amazing. Can we get the lasers on just to get some
of course.
broad measurements? No, that's fine. Thank you. Where are they now?
Let me put them on the front coral. There you go.
Oh, thanks. There we go. Yeah. Just to kind of get an idea, maybe around the base of some of these. Just
we'll go to the right. That's a nice exposed base.
Sure, thanks. Just thinking ahead for the person that might want to annotate this video. They'll probably have to count each individual coral, and they might even be asked to do their best to measure them. So try to help them out.
Another base.
Wow.
All right, we'll just
look at all these. Yeah, I'd love to scan this area just to get an idea of the extent.
Try to light them up for you.
Thank you. This is fantastic. So yeah, I think we'd call this like there aren't any hard and fast rules, I don't think, but maybe a coral thicket. Just a really dense population of these.
I like coral forest.
A coral forest, sure. Yeah, I mean, if you're the size of a sea star, it is a forest. And even some of these fish we've seen kind of associating with these corals. Every now and then we've kind of
I think it's time for me
scared
to
one out.
come up and move on.
Sure. Wow. Really big rockfish coming into view.
What was that video?
Big fish in between
So you
corals,
can see why,
partial zoom
yeah, an
request.
area like this is of great interest. You see lots of smaller rockfish kind of scattered, especially close to the bases of some of these corals.
45.
But you can see how they would just completely change the dynamics here.
Yes, sir.
Give smaller animals a place to hide or rest, and maybe even brood.
I'm hauling in right now.
Roger.
They've been running close all day just to be able to see through the...
Not this close.
Wow.
Do you want me to go keep a larger delta?
You know, and we are seeing if some of those Hippest area sea stars predating on the corals, but in general, a lot of these are pristine. A couple of different sponge species in between, and lots of these almost, they look miniature compared to the Primnoas, but these little hydrocorals also in pretty high abundance here.
Hey, Nav, would you compare,
wow,
does the CTD reported depth for D2 and Sirius agree with the delta that we're seeing right now?
Stand by.
Thank you.
Yeah, I would say so. Yeah. So yeah. Sure, yeah. So this is yeah, the area that the coral distribution model that we were going off of to kind of use for maybe a site selection when we were picking out this place, you know, took in all of that different data of catch records from bycatch, from fishery surveys, as well as a bunch of different pieces of environmental data. And it did highlight this area kind of as the high point, you know, is one of the darker red spots out of the whole Gulf of Alaska. So we had a pretty good idea or high hopes of seeing corals here. And if not, it was really useful to have to kind of ground truth this model. But we're definitely seeing what I was hoping for, which is, kind of these really dense thickets or forests of Primnoa pacifica, which was one of the species selected out, or the primnoid corals, these larger structure-forming corals that the model was looking for and looking to kind of describe the distribution. So this is kind of giving us a lot more information about the small-scale distribution and where we might find these corals in other places where we can't get to with something like an ROV. We can use it to fine-tune the model and help us predict places that might be good habitat. So I'd say this is a really dense coral population that the model predicted would be here, and now we're seeing it. Now we're seeing imagery of it. It's been a very, very useful observation, and just incredible to document such a dense amount of these really large, long-lived corals. We just keep on seeing more.
Camera doesn't seem to enjoy this lighting.
So I think all your lights are off, right?
Looks like it.
Yeah, it's not
That'll
really
explain it.
So seeing a little bit of lithologic difference here. Just we initially came up a more massive outcrop where we saw the big forest to our, which is I believe to the left here on the view, and then over here to the right we see a ridge that looks a little more. It's got traces of bedding, or perhaps those are partings from the schistosity or the foliation in the rocks, and not so many corals on it.
Let's check on deck the starboard lower swing arm.
What is?
Looks like it's wiggling.
Yeah,
Schistosity. Schistosity, yes.
Schistosity.
That's refers to the texture of a schist. It has that, we call it a foliation, but that general flat parting.
So it is
that's
to like when
that's due to the minerals that make it up.
That forward is kind of right there.
Yeah.
We noticed this last cruise shakedown, one of those.
Yeah, but I thought the port.
But we didn't come up with a good answer.
I thought the port one would also kind of see it. No?
Maybe. I remember the starboard though being.
Okay. I just remember those swing arms were coming loose every time. All the time.
I think we checked them and it seemed secure, and I'm not really sure why it was wiggling, but I mean we should check them again.
And it's interesting, the little hydroids, corals seem to stick pretty close to the bigger primnoas.
Would prevent it from the starboard side.
So, kind of losing the bottom here.
You're going to do, let's see, moves halfway through.
Well, there's more corals. Unfortunately, I think this area is a lot of undulations.
Yeah.
And with these tall corals, it's kind of hard.
ROV watch lead.
Yes, sir.
Yeah, I believe we're kind of to the top of the last ridge, and we're going to be going down. If we keep heading west at 270 for a while, we might want to make a right turn and head kind of northwest up this ridge, or and eventually there we will kind of top out as well. But
sure. Yeah, I think
that might be the best. Might be the best way to go.
We'll head there.
We'll do some spins. Do you want to finish this ride out this move? There's 5 meters left.
Sure.
Okay. I guess look north.
Yeah, we might want to continue down this a little bit, and at 270 for a little bit till we get to the end of what I think is the outcrop, and then we can maybe turn and head north from there.
Okay.
Although, yeah, like I said, we would be heading down, so that might not be the best way to go.
Yeah, down is hard for us, especially in this visibility.
I can see almost immediately, it's getting dark.
Yeah, we sort of just look into nothingness when
right. So yeah, this might be a good time to make a turn to the right.
Yeah, I think that would be best for us, watch lead, if you're okay with that.
No, that's good.
What is that object? Lower center.
Snap.
Move complete pilots.
Copy.
Sea cucumber?
Yeah, that looks like a great sea cucumber.
Yeah.
Is that what they're called, or is that just a really good one?
This is just a really good one.
Okay.
Hold on.
Yeah, I'm going to come to starboard. I just don't want to take out a wall of coral while I do it. Kind of just trying to point that way. Okay. Yeah, you can start cheating over.
Okay. Okay, I'm almost there. All right, I'm due north.
I think we can start going that way.
Copy that. You just want to start with 20? Due
Sure.
north?
Sounds good.
It's coming.
Did you want a closer look at the cucumber video?
Sure.
Yeah, that'd be great.
What's the current feel like? We're not getting good.
It's coming from behind on me now.
You're getting a little stretched out too.
Yeah, I got hold here. Go ahead.
Nice trail of droppings.
Yep.
Doesn't look like he's made a whole lot of progress.
No.
Kind of just hangs out right here, and he's found a nice place. He must be
Beautiful
finding
texture.
plenty to eat.
I would imagine there's a lot of little organisms in this sediment or debris on the top of the rocks that he could eat. Earlier you were asking, Jamie, whether the difference between a nudibranch and a sea cucumber. A nudibranch is a mollusk, so basically you can imagine that as like a sea snail without a shell, while the sea cucumbers are echinoderms, and they have, usually when you hear echinoderms you think of the radial symmetry, the pentaradial symmetry. In sea cucumbers, you don't see that on the outside as much, but inside their breathing structures.
Oh, interesting.
Where you
Okay.
see that symmetry.
Full screen.
Yeah, but basically these guys just cruise along the bottom and filter through this detritus and sediment, and
just it wasn't good for the.
Yeah.
It wasn't holding.
Little vacuum cleaners on the bottom of the sea.
I just didn't have enough.
So if I think of it as kind of a one-armed sea star, inaccurate as that might be in detail, it'll help me remember.
There's a little lobster or crab underneath an enemy.
Red thing, an enemy. Got it.
What's this here?
Okay, I'm rotating down.
And that looks like a
crab?
decorator
crab, yeah. With
a
an
decorator
anemone
crab who's
on
decorated
top.
himself with an anemone. An anemone.
Or is he just hiding underneath?
Very interesting. Yeah.
Video when you've got
That's interesting. I wonder how well that actually works for the crab.
Yeah.
Or is he just hiding underneath of it?
That could
that's
be.
true too.
Tether looks good.
Yeah, maybe he's just hiding underneath there. It's funny, you pan out and you see, oh boy, not much going on, but you zoom in and
every now and again,
I think we're passing through to the other side, one of those, that coral forest we saw earlier.
Yes.
Well, he stripped that, didn't he?
Chunkiness on the control video.
I slowly
Sea Star is
adjust
eating the middle
control
out of that one,
very
but
slowly. It doesn't respond, and then it makes a drastic change.
Gotcha.
See that bright reflective thing? The center screen right.
Oh, there goes
Tina 4?
a jelly.
Don't. I need to straighten up though. Sorry, Roland.
I think we may be starting to get into very small fish.
Something just out of view to the left.
Reflective.
Don't know.
Like quarter-size hatchet fish.
Roger. I can look at the next one. I was getting out of position, especially with my upper swing arm out.
You want me to pull them up a little bit?
No, just where I was turning and how the tether's pushing, or the current's pushing the tether forward of me.
I thought I saw a big line in the Sirius view. I wasn't sure if that was our tether or a fishing line.
You guys wanted to move? Looks like you're...
Yeah, I think so. He's getting out front.
You happy with north still? Ready for another 20?
Sounds good.
Yeah. It's coming.
We have an identification of a sedipid Tina 4 that went through the view at one point.
So I'm kind of in this trench here. Watch leads. You can see on my 2 rail cams. Very much of a
wow, yeah.
shoot that I'm in.
Yeah, this really deep kind of trench feature.
Very nice, yeah.
We're seeing, you know, kind of steep rock and coral on either side of us, with lots of sediment kind of filling it in between.
So I think I'm gonna come up on this starboard one. Get back more to center on your screen.
Copy.
How much bottom time do we have left, Sean?
A lot, I would think.
About an hour 10 minutes.
Okay.
Have you guys seen the flabbermouse view?
Yes, I have.
Is that a shrimp at the bottom?
Where?
Or no, that's hydroids or something stunned. I couldn't figure out what that was with the arms sticking up way into the water, but it's a hydrocoral with something next to it.
Yeah, I think there's the base of an older dead coral stalk and some other little things growing at the base of it.
I mean, that could be...
Thanks, Michael, for joining us in the science chat. We've really benefited from your observations.
Yeah. Watch leads, if you take a look at our blue view, you can see these are really, looks like 3 parallel mounds.
Oh, wow, yeah. Oh, very cool. It'd be
wow.
cool to to go over top of them. Yeah, in the sonar view, we're seeing 3 really big stripes kind of
We were
returning.
just between here, this first one and the second one. That's kind of that trench we were in.
Right.
Okay.
I just came up and over on the right side of it. And you can see it's really almost 3 parallel ridges.
Yeah, I think that is reflecting it's a compositional layering in the rocks where you're getting a little more resistant ledge and then interfingered softer parts and that are the low spots. I'd be interested in getting a piece of some of this stuff, especially where you can see that foliation or that bedding, those alternating ledges, but there doesn't seem to be much around here loose, nothing.
If it was, it might be kind of in between, and it's just full of all this sediment and detritus.
I think the last rock sample we got was probably this stuff, but being that it was out on its own, you're always never quite sure, but that's also why you grab what you can get when you have it. So
pushing past the skids.
Okay.
So we're seeing lots of these Hippasteria sea stars now.
Yeah, I'd say another one is still pretty far out in front.
Oh, and a cute little fish kind of at the bottom. Maybe another one of those sculpins. So a lot more damage from predation or grazing by this this sea star, and more of those kind of pyramid-shaped gastropods, Calliostoma, that we've been seeing. RV, could we zoom in on that stripy fish to the left of the lasers?
Stripy fish, left of lasers.
Yeah, is that what that is?
Let's come in.
Ooh.
It's a stripy
fish.
Wow, with the little, mask, little banding around the eyes.
I'll set up closer.
Very cool. That's something a bit different from what we've been seeing.
Yeah.
You want me to pull in your port lower?
No.
Is that another one to the right?
That one looks more like a rockfish to me.
Okay, yeah.
But this one...
lasers on as you go in.
There's a fish I know that we've seen in some of our other imagery called a war bonnet or a decorated war bonnet. Chirolophus.
Copy.
Wondering if it's maybe something like that. Yeah, it could be. I think I might see a little bit of a tuft on the top. It's kind of where it gets its name.
Oh.
Yeah, those little kind of embellished appendages that are protruding above the eyes.
Well, those probably sensory.
Is that full zoom?
This is my
or just
very
decoration.
vague guess. Yeah, I would guess that they have some kind of a sensory application, but
take it up to 60, please.
if they're really spiky, I suppose they could be for defense or
the water out. You can kill it.
To blend in a little bit. It does resemble some of the hydrocorals we've been seeing, I guess.
Do we want to get closer video?
This is
I'm happy with this.
Beautiful.
And try to reset up, get
So yeah,
real
nice
close.
view of the patterning. The only other times I've seen it, I'm looking at the pictures in my guide, is also in association with Primnoa. So it's kind of an interesting one.
Hold there. I think this is a good setup.
And a couple of different polyps in behind it. Maybe a different kind of stoloniferous coral. It's hard to see. It's barely reaching out from the layer of sediment and detritus that's on these rocks.
Video's clear.
Roger. Let's come wide then.
Did
I'm not close enough for still.
Okay.
Wow. Again, you get a sense of how
down a little bit
impressive the zoom
60
is on
down.
this camera. Something that small. Could we get the lasers just for a general scale?
Okay, we'll leave him, move on.
That's great, thank you.
You're full wide.
Wow, quite a bit of damage to some of these Primnoa colonies. And I think by now we know who the culprits are. But lots
Squid
of really
through?
big, oh, teeny tiny squid.
Bottle it or no?
Very cute.
No, we'll just let it kind of swim through.
Another rockfish.
Oh yeah, a couple of them around here.
Couple.
So it's possible the snails we've been calling Calliostoma, we've just had another suggestion for an ID of Acoya platinum. So a different genus, but in the Calliostomid group. Calliostomid. There we go.
Roger.
So if we have questions about this
Sorry,
snail,
Andy,
please
last time I was just,
let me know in the
I was
science
watching
chat
the
if that's something that we
communications
should potentially target,
and the
because we
information
haven't so far.
stopped processing.
Of course,
So
there's
then
always
it
a
jumped
chance
to
we
the
got
new
one on the
setting.
rock, but feel free to type in
Okay.
if that's something that you'd like us to collect.
Like
Otherwise,
we've
I think
seen
we'll
one of these?
continue scanning. Yeah, we've
Yes.
seen those. Those
Okay.
are those little sculpins.
Moving on then.
Thanks.
Gonna rotate a little bit to keep you in view.
Yeah.
Pilot.
Yeah, you're shooting a little that way. Do you want
So thanks
Seems
to
our next
Chong
move
Cheng
will have
for
to
that
Copy.
identification at Jams Tech. Not sure what that acronym stands for. I'm just barely getting a handle on the Canadian government
I'm sorry,
acronyms, so you'll have to forgive me.
Downhill.
I'm following you down.
Wow, some really dramatic,
Back to a delta.
you know,
10.
almost like boulders with these geological
Be careful
features
falling
at the
down,
ends.
Andy.
You have some terrain on your.
This is almost
port
something
side
we
there.
might be able
Yeah,
to find a
I've been
sample
watching
at the
sonar
bottom of
and
if
now
we
it's in
can
there. I won't go too deep.
get to the bottom of it.
Mm-hmm.
Feel
Oh,
like
there we
once it
go.
gets-
Japan Agency
just
for
once
Marine
it
Earth
gets, like,
Science and Technology.
even right now.
Thank
a
you for the help with
delta
the
of 11,
acronym. Oh, could we get a zoom on this
all you
white,
can see is the ox.
the white color morph? It's not something we often see together. A nice big flatfish swimming by. But yeah, if we could zoom on this white individual.
Roger, watch lead.
Thank you.
Just trying to. Yeah, maybe a move towards me. Yes. Keep
Like-
going at 340.
So that's the same
10
thing,
meters.
only
10
in
meters,
white.
0, 0 coming up.
Yeah,
Or
you know,
20
really
meters
stark
is fine.
difference.
20
Mhm.
meters,
Um,
0.
There's an area, one of our study sites, that has almost exclusively
So
the
it'd be
white
340?
variant,
Yeah, I'm at 340 right now.
and it's
That says
always very
something else.
interesting to see.
maybe
Oh, look at that Tinophore going
Oh,
by.
wow.
That was neat.
Or a siphonophore, maybe? I didn't
Siphonophore, I'm
totally
sorry,
catch it.
yes.
And I am also just starting to learn the difference. I brushed up on my pelagic IDs in anticipation of our midwater dive, and so I'm a little more on it than I was at the beginning of this survey, but not by much.
All right, video. Let's go from here. When I get a little more tether, I can.
So
come
it's
around.
always helpful to document when we see something a little bit different, so we know that there's a variety between a white color morph of Primnoa pacifica and the really dark pinks, oranges, and reds. So it's very cool to see them, you know, right side by side for comparison. We can see a similar polyp structure, the kind of highly armored polyps of these Primnoas. You can see almost individual sclerites, or those hard calcium carbonate skeletal structures embedded in the tissue of these corals.
Right, everyone, we've got, let's see, 1 hour left in bottom time.
And
today.
there's another one of those tube
Thank you, Nat.
amphipod tubes, those little tiny crustaceans that are making those little
Oh, yeah.
mud tubes.
Oh, they're making that, not
Yes.
just sitting there?
Yeah, I believe
Oh, interesting.
so. Yeah, this isn't something that I've ever seen before, but I think Sean Rooney piped in about it early on in our expedition, that it was a thing, something that he sees quite often. And now we've seen it in situ a couple of times.
Okay, gonna come around.
Beautiful. Yeah, look at all of the colors.
Let's bring the lights up to about 60-70%.
Lights up, Andy.
Very interesting. Just to see, it almost could be the same individual, you know, we can't really, there's so many different attachment points down at the bottom, and they are known to grow, you know, newer colonies will kind of grow on the old stalks of dead ones, so there's a lot of overlap.
Let's get lasers on momentarily.
And this one's relatively intact. It hasn't been predated on to quite the degree of its neighbors, so this is a really beautiful, you know, healthy-looking colony.
Okay, gonna hold here just for a second, and then I gotta turn back out.
Yeah, this is fantastic. Just getting the gradient of the different colors with the kind of dark, open water in the background. All of the marine snow floating by, and like a storm of these ketognaths, these long little arrow worms that we see kind of flicking around in the water column. Just stunning. Wow.
Okay.
There are tons of them. If we're seeing this many, I'm wondering how many there are right up in front of the lights, which is where they really like.
Yeah, every now and then we pan up and to catch a glimpse of what's up there, and they're just like moths.
Let this go.
Fish sitting in the right-hand coral.
Can snap as I turn out.
Oh, another one of those. Yeah, I think it's something at least very similar to the decorated war bonnet. Is the common name for this fish. The species I have
interesting.
Yeah, so they really seem to like, you know, the Primnoa.
Okay, let's go in.
Very closely associated here.
You got time.
Wow.
Neat.
Yeah, so I think Chirolophus decoratus is the species name that I have for one of these fish that we've seen, you know, not too far away in BC. So we could be looking at a different species, but it's definitely something like Chirolophus decoratus. just looking up, checking out its hair. You look great.
I think at the end of that move, I started to get into
remarkable.
Much deeper
Oh, these
water.
are beautiful.
Not much deeper.
So again, a really great example of how these corals create habitat for other kinds of organisms. Here is a really big fish that's just nestled right into the branches.
And of course, Chris Ma is only noticing the out-of-focus starfish, sea star.
Right.
Sorry, Kip, I'd say.
Let's get several seconds of this. I was just saying at the
I try pretty hard not to call them starfish, but it's good to know that that's like a perfectly acceptable common name. I think most people refer to them as starfish, knowing full well that they're not fish.
Right.
I'm floating here, so
that's beautiful.
ready to move on.
Very smooth float.
Even this kind of pan back is really stunning.
Okay.
It's the arms of another sea star at the bottom. Not sure if it's the same one or maybe something like a Solaster. Beautiful.
Love working with you ace pilots.
All right. Let's find bottom again. Back in center. Keeps going down. And I heard you say
another little squid
gained altitude.
Yeah.
Swimming on by. Possibly
Turn
a
back to
gonadid.
270 maybe, or like 300 or something. Cut it.
Yeah. Want to look around a little and see what works for you.
Okay, I'm gonna go over to 3.
Ships all stopped.
100.
Like a little jelly. That one might be the Solissimus. a little dinner plate jelly. Wow, look at the marine snow coming down
45
here.
right now on the rotator.
How's the current in this area?
It's pretty strong.
Yeah.
We still have our front lights on. That's why the snow is more noticeable right now.
Oh yeah, we could.
But the current has certainly come up a little bit, I think.
Okay.
We can bring down the fog lights
as we hit these different chutes of flow.
Yeah. Yeah, it seems to be really coming down here. The
What do you think,
marine
co-pilot?
snow.
Do you like that?
Heading?
So
Sirius
where you're?
looks pretty good. Oh, I'm sorry, I was looking at D2 sonar.
Yeah, I'm pointing due south again.
I mean, the high point is over, like, kind of where you are, where you were.
Yeah, I mean, that's what we just came off of.
Yeah. So
I can come up some. I'm coming up.
Okay.
Just to cast our. Do you want to put more range on nav?
Got it.
Gonna hold here.
Okay, we got a 10-meter delta.
I mean, it looks like if we continue this direction, we'll find it again. Find bottom.
Where you were at 240.
So we've got a little bit of a dip here.
The other way
ahead
you're
of
so I'm matching your
where
heading at
we're going. So get a
It's
little
out
bit of
there.
a view of the water column instead.
I had
Of
problems
course,
with
we've
that
been
before I
seeing just
reselect.
thousands of these little Ketignaths flickering through the water
Yeah, I
column.
think let's just push forward.
Yeah.
And keep going. Yeah, maybe.
Yeah,
west
there's
again.
a little indent, but
Oh, lots ahead of us. There we go.
we can see it on the sonar.
Yeah, the sonar is really lighting up, so not too far ahead of us. We're seeing another big rocky area.
200, okay.
Lots of little fish down there, I think. Could be myctophids or something else.
Earlier, I had to reselect it on the KVM because the mouse wasn't working.
Yeah, I don't like that.
I think I lost mouse control.
On all stations, huh?
Yeah.
Try it not on the black, maybe?
So we've been given an ID of
Is that it?
Euphisa anthomedusa for one of those jellies we saw earlier.
All right, Andy, I'm going to push in.
Okay, I'm pushing down.
Kind of to whatever this is.
Thanks for that, Dougal. Lots of Ketignaths and amphipods. And also possibly a Euplocamus.
I think we'll just go another 20.
Another species of
70.
comb jelly or ctenophore.
Copy that. I'll put it in once the ship's done moving.
Okay.
Roger.
All right, I'm cheating over to 270 now, pilot.
Roger.
Okay, I'm pretty much there.
Okay, so coming up to another one of these large rock features we were seeing lighting up in the sonar, and again, it's just covered in some really large Primnoa colonies.
I'm pretty far out there, so I need to see if I can find something to look at.
Smaller rockfish hanging around.
Looks like there's
some kelp.
Something kind of tall off.
Caught up in the base of one of these corals.
Yeah, I think that's kind of where we were.
And a lot of those kind of yellow splotches on the rocks could be the bases of older dead Primnoas. Could possibly be some encrusting sponges, but they look like they could be Primnoa holdfasts from large ones. And of course, some of these smaller rockfish may be juveniles of some of the larger species we've been seeing. Wow, look at all these. ROV watch lead.
Go ahead.
How do you feel about trying to suction sample some of these little snails? The snails that are kind of eating the coral tissue.
In the coral.
Yeah, there are some on the ground as well, if those are easier. I'm seeing lots.
They're
the little
easier
pyramid
to
shaped.
find,
yeah. anywhere that you see them.
I think we can try.
I think we will try a collection if
All right.
we have some
we got
suction jars open.
We got some time as well.
Yeah, that's kind of what I was thinking. And since there's a little bit of confusion about what genus and species they might be, I thought we'd pause to collect maybe the ones on the floor, and then we could suck up a couple of little extras. What do you think?
Roger. So you want the ones on the ground as opposed to the ones in the coral?
Yeah, I think so. Even though I'd love to help the coral out a little bit, that's not my job.
Plus, you'll wind up with a bunch of bonus brittle stars.
That's right. That's right. I'm trying to make the echinoderm scientists as happy as we can. We've really benefited from their expertise and their identifications on this survey.
I have a great feel where I can set down here. I guess just get a toe in.
Well, good. We have a second vote for collecting some of these.
Do you want a suction while I drive?
Up to you.
Do you feel comfortable trying that?
Always.
All right, let's do that. Why don't you take the craft?
Hydraulics are up.
Is stopped at this time.
Copy. And would you also pull up the jar cam?
Yes.
I got that. 12.
Ooh, that's 12, that one.
And can we bring up the jar light?
Yes.
So I see one under the big coral there, two more on the left, one on the dead coral.
We're going for snails.
Yeah, a little white one right there, a couple more there.
Yeah, kind of take your pick, and then maybe we'll collect a little bit of extra
hydraulics are up.
Encrusting stuff around it.
Yeah, so we're going to start.
Arms hot.
Going to start with this one below the coral. Get lasers on.
Beautiful. That's the one I would pick too.
You guys are
Can we get a zoom on it before we start?
Sure, we can snap while they're bringing the arm out. Oop.
Nice. Confirmed. That is the target.
All right, let's come wide, please.
Then I can push the drawer out whenever we're ready.
Yeah, I'm ready. I'll back off a little bit.
Okay. Okay, going out with the drawer.
So again, these are possibly in the genus Calliostoma, but we've had another tentative ID of Akoya platinum.
Do you want to back in real quick?
Yeah.
So it'd be very helpful to get a sample to confirm.
That's good.
Can be a bit more precise when we talk about the distribution of these snails and what they do, what they eat, and the kinds of environments they're found in. Can help us gain a better understanding of the dynamics of these ecosystems. So these snails will make their way to the Smithsonian, where they can be accessed from people pretty much anywhere in the scientific community who are interested in borrowing them, and they can be further studied based on their morphology. So the physical characteristics we can see under the microscope.
So there's one just to the left of your nozzle.
And also for genetics, so we'll be taking a tissue sample
Yep.
and
So there's one
running
there,
them
and then
through the barcoding process, at least if there's not some kind of specialized application.
Okay, I've got a suction of
and we
300.
can compare them to other
and I'm
hold here.
going to go up to 500.
Specimens of the same species and different ones to see how closely they are related to each other.
Suctioning, I'm going to suction a little more. That's 1200 RPM.
There we went.
Okay, that looks like one.
There we go. We'll keep an eye on the jar and wait for.
Okay.
There it is.
And there it is.
One by the
Beautiful.
coral. Stand
yeah,
by
a
one.
couple would be great.
Standing by.
You square it up.
It's the one right underneath.
Yep. All right, I'm stable.
Okay.
Stable-ish.
Suction's active.
Did it go in?
I didn't see it.
It went in the funnel.
It did?
I thought it went in. Did it not?
I thought you pushed it down into the bottom.
Oh,
there it is.
There it is. Yes.
All right, we got two.
Beautiful.
Let's come around.
Maybe one more for good measure.
All right, there's a couple up here on top next to the coral that we can go for.
There's one
yeah, I see them.
One on a dead coral.
Or would you like the one on the dead coral, watch lead? Or does it matter?
Nope, doesn't matter to me.
All right.
Take your pick.
Coming up. Saddling left. Okay, I got a toe in. I'm stable. Think you can reach that. I'll bring my heading over. Center it up.
Do you want me to tilt the mini Zeus down a little bit to make sure we're not pulling it out too far?
Sure.
I guess we can see in the manip. There's the black tape.
Is this view okay, Sean? You want me to get you closer?
Oh, that's good. Sorry, I pushed
Coming up. Yep, coming up.
Me.
Yeah, I'm gonna come up a little. So, there we go. Okay. I'm stable. I thought I was stable. Okay, go ahead.
Okay, suction active.
Got him.
It's inside.
It's another one, right?
Yeah,
I think so. I think.
Three total now.
Yeah. Do we want more?
How's
No,
that,
I think that's perfect. Thank you.
All right.
Okay.
That's great.
Let me go back out into the water column.
The Smithsonian collections also thank you.
Okay.
Yeah, getting some
You guys can do
excitement
draw
from
when you're ready.
the science chat about that one.
Ready? Yeah?
Let me straighten up a little bit.
Okay.
Let's see. That looks a little crooked. How's that look?
So apparently,
Looks good.
at one
I'll go out
point,
slow.
Aquaria platinum
Andy,
was
did they
in the
also
genus
pass on to you not to
Calliostoma,
pass
so
over
they're all
jar
obviously very
two?
closely related or easy
too
to
late now?
confuse.
No, you did right.
I don't have control. Well, I guess I can
Squid
jog
off
it.
port.
Oh yeah.
All right, let's get this towed.
So I have sample ID
push it
EX2306 underscore D11 underscore 11B
That's good
in
there, Andy.
Okay. hold on.
canister 3.
And pull the jar in.
Nice job, everyone.
Yeah.
Beautiful
Fine. I
collection.
like it.
Thank you. Okay, so I think we'll continue to
Continuing
explore this area
Okay, arm is
and
off.
maybe
you,
with
Sean.
the lasers on when we can, just to kind
Lasers
of.
are on.
Are they? Wow.
I just turned them on.
Okay, I was like, I cannot find them in this.
Don't see the dots, but I see the.
I do
the
see the
shiny
line.
in the
Yes,
water.
yeah, I do too.
So it's some kelp down low stuck in a
Yes, I think
coral.
so.
There we
Yeah.
go, on the coral.
Beautiful.
The two laser dots.
There we go. Thanks.
I think we're ready for a move. They're probably done with their ship, their heading change.
Yes, they are. All right. Do you still like 270, 20 meters?
Yeah.
Let's do it.
I'm already there.
Could we do a snap on that kind of cucumbery, wormy looking thing under the coral?
Let's go ahead, video.
Right to the right of the lasers. Okay, another one of those sea cucumbers. I think it was Aposticopus, something like that.
Wow, yeah. That was the last one that was really long for a sea cucumber, I thought.
Mm-hmm.
I'm gonna.
I was just saying, don't forget to record all that, the sample.
Let me.
Are these any loose rocks over here to the left? Kind of, well, now coming into the middle.
Below those.
Yeah, I was definitely distracted. Thank you.
You're welcome.
Wondering if maybe we could grab that guy that's coming right into the middle of the lasers. I think the lasers.
That's about where you were.
Yeah, we can take a look.
There's also one that's now coming in towards center as well on those.
Yeah, that's possible.
On that little sediment chute.
Mm-hmm. They're not making it easy for you.
I got to see a few meters that.
Right. Cool.
Okay. All right. Yeah, I'm right above where we collected the snails for you, Nav.
Yeah, copy that.
It's right here on this outcropping. All right, watch lead. Sorry, we were doing some. Look at all these little baby
Aw,
squids.
cute. Oh, there's 3 of them.
I heard you're interested maybe in this rock.
Yeah, that one looks, I don't know. Yeah, as good as the other one. Maybe a little blockier and.
Okay, we can try to reach out and grab it.
I think it's, well, already I can tell it's bigger than I thought it was as we get closer with the lasers, but I think it'll be all right.
Might have a Primnoa stalk attached to it, which would be very cool.
Oh, now it's getting bigger and bigger.
It is. Well. What do you think?
It's a maybe as something that's not too big. I think it's all right.
What do you think, ROV team? Will this fit?
We can give it a. It should fit.
Yeah.
I think it's okay size-wise.
Okay.
Yeah. Then that looks like it'd be a good one.
You want me to pull the port wing inboard?
I think we need to go into port side.
Mm-hmm.
Yeah.
Yep.
Port side rock.
Okay, hydraulics coming on. We spun up.
Hydraulics are on.
Okay. It's loose. All right.
Wow, there's also a huge nudibranch on a dead coral skeleton behind. Kind of
All right,
really blends in with these corals.
Bring the box in.
Okay, coming in.
You might already know that. I just wanted to remind you. In case.
No, it was good.
So these samples will help us do a little bit of geology out here in the middle of the bay, where in the last mapping that I'm aware of that was done, the contacts are left hanging as soon as they got to water. But the geology obviously continued down into the water, and this will help us kind of bracket. There's kind of 2 choices of rocks. The rocks to the west of us, the islands to the west of us, and the rocks that are on them
Nudge it
are a little bit younger, Silurian and Ordovician. The rocks to the east are a little bit older and Cambrian to Proterozoic in age. And with some isotopic studies, oh yeah, look, we're going to get that stock, maybe.
Excellent. Could potentially do an aging study.
Right.
If we have the base of that coral.
And we can maybe tell the age of the rock. And in doing so, will tell us a little bit about the structures that separate the rock type that in between the islands on the east and the islands on the west of us.
Beautiful.
Very nice. Sample ID 2023. I'm sorry. EX2306 _ D11 _ 12G.
Port rock box.
And the port side rock box.
Watch leads, did I hear correctly? You just need the base of that coral?
That's correct,
that
yeah.
kind of dead one, so if these branches break off, that's all right?
Yeah.
Yep, doesn't bother me.
You need to kind of tuck it down a little bit then.
I understand.
Yeah.
Crunch.
And as I expected, we might lose.
Yeah, no, that's totally fine.
All right, perfect.
You never know, they might come up on the instrument tray later. I've had stranger things happen. It's another one of those Henricius, the white little sea star to the left with a red dot in the middle. So they're nice and easy to pick out. And I think we might have a different kind of
arm secure
sponge also behind where that rock was. It looks like it could be a Faria glass sponge of some kind.
Oh yeah, let's zoom in. Let's see what we can see.
That's
EX2306 D11-12G.
Oh, okay.
Frame it up.
Oh, great. Yeah, definitely a Faria of some kind. It's pretty sedimented. I think it's struggling a little bit here. But, yeah, one of the thin kind of branching tubes and lobes makes you think it's a Faria. There are several different species in this region. That's great. Thank you.
Yeah, once that move starts, you won't have too much time.
You see them often associated with the sponge reefs, although they don't have quite the same skeletal structure. They don't create quite the same enduring large reef-like structures that the Afrocallistidae do. But very cool to see. I think that's the first one in this area that we've noted. Wow,
uphill.
Just keep on going. Look at these.
Moves in.
Roger.
Oh, could we get a zoom on these little starfish on in the foreground here? That red one, starfish or a big brittle star? Kind of can't tell.
Thank you, co-pilot.
We had a request for a zoom earlier, and I think we got distracted chasing that chimera. So this is a great opportunity. Yeah, I have no regrets.
Oh, that's pretty. Have we seen one of these before?
I don't think so. No. I wonder if Chris has any thoughts.
That's pretty.
Yeah, just the interlocking those kind of big scales that make up the dermal layer of this. Okay, we have an ID of Stegophora nodosa. So I think if we know what it is, it's not a sample request, but some really nice in situ imagery is always really helpful. You can get all the way out to the tips.
I think I see some hexagons. Okay.
Apparently they feel like cookies. Which is helpful information to have.
Interesting.
I bet they don't taste like them.
I have another second here. I want get just a hair closer.
Yeah, you got plenty of time.
So it looks like we've got a little bit of damage to the arm that's at the pointing at 12 o'clock.
I know we'll lose our light, but
In situ video is very useful. Chris has always wondered what they do. And now we have a bit of an idea associated with this soft sediment and staying very still. Wow, look at the texture on this. That's I call it the central disc. You know, that round part in the middle. It's just beautiful.
And
yeah, lots of little, polygons, pentagons, hexagons, kind of making up the geometry that you see.
Well, let me know as soon as you got this, because I'll need to move on.
Clear.
Okay.
That's wonderful, thanks.
And before we zoom too far out, I'm kind of interested in the rock that's up and to the left. So we need to back out a little bit.
Science, this is on your science channel. That snail on the left edge of the screen. If you look back at the video later, that actually jumped and started swimming in the water column and then fell back down there.
Oh my.
Oh.
All right,
okay.
See if we can see this rock.
Apparently we missed a swimming snail. That is cool.
Dark looking thing.
You'd like that sample or just a look?
Just a zoom, a quick zoom.
Okay.
It's a creature walking on it.
Looks like a spider.
Does look like a sea spider, a pycnogonid.
Coming back.
Different species from
Go ahead, you can
the
take it.
Colossendius, which I practiced pronouncing a little bit after trying to before. But a different species, but definitely a sea spider. And maybe...
Alrighty.
Is that good for you, Watchley?
That is good.
All right, let's come on. Time for me to push forward.
Right.
It almost looked like a piece of a bamboo coral skeleton next to it as well. But I haven't seen any on this dive yet, so it would be hard to say. Wow, look at these. There's even more. Just giant. Whoa, okay, the view from Sirius. Everybody should definitely go over to stream number 2 to check it out. You can just see beautiful imagery of D2 approaching this large field of Primnoa pacifica, these really large tree-shaped corals. Just kind of a dense forest of them. Lots of different associated animals, rockfish, sea stars, nudibranchs, a rockfish in the coral right there. Big anemone. Look at that. So we've seen a couple of fish now that are just kind of nestled into the coral, either resting or, using...
That is really good camouflage.
A little bit of camouflage and protection, yes. Yeah, I'd love a snap zoom on this. A sponge.
Snap ID.
Bit of a different one. Let's see. Ooh, a different kind of Feria. This one looks like Feria oca to me. It's kind of the largest species in this group. So it's kind of easier to pick out. And maybe another sea spider inside or something inside. Very cool. Oh, wow. And a really buried, it's just been covered in this sediment, a Primnoa that's been knocked over. Wow, lots of Liponema, these big pom-pom anemones just everywhere all of a sudden. And a couple of those Balticina sea pens in the foreground. And just, I love seeing that imagery of from Sirius looking down on D2 as it's exploring this really beautiful seascape. I think all of the coral scientists that have been following along and will probably be reviewing this video in the next 24 hours will not be disappointed with this dive. We've seen, you know, fields of these primnoas.
Pilots, you want to move?
Yeah,
keep it moving.
You're looking.
Yeah, I think based on all of those environmental factors I talked about, and you know, kind of extrapolating those
like
from the different kinds of catches that we've recorded across the Gulf of Alaska, predicted this area as a hotspot, and we didn't have any actual evidence until now that it's proved to be pretty accurate. You know, we've yeah, this is definitely an area with an incredibly high abundance of Primnoa pacifica and lots of other things, but
it pays to explore. Huh?
It sure does, yeah. It's very helpful that I know we had to change our plans, and this was kind of a backup, but I couldn't be happier with this plan B. You know, we ended up seeing a hugely abundant coral area that I'm sure scientists will want to keep an eye on in some way going forward. You know, okay. Just getting a little bit mesmerized by this ctenophore that's in front of us. But this is why, you know, we do the kind of exploration that we do. You know, you can sit with your algorithm all you want and use these predictions, but to actually be able to see and, you know, yeah, count and measure all of these individual corals is something else. It, you know, if you were ever looking for a study site to study primnoas, this incredibly important habitat forming coral, you couldn't ask for a better one than the one that we just discovered today.
Yeah, this is just kind of taking it to where you need to take it. Models are great.
What do we think?
Insofar as what you can do with them, but in the end, you really need to be able to validate them and see how useful they are, so you can move forward from there.
Yeah.
Do you want to look north again? What do you think after this?
Yeah, so you could, you know, the area that the model covered was the entire Gulf of Alaska and reaching out into this into these little channels a bit. So you could never visualize, you know, you could only ever do a very small fraction of the amount of ground that covered. You could never visit at all the amount of, you know, people and time and energy and equipment that go into a dive like this is pretty astounding.
Sure.
So you have to kind of find ways of narrowing it down, and the models do that.
Well, that's why the models are so valuable, for sure. They do. But that's why, at the same time, it's important to know that they're working right.
Yeah, and this one was, you know, pretty much spot on.
Okay, I'll start cheating over.
Yeah. So I think some of the authors that did all of that work in 2017 will be very interested to see this kind of come to life. And also interesting where we didn't see them, but instead saw a few other things, like those really abundant black corals from the first dive that we did in this area.
I don't know what that is.
Yeah, maybe a piece of wood. That's my best guess, like a little plank of some kind.
Someone turned bridge down to -30 on mine.
There's some nice looking boulders for you.
Deselecting bridge.
Yeah.
Wow. Oh, maybe.
Yeah, we're full though.
Yeah, we're full.
I think so.
Can we zoom in on that sea star up there?
Just
the one straight ahead at the top.
Straight ahead?
Yeah, those two.
Lasers are on? Okay. Roger?
They're just cute.
Okay.
I mean, they're viciously attacking that really nice looking coral, but...
In hyper slow motion, but...
Yeah. Look at those.
Okay, pilot, I'm looking north, and my rotator's at 45 degrees.
Roger.
That's great. Just wanted a quick zoom while we were on the way. Thank you.
Right,
wow.
So
wow.
Really neat little boulder crop.
Yeah.
A little lighter start.
There's another fish in that one, maybe, or 3 nudibranchs for sure, and then maybe a little fish at the top.
Sounds good.
Oh, 4 nudibranchs. There's one near the base.
Oh my!
And is that a fish? Could we snap on that? Yeah. So think a little juvenile rockfish. Just beautiful. That's amazing. So again, even this this coral that's being heavily predated on by nudibranchs and probably other things has a little juvenile rockfish nestled into the branches. Really beautiful to see. Just to document, you know, when people wonder why we study corals and why we're, in some cases, setting up protected areas around them. You can imagine how vulnerable these would be to certain kinds of activities. So it's always really amazing to see them in real life.
And so as we were discussing with the biology, this is kind of exploration is a great example of what we can see with the geology as well. We're down in a bay, as I mentioned, in a place where people didn't try to map the geology back in the day. All the geologic contacts more or less ended at the shoreline. We know we had different kind of rocks on the nearest shores that are to our east and to our west, without really any idea of where the boundary between those two units are or is. And we've picked up a couple rocks here that will help us kind of settle that question. And it's something you can't do. Until you get down here, and until today we didn't even have the baseline information, which would be the mapping that would allow us to even get in here and explore.
Keep moving.
So it's really a neat example in a encapsulated way of like how you can make that push forward. Collecting both the mapping data and then immediately getting down and getting the hands-on look and sampling to get some new information.
On top here, a lot of jagged edges here.
Okay. Rotating over slightly.
Yeah, it's pretty amazing to be able to do all of that, you know, over the course of 24 hours, filling in all of those gaps in the map. Looking at the species distribution predictions and picking kind of drawing an imaginary line and then going down and doing it. It's been pretty amazing.
Now we've come up and reached another pinnacle.
Wow.
Nice little, even kind of a spire.
Yeah.
You can drive
some of these overhangs, it's hard to imagine what's keeping them up. Look at that.
Yeah, I think it just kind of keeps going.
Yeah, you can imagine that there was a boulder-sized chunk of rock in there that's dropped away, perhaps during some earthquake.
Right. Oh, look at this.
I mentioned the Queen Charlotte Fault has had 7 magnitude 7 earthquakes or greater in the last 100 years. The 2 most recent were in 2012 and 2013.
Lasers on the fish.
Oh, okay.
Lasers are on.
Just a bit before I moved to the West Coast. Wow, look at that huge rockfish. So this has to be one of the older ones. Okay, might even be gravid, I don't know. Definitely have to take a look and make a note of the video. It looks like it might be. And I know for rockfish, I'm pretty sure that the older they get, the more young that they have.
And it looks like it's
so I think they, yeah, if you're
Sorry, I thought the ship was
kind of looking at a larger, older rockfish, they are the more reproductively active ones than the
Sounds good.
kind of mid-sized ones that you would think from other fish species.
Looks like you found another cave similar to that one we stumbled upon before.
Some really big dead bases of some large primnoas here.
Bring
Not
the
a
lasers
lot
back
of,
on. Thank you, Byron.
I think, you know, kind of the remnants of larger species versus small ones recruiting. More of that sponge that we sampled earlier.
There's comment.
I think those are a little patch
Look
of
at that
zoanthid
patch right there
anemones.
between the lasers.
Go ahead.
Just a snap will be enough, I think.
Yep.
Oh, neat.
Yeah, definitely. So again, they're like the big solitary anemones we see, but they're connected at the base, so they're a colonial anemone. And, you know, they have sort of a long, thin, we call it a stolon in corals. I don't know what it's called for zoanthid anemones, but a piece of connective tissue that kind of encrusts over these rocks and connects all of those individual zooids.
Thud.
Wow.
Lights
Look at that.
60%.
Wow.
Several different kinds of rockfish, some smaller looking individuals, so maybe juveniles of some of the larger species we've seen, like that one.
You found him. Or her.
Wow.
Really?
Huge.
Really interesting the way somebody stacked these rocks in here.
Uh-huh. Okay, a really curious rockfish as well. Somebody noted the black spots. I know some of them naturally have black spots on them, but some others maybe, you know, some kind of scarring or damage. And that one looked like it might have been gravid, so gestating. Watch leads here. Maybe through the, well, how are we doing for pinpointing the scattering layer on these?
I don't know. I'll have a look and see if we can spot it.
Okay, stand by and we'll look into it.
See if I can spot it.
Okay, I wanted to double check before I talked about this, but I did think, and now I've just kind of confirmed that all species of the genus of rockfish that we're seeing, Sebastes, are viviparous. That means that they give birth to live young instead of, you know, laying eggs and leaving them to hatch. So they'll give birth to thousands or millions of tiny larvae. Very cool. Just want to make sure I get my fish facts straight before I mention them. Looks like that Hippasteria kind of is taking advantage of this
Looks like
really large primnoa, or possibly had something to do with it.
He's
this branch
tackled
toppling over. Wow, that one's huge. But there's, you know, no shortage of primnoa growing here. You know, with these really large colonies. Another one of those sea cucumbers. Some smaller rockfish.
Yeah, about 8 minutes left.
What do we think about the Niskin? Did we? It's kind of hard to say. Maybe we do take one on the bottom.
This isn't a call I can make. I don't see anything that jumps out at me.
Okay, ROV, will request a Niskin while we're here.
Roger that.
Thank you. Get some really good eDNA data on a very unique and dense population of primnoa and the kinds of associated fauna that we see in the area.
Okay, when you guys are ready, I'm ready for the Niskin.
Navigator's ready.
All right, resetting and firing in 3, 2, 1. Good fire.
And 314 meters depth. Sample ID EX2306-D11-1113W.
Wow.
Right, Pat, I'll put that move in
I don't think I need to request a zoom. Let's take a look while it's still.
Go ahead, video.
Excellent opportunity. I've never seen one just kind of hanging out on the sea floor like this.
Oh, fantastic.
That is very cool.
I'm going to slowly try to creep in and get you stationary.
Yeah, did I see it change color?
Yeah, it was a little lighter when we first spotted it, I think.
Yeah, because we've had a question asking if it was alive. Because yeah, I've never ever seen one that's just kind of hanging out like this on the sea floor, but I definitely see it moving now.
All right, go ahead, video unstable.
Wow. Oh, this is an opportunity
you can take full zoom or whatever you want.
To get you know, a nice zoom and imagery of this squid that almost never stay still like this.
How's your lighting? Do you want
you
some
only ever
fogs?
see them really quickly darting through the water column.
We're at 60 right now. I can bump it up.
And you can see all the little, I think they're chromatophores just pulsing. Look at those dots on the mantle and on the skin of this squid. You can see them kind of
okay. We'll go back
pulsing different colors. Just stunning. So other families sit on the bottom, but I'm glad Michael is.
That might be it.
Whoa,
and there he goes.
Wow.
That was neat.
So a gonadid squid of some kind. That's very cool. So also, we have Gonatopsis
Looks like I have a little more
borealis.
tether now to
Gonatopsis
you
borealis. Yep, I guess it is alive. There's a little bit of ink left behind as well. Wow.
There he is again.
About 5 minutes.
Okay. So very capable of swimming very fast. Just taking a little break. Doesn't sound like any of the scientists in our science chat have seen that behavior before, of a gonadid squid just kind of resting on the sea floor.
Press down.
Wow, it just doesn't end with this dive. Look at all the all of the things that we've seen. It's just checking out.
That's looking way down.
Really nice view of the
we've lost
marine snow. You're kind of taking a look ahead.
It is off to port. It's still that ridge line. But it
just seeing if we could get to the bottom of this feature, but we're kind of pointed the wrong way.
We don't have much time on bottom anyway, right?
4 minutes.
I'm happy to spend it looking at
looking
at this feature.
At this area.
Sounds good. I'll just let it ride.
Yep. I'll just cheat off to co-pilot support.
Okay.
If you want to turn or anything, you can.
I'll turn and I'm fading out as much as I can. Get a little bit closer to you. We got a pretty big delta right now, but setup looks good.
Wow, quite a few shrimp and arrow worms.
Let's bring the fogs back down.
Going back down. That's secured.
Can we get the lasers back on?
Lasers are on.
Thank you.
There's those yellow.
Wow. Oh yeah. Could we get a quick zoom on those again? We saw them before, and I still don't know what they are.
Roger. All right, video. Let's go for a zoom. You might want to bring the fogs up just a hair for this.
Would we have time to attempt a sample?
I think so. Do we have a jar open?
I think so.
And you have a canister.
I think we have C4.
All right, let's come wide then.
Okay.
And I heard ship is stopped.
Ship is all stopped.
Roger.
And so jar 5.
So I think, yeah.
Both jar 4 and 5 are open.
You know, we've passed by these little unidentified sort of stacked discs. I think they could be eggs from something, but I really don't know. And it looks like there are a couple of brittle stars nearby that might get opportunistically sampled as well.
All right, packs coming on.
Safely,
HPU
of course. But
coming on. HPU's up.
Roger. Let's get HD2 up. All right, I can get it.
Arm is on. 3, 2, 1. Arms live.
2. Okay.
Okay, ready with the drawer. Do you want me to put HD2 on the minute?
Yeah, I would like that. Imagine Sean wants it too.
Sure, yeah.
Okay, we're gonna go for a suction, so let's get the drawer.
Okay. Ready, I'm pushing out now.
Okay, I'm backing off.
I think we have to call these unidentified and maybe rare taxa. Yeah.
That's good there. I can grab that.
All right, hold in there.
Use Zeus down a little bit.
Zeus coming down. Yep.
Ah.
Okay, you can drawer in.
Okay, drawer coming in. It's fully retracted.
Kind of just straight ahead, behind your
Going
hose.
What am I going for?
Those objects right there, where your nozzle are. Those yellow objects. All right.
Yeah, kind of little
Stabilizing.
stacked discs.
I'm inching you in slowly as well. Okay. Holding here.
Okay, engaging suction.
Pushing back in and up.
I see one in the jar.
There's 2. Good job, Sean.
That's great, actually. We don't need all of them.
Okay.
Yeah.
Sounds good.
Okay, suction
Perfect.
secured.
Okay, backing off.
Sample ID EX2306-D11-14B in suction sampler canister 4.
Happy with that alignment, Sean?
Give me one sec. Yeah, let's go for it.
Okay, push and drawer out.
That's good there.
Holding.
Okay.
Tracting drawer.
Wow, some really big Hippasteria sea stars.
All right, arm is off.
Thanks for that. Very excited to find out what those are.
That's the dive.
That's the dive.
HPU secured.
Whew.
Nice job.
Wow, what a dive. This has been wild. For somebody that studies corals,
Andy,
this
I'm
has
gonna come
been
off the wall.
astounding.
I'm going to line up.
You know,
my
we
tail
kind of
towards
started
you.
off the dive with
Clear view
that
for swing arms.
sort of sparse sightings of corals, and then at some point we just hit the right depth and we hit the right conditions, and we have found an incredible abundance of Primnoa pacifica, forests of them basically, some incredible imagery, incredible opportunity to ground truth this model that I was given access to.
I can do the lowers.
And yeah, I couldn't be happier. I think we'll be studying this dive for quite some time.
Yeah, yeah, and I think there was a nice contrast between the early in the dive, the steeper, cliffier parts, where there was certainly a contrast in what we saw once we got to the top, and so possibly that's information that would go into improving the models a little bit.
Yeah, definitely.
If they get, if slope is not already part of it, then it's certainly a little piece that they can think about is going forward. So yeah, kind of neat. And we also just got some nice samples for some just geology ground truthing, and making a little progress with the seafloor geologic map. So
if I just come to starboard,
all in all, a good day.
Just have to do a half turn, right?
I'd like to extend extra special thanks to the GFOE team, the ROV crew, and the video crew for the excellent sampling and imagery we were able to collect today.
All right, so you want me to turn?
Thanks also to the scientists that tuned in on Labor Day, no less, to help us with the identifications and advise us on collecting. We always appreciate your observation and expertise. So thanks so much for joining us.
Ready for that.
I'm turning now.
I'll send a preemptive thanks to the lab crew, everyone that's going to be working in the wet lab a little bit later today than the past couple of days, going over and meticulously cataloging and processing these samples. And of course, NOAA Ocean Exploration and the Okeanos Explorer for the platform and for making this all come together and happen.
I'm starting to come up.
So I'd give everyone a preview of what we were looking for tomorrow, but I think it's very, very weather dependent.
Yeah.
So you'll have to stay tuned to the social media and to our emails if you're on the shore-based science team for an update as we decide where to go. I think the weather's opened up a little bit back in the Gulf of Alaska, so we might be back on to our close to our original plan. So stay tuned for that information.
Yeah.
Thanks everyone for watching.
Yeah. So, all right. Yeah. Thanks everybody for watching, and tomorrow I'm interested in finding out where we go tomorrow too. Yeah. So I know we'll be back in the open sea. The question is, we're actually still dancing around some coming storms later in the week, and so we'll probably have a big meeting tonight and kind of work out ways forward.
Right. After the lab. After things are sorted, hopefully.
Right.
Or
Looks
during.
good
We'll
from
see.
D2's end, and looks clear from Sirius end.
So till tomorrow, yeah. Thanks
deal.
everybody for joining us, and
All right, let's come up to speed then.
we'll see you next time.
I'm selecting ascent.
Hauling in. I'm at 30 meters a minute.
Roger. Delta 5, put a little bit forward. Tether looks great. Getting the arms stowed.
Pilots, you good if I have them start coming decimal 5 through the water? Should push us forward relative to what we're doing now.
Yep.
Sounds good. I'll take auto sway out next, but it shouldn't affect anything from what I'm seeing.
Yep.
Pilots watch streaming as we start coming out.
I secured HPU.
Did. I got him a squid.
Serious gain has been returned to 0.
Ship moving?
Yeah. All right. We're making forward,
Starting
right?
to.
Yeah. So let's see. I kept swaying it this time, so it looks like we have slight positive starboard. So if we bring that out, it's going to push us this way, right? So I should leave it in for now. Do you agree?
Starboard.
Right, we positive starboard way. So if we go off that, we would go this a little bit.
Starboard means the water's coming this way. So if we come off, we should go to port, right? Or do I have my signs reversed?
Or do I have my signs reversed?
So like forward, positive means the water's coming.
So if we stop forward, we would drift back. If we stop going starboard, we would drift this way, right?
Why can't I make up this delta? I'm at 93. It must be. They're moving. Although I'm still... no, I'm moving. I'm going to take off my forward way. It'll help my verts. Roger.
At this time, backgrounds are going to come off.
Sean, those samples, double great.
Yeah, that was awesome. I think that was a great way to do it. It's probably hard for you to see that monitor though. I was trying to. I was watching. Yeah.
I was glancing up at Sirius because I was kind of piloting without having to worry about the arm. Well, when you were going in for the sample, I had to focus on the arm, but while you guys were doing like draw in, draw out, I was watching all the conditions.
It was way faster than we would have been able to do it otherwise.
The conditions just didn't allow it today on either of them, the snails or this last one. Well, this last one was partially because we needed to do it quickly, but there weren't great holds for it either.
Yep, thank you, Nav. Okay, holding at 50 meters.
Good catch.
Yes.
I think, yeah.
This time we request to disengage auto sway. Once that super, I have got it, a little bit that bridge. Yeah, bridge, correct. Yes, at this time please disengage sway. Just one set there, so sorry. Hey, Dive Super, this is your navigator. I'm ready.
Dive Super Pilot, loud and clear.
Dive Super, go. Yes, vehicles are just about 50 meters. They're holding. Let's see, we've got 5 to 6 knots of wind just off the profile of the ship. Ship's doing a heading change to 175. And then current is very light. You see investment 1, because it's so light, you have to be variable, so we now have direction for surface current. And we are making about 175 through the water, water surge and sway, disengage, just times, put throttle to rudder, disengage.
Permission to continue with the recovery?
Yes, you do.
Pilot, dive super.
Dive super, pilot.
Is the control room ready to continue with the recovery?
Control room's ready.
Sounds good. Let's go ahead and ascend up to surface, 15 meters per minute, calling out every 10.
Roger.
Ascending. .40 meters, wire out.
T245.
.30 meters, wire out.
D232. All's well.
.20 meters, wire out.
D223.
.10 meters, wire out.
D215. All's well.
All right, pilot, we have good eyes on.
Video.
Streaming directly aft.
Pilot has visual.
Good copy. Let's go ahead and lock in auto heading in versus ship.
Auto head in.
Okay, Deck, let's go ahead and recover Sirius.
Did you click on surface, Nav?
Yes, vehicles are on surface.
Okay, pilot, target lines are on. Please provide some mass thrust as Sirius comes up and over the side.
Roger, dive suit.
I just turned off your CTD pilot.
All right, pilot, that's looking good. Let's maintain.
Pilot's 40 back.
It's looking good, pilot. Let's maintain.
Backing 30%. Pilot 20%.
Good copy, pilot. Winch, let's take back local control. Check A-frame.
Remote disabled.
Sirius cameras clear.
All right, pilot off stick at this time.
Pilot off stick.
Go ahead and leave auto head engaged.
Auto head in till halfway down.
Super nice.
Okay, pilot, let's disengage auto head at this time.
Auto head out.
Pilot off stick.
Thrust 0.
Okay, pilot, recovery line's clear, daisy chain's free. Okay, pilot. Lift line's going tight. Let's go ahead and lock in auto heading, inverse the ship, start backing towards crane.
Auto head inverse, backing.
Give us a slight bump to surface, pilot.
Bump up.
Let's ease your back a little, pilot.
Easing.
All right, pilot, you can pick up speed back again.
Picking up speed, bump to surface.
Good copy, straight back.
Straight back. 40%.
Looking good, pilot. Let's maintain.
Maintaining. Bump to surface. Bump.
Good copy, pilot. Keep coming straight back.
Coming straight back, about 60%. Bumps up.
Good copy, pilot. Keep coming back, almost under the crane.
Back in 60.
Give us a little port lap.
Come in port.
Copy, thank you. Looking good, pilot. Just port now.
Port only.
And let's ease on that port.
Easing.
And you're under the crane.
Backing.
Let's give us a small twist to starboard.
Twist to starboard.
And pilot off stick.
Pilot's off stick.
Pilot, secure power.
Deck, E-stop. Winch, knife switch.
Knife switch.
Voltage reads 0.
Vehicle's cold. Pilot, dive super.
Dive super pilot.
Trolley straps are going on. Feel free to wrap up in the control room and come on down for post dives.
Roger, dive super.
Good recovery. Winch, dive super.
Go for winch.
Feel free to secure the HPU at this time. Come on up, help with post dives, and please hold off on turning on the fans until after we're done.
Good copy all.